By the way, we should also be careful about timing because the way the webinars are set up is that by like 15 minutes before the end of the hour, the next webinar starts, and then they will kick us out automatically because they will use the same account. When the next webinar starts, our webinar will terminate automatically.
So the speaker has 30 minutes slot, no?
We should quit by the 50 minutes before the
Hi,
hour.
Simon.
Hello.
Hi, Simon. Hello.
Hi.
How
Can
are
you hear me?
you? Yes,
meet
very
you.
well. Thank you.
Cool.
Thank you for saying, for accepting the invitation and everything. Thank you.
Yeah. Well, thanks for having me. This is really, it's kind of my first time with this neuro match, but it seems very exciting.
Yeah, it's also my first time.
Uh-huh.
So you were saying, sorry, you were saying that he was telling me about the timing, that we have to be really, really on time because they will kick us out. You know, they use the same account and then we are out from the Zoom.
I see.
So you have 30 minutes for talking and 15 questions, no? Is that the case?
I think yes, right? No?
Mm-hmm. Right, so at the start of the hour, then we should be out maximum by 15 minutes before the next hour. So 45 minutes. The whole session should...
So Simon, if you can be like 30 minutes or so, and then, you know, even if there is...
There will be Q&As as well, so we will also
yes.
questions.
So, but you were already expecting this, no, Simon?
Yes. No, I think I calculated for about 30 minutes. Depending on the level of nervosity, maybe 31. But
okay.
I still, you know, I do this now for a few times, but I think it's still strange, right? You're talking to your computer, you don't see anything.
It's very weird. It's very weird. Sure.
By the way, will you be sharing screen?
Yes.
Maybe we can test that before.
Right. Let me just...
Perfect. Yay, it's working. So I'll go off screen throughout the webinar. And if you need me, technical problems, I'll be here and I'll be monitoring the chat as well. If there are some disturbing situations, I'll be handling the chat. But other than that, your MC, Dr. Bendito, will run the show.
Sure.
Okay.
So you tell us when we have to be... when we are online or when we have to start.
Right. In 7 minutes, I'll push the button and we'll go live. Okay.
All right. Good. I just make sure this PowerPoint works.
Yeah, you have to. Yeah. How are you, Simon?
Oh, it's okay, but it's actually a bit frightening what's happening right now across Europe. You know, we were supposed to be in around Basel. You know, Denis wanted to organize this.
Yeah.
I think this event, right? It's actually very close where I grew up, and I was looking forward to. So I was hoping, oh, that's great. You know, meet all you guys.
Yeah,
but now it's crazy.
We just need to, you know, change our mind of how things were done before, and you know, and adapt to this new situation. I mean.
Yeah. But how is in Alicante? I mean,
in Alicante is good. We don't have, you know, we have this map of Spain with colors. Green is that you are more or less okay. Red is that you are. So we are green so far. So Alicante is good, but even within Alicante, the you know, the province, you can now start to see, you know, small patches of red lights. So yeah, I don't know.
I'm always wondering. I mean, it seems too early. You know, if it would be super cold and winter, I would see it. But now it's still okay, right, weather-wise, and it's just exploding. I think for even Austrian and Swiss circumstance, it's crazy. I mean, I think Switzerland has the highest rate in exponential growth right now. Yeah. And,
wow.
Yeah,
I didn't know that. I didn't know that. That was like this actually.
Right. And in Austria, we also now we are, I think, close to red, so orange red status already. Many areas, yeah. So it's it's stupid because I mean the winter is only coming. So what happened in winter, right? I mean, and how long is this gonna be, right? This is the most annoying part.
It's crazy because we have already been like this for 8 months, 9 months already.
Right.
So I don't know. It doesn't look like it's gonna get better in the next few months. And
How is school? So for you it works, or
so for us in Spain, or I mean for sure here, is working very well, very well. So the majority, only 1% of the of the people that gets it is from the schools. I mean the majority is coming from the from meetings with friends and at home, and also they cut from yesterday. They cut the opening hours of restaurants and so on, so they close at 11, and we cannot be on the streets until 6 in the morning. So you have to be at home.
it's funny, they don't call it lockdown, but maybe slowdown or something. But in the end, we are going back to where we have been, right? So it's
yeah. And probably at the same time, we will be around February, March.
Yeah.
So hopefully the vaccine will come at some point.
Right. Well, hopefully soon. I mean, I hope this is the only thing that could, help. Otherwise, we're going to go up and down, right? I mean...
Yeah, we will be like this. Yeah. I don't know for how long.
Yeah.
Maybe forever.
But how is the science going for you?
It's going well. It's slowly, because we have been slowed down.
Right.
A lot, 40% of my mice were sacrificed during springtime because the animal house could not cope with all the, people were getting sick and people could not, change the cages and so on. So basic stuff. And but yeah, science-wise, I mean, we have very nice results, projects, paper comings and so on, but it's crazy.
Yeah. Yeah, no, I have people that should go in a job market, but I mean, I don't know how they can do it, right? So.
By the way, if you have some people that may want to come to Spain to my lab to do a postdoc, I have opening positions. So.
Yeah, I saw you got this wonderful grant, right? So
just keep it in mind. Keep it in mind. You know someone or.
I just had 2 graduates. One is getting a baby now, so she probably is not. And the other one got a job in industry. But I have one more graduate soon, maybe next year. I will tell him.
Sure. And we have to think about what to do with the Axon meeting, eh?
Oh, yeah. I think the enthusiasm for virtual format is not that big with, who said it, Alain or Rüdiger?
Yeah, but I think we should do it online. Yeah. It's not a big thing. Anyway, I think we have to start this thing as soon as
Yes, I'll start in 30 seconds if you're ready.
Okay, yeah. Are you ready, Simon?
Okay, I hope.
I don't have to do too much. I mean, it's you.
Yeah.
Okay, I'm pushing it. Good luck. Have fun.
Okay, I hope we are online. Good afternoon. I am Guillermina López Bendito from the Institute of Neuroscience in Alicante. And it's my pleasure to be joining this NeuroMatch meeting for the first time for me. And also it's a real pleasure to have Simon Hypenmeyer as a speaker today. And Simon and I met a few years ago, and we are good colleagues and so on. And so Simon, since I think 2019, 1 year ago or something, got the full professor position at the IST Austria. But before that, he was also in the IST for a few years as an assistant professor. And Simon is a very well-known worldwide as I would say one of the fathers of this technique, Madam technique for labeling lineage tracing of neurons and cells in the brain. And this is a technique that he used widely and to show how the cell diversity occurs in the cortex and other brain regions, and in which he had been using for a long time. So just a little bit of a background for the ones that don't know Simon. So Simon did the PhD with Sylvia Harber, and then he moved to Stanford to do a postdoc with Li Chun Lu in several years. I think 5 or 6 years he was there, 6 years maybe. And then he went back, as I said, to IST Austria, and he got also one of these prestigious ERC grants, and he had been working in very nice projects, so on, regarding the molecular mechanisms for neural stem cell production and lineage progression. Also, he has been working in genomic imprinting that maybe he will be telling us today in the cerebral cortex. And I think he has also future plans for looking at lineage progression during evolution, I think. So thank you, Simon, for accepting the invitation to talk in the NeuroMatch. And thank you, everybody, for connecting. And I hope you will enjoy Simon's talk. So thank you.
Okay, thank you, Guillermina. I'm going to share my screen. Okay, I hope you can see this now. I'm not seeing anyone anymore, but thank you very much, Guillermina, and also all the organizers for this wonderful initiative, this NeuroMatch for us developmental biologists or neuroscientists is actually, I think, the first time happening. But I really think in these days, this is a wonderful initiative. So thank you for the invitation also to speak here. Okay, so the main question, or one of the bigger questions we are addressing currently in the lab, is really how to produce a brain with correct size and how to generate all the different cell types that you find there. And we currently focus on the cerebral cortex, and in particular, we are using mass genetics to address this question. So already a long time ago, Ramón y Cajal, of course, noticed that not all the cells in the cerebral cortex are the same. And today we know that there's actually specific factors or transcription factors or even transcriptome signatures that define the different cell types. This is now an image from the mouse. So you can see the laminated organization, which is actually, yes, evolutionarily conserved of these different cell types. And importantly, the laminar position dictates the wiring of these cells. And we do know if something goes wrong in the production of not the right number or the different cell types, basically due to genetic mutation, that this can lead to neurodevelopmental disorders such as schizophrenia and autism. Or in much more extreme cases, it can really be the mutation of a single gene that leads to a drastic disease, microcephaly with a smaller brain compared to normal, or lissencephaly with complete disorganized laminar structure, and the folds are also very much not there. So there is a huge sensitivity in the process to produce all the right cell types and the right amount of cells. Now, while in most of these diseases, the etiology is actually not so clear, the nature of genetic mutations is becoming clear increasingly. And we believe that this can give us maybe some insights, but also may imply some general principles. Now, in our lab, we take this as an inspiration and are really interested in how neurostem cells during development are able to produce all the different cell types here in the distinct layers in the cerebral cortex. And as Guillaume also already mentioned, we're using mainly quantitative lineage tracing and genetic manipulation at single cell level in mice. And the prime methodology that we are using is called mosaic analysis with double markers. Again, this is a genetic method in a mouse. And importantly, what MADAM, this technology can really do is it can capture progenitor stem cell division patterns at single cell level. And this is really a method that I got inspired when I was in Stanford, a postdoc, and that was originally developed by Huisson in the laboratory of Lichun Lo. Now, here is really the principle how MADAM can work. So you're starting with a cell that is not labeled, but then you induce this MADAM event. I will not go into technical details. I mean, it's published quite a while and there's literature you can read. But the important thing is that the two sister cells express two different markers, one's red, one's green. And depending on the further proliferation pattern of these sister lineages, either more symmetrically or asymmetrically, you can really infer the cell division patterns with high accuracy. Now, on top of it, we also can introduce genetic mutations of interest, specifically in one of the lineages, here in the green lineage, and then compare the effect at single cell level of that mutation when compared to wild type. And for many years, actually, we're working towards a resource that we now completed. It's actually here in bioRxiv by Contreras et al. That allows more than 96% of all mouse genes to basically subject to such an assay. Now I show you one clone here at higher resolution, and this is really what it is. So basically, all the cells that are labeled here is the result on the output of a single stem cell, which we induced at this MADAM event at embryonic day E11 and analyzed at 3 weeks postnatally. You can appreciate the quantitative nature of the approach. You can go and count the different labeled cells here in the different layers. There's also glial cells, not only projection neurons. And over the last years, we really have analyzed hundreds of these clones with the aim to see whether we can get to a general framework of lineage progression in cerebral cortex. And this is the result, the summary, basically, what we came up with. So we have now an initial phase of neural stem cell division patterns. So this is symmetric divisions before the radial glia progenitors then start to produce neurons of all the different layers by asymmetric division. Now, the intriguing part about this finding, at least to me, is that once these radial glia cells start producing neurons, we can predict the overall output, which is about 8 to 9 neurons in total that then assemble here or distribute over these different layers. Now, not only that, also about 1 in 6 can be predicted to retain the potential to produce glial cells such as astrocytes and oligodendrocytes. Now, while there's many unknowns, of course, in the framework here, we are now really interested in the molecular and cellular mechanisms that drive the lineage progression or the switch from symmetric to asymmetric, the sequential production of neurons, and then the switch to glia production. And today, actually, I will tell you 2 brand new stories. So unfortunately not imprinting as Guillermina was maybe expecting, I'm sorry, but I really want to present recent data that we got in the lab. But basically, the inspiration for the first project I'm going to tell you is really that the laboratory of Denis Chabaudon in Geneva recently found in a very beautiful paper here, published about a year ago, is that there's a temporal patterning of radial glia progenitor cells as they produce the neurons. So in other words, they isolated with single cell resolution the transcriptomes at every step of neuron production here, and they could identify intriguing fingerprints that seem to pattern the stem cells when they produce the neurons. Now, we have then carried out a candidate gene approach where we looked at EED, or which is part of the PRC2 complex, and found actually that lineage progression in such a mutant is affected. In other words, you anticipate way too fast the neuron production, and this results actually in limited output of neurons and a severe microcephaly. Now, before I go into detail, I want to tell you a little bit more about this PRC2 or polycomp repressive complex 2. It's actually regulating chromatin, and it's required for cortical development. So this was known before, but what is PRC2 complex doing? It's actually methylating histones, and that leads to gene silencing. So in other words, you're repressing gene expression. Now, if you're losing part of the complex, namely this EED, for instance, as we did together with Denis, you're actually losing also methylation, and you have no gene silencing, and you have increased accessibility and expression of target genes. And thereby, this correlates with a very severe microcephaly, as you can notice here. So we then measured cell cycle, and indeed, so there is an increased cell cycle exit during especially upper layer neuron production. There's also a thinning of the ventricular zone, so there's less stem cells over time, and as you can see, this results in a severe microcephaly. So now we were really wondering what is going on and what are the mechanisms of the loss of function here. And we're addressing three questions now in this part of the presentation, namely, what are the mechanisms of this PRC2 complex in temporal lineage progression? And also, to which extent is that function cell autonomous or necessary at the global tissue-wide level? And we have seen that there's a problem with neuron production if you're losing that complex, but there may be other functions in lineage progression, which we're also looking at. Now, this is the project really driven by a very talented postdoc, Nicole Amberg, together with Florian Pauler, who is a bioinformatics expert. Now, the paradigm that Nicole was using is actually really to try to address, firstly, that question, namely, how we can dissect intrinsic gene function and the global or stem cell niche tissue-wide mechanisms due to loss of function at single cell level. And for this, we're having two paradigms here. In the first one, we're knocking out a candidate gene. Now, in this case, it's going to be EED from the PRC2 complex, just in single cells with a normal environment. And we can trace the phenotypes of these mutant cells with high single cell resolution and thereby infer a cell autonomous gene function. Now, as you know, usually we can also induce a global knockout or a conditional knockout. And also together with this labeling, the MADAM labeling, you can see that these mutant cells are homozygous mutant for that gene, but it's the same like these cells. And any difference in the phenotype of the green cells in such a paradigm where we have a mutant background in comparison to this paradigm where we have a normal background actually can provide some hints at these non-cell autonomous effects or mechanisms. So this is what Nicole then did, and here is the summary of the data. So we have a control here with MADAM labeling. It's a bit low res, but here it's at higher resolution, so all the cells are wild type. Then we have the conditional knockout. You already have seen we have a dramatic microcephaly here, which can reach up to 50% of the normal cortex size. And then we also induced the genetic mosaic with sparse deletion of the gene in single cells here with a normal environment. And we didn't see any microcephaly here, but even more surprising was when we counted mutant cells and compared them to wild type cells, we saw a ratio of 1:1. So in other words, there were no less green cells than red cells. And also that shows really that cell autonomous loss here of EED does not affect neurogenesis as such, at least not to the level that we have when we mutate the gene everywhere. So this is really providing evidence that EED itself or the PRC2 complex is not required cell autonomously for cortical neurogenesis. Now, to address this a bit more in detail, we went back to single clonal analysis, so where we really induce single stem cells. And you can see that if we do this with a conditional induction of these MADAM clones at embryonic day E11, look 2 days later, we basically don't see a difference between the number of EED mutant cells and wild type cells here at this level. We also look a little later at E16 at the end of neurogenesis. Also here, when we compare mutant cells to wild type cells, it's roughly the same, so there's no significant difference. So that really shows that at a single radioglia progenitor level, introduction of a mutant lineage here compared to the wild type lineage doesn't affect any of the output of these cells. Now, this really shows that there's no cell autonomous function, but then we still measured the total output. I told you earlier, there's a unit of about 8 to 9 neurons, and which you can really count here when you induce the tamoxifen and induce the madam event at about embryonic day 12, when neuron production starts roughly, and then look at 3 weeks of age. Now, also in this case, the total unit size here is not changed. So black is EED mutant and wild type is the same. It's about 8 to 9 neurons. And also in superficial or deeper layers, there's no difference in the total output within single clones here. So that really seems to confirm that cell autonomously PLC2 is not required for cortical neurogenesis, and that the dramatic microcephaly we see in the conditional knockout is rather the effect of a systemic loss of that gene. Now, this is summarized here. So basically, having single cell knockout, you have normal neurogenesis. With a conditional knockout, all cells mutant, you have a major defect in neurogenesis and microcephaly. And so we wanted to know what's different actually in these two paradigms. What's different in the green cells when all the cells lack that gene, or in the green cells with a normal environment. To this, we then isolated tissue, dissociated the cells using FACS sorting, and then extracted mRNA and did RNA sequencing to get a transcriptomic fingerprint of those cells and compared to control, and then again the mutant cells in the mosaic or mutant cells in the conditional knockout. Now, as a control first, we measured EED expression. So in all our paradigms, regardless if it's sparse or conditionally, we actually have downregulation or ablation of these EED transcripts, whereas in the wild type or in the control, there's a robust expression level at the times that we measured. Now, when we look at deregulated gene expression, it's actually dramatic. So now when we compare the green cells from here with the green cells from there, we saw that at about embryonic day 13, which seems to actually mark the onset of the problems in neurogenesis in the conditional knockout, we start seeing deregulated gene expression. And that is quite dramatic if you look in the time course. So there's more than thousands of genes that are upregulated, which in principle makes sense because it's a repressor. So you're losing repression, you have ectopic upregulation, but we also have downregulated genes, and these likely are secondary effects that may also play a role. Now, if we look at pathway enrichment, there's all the famous pathways that seem to be affected, the Wnt signaling, which we know is very important for neurogenesis, there's p53 signaling pathway, cell cycle, etc. If we look a bit more in detail, especially at E13.5, when the microcephaly seems to start, we see there's lots of deregulated genes, but most importantly, if we measure cell cycle dynamics or EDU incorporation into the madam cells, we see specifically in our CKO, where all the cells lack EED, there's a sharp drop, but not in the mosaic. Again, this shows that cell autonomously, cell cycle is not affected if you're losing EED, but then systems-wise, it is affected. The same is true for cell death or apoptosis-related pathways and genes. We see in our CKO, there's quite some upregulation of the apoptosis. This is also measured here, and this is not the case in the single-celled or sparse knockout of EED. So this shows really that there's a dramatic difference in cell cycle and apoptosis, but only if you're losing that gene in all the cells, but not in sparsely the single cells. Now, microcephaly is often associated with mutations in these genes, regulating cell cycle and DNA repair and chromatin organization, as I just told you. But then in mice, what happens sometimes is that if people do loss of p53 experiments, that can rescue many of these microcephaly phenotypes. And so we ask whether our microcephaly phenotype can be also rescued by loss of p53. Now here's the data, and this is not the case actually. So I walk you through. So basically this is a wild type scenario. We have a regular cortex, a certain thickness. If we're losing p53, not much happens actually. So the cortex is more or less intact. Although we are not having dying cells so much anymore, so we measure that, of course. But then if we compare to, oops, what happened now? Sorry. If we compare to the EED CKO, we have this massive microcephaly here, as you can see. This is also measured here. But then also losing p53 homozygously doesn't do anything, and the microcephaly is similarly severe. So that really shows that whatever pathways are affected, it's not acting through p53. Now we were interested to find out, okay, what target genes are we actually talking about that we now having ectopic expression? And to this, we went back to a study from the Huttner lab where they actually measured by ChIP-seq all the histone modification tags here. And so the rationale was, if you have this histone methylation, you wouldn't have gene expression at all. And if we combine this with our POC context, if you have deregulated genes where you don't have a tag, this would be secondary effects. And if you have normally histone methylation tag and you would have ectopic expression, this would be the POC to direct targets. So this is shown here. If you're losing the activity, you would lose the methylation. You're having ectopic high gene expressions. We have already seen this. There's many genes. And now combining this with the Huttner data set, we really can see that downregulated or upregulated genes where normally there's no histone modification, this would be secondary effects. And if there were a histone modification close to this gene, this would be actually the primary targets. And this is the data, so it's actually quite dramatic. So if you focus on the direct target, they are massively, there's a huge number of direct targets, about 180, and very little in the mosaic, but these seem to really repress genes for basic biological developmental processes. That was to be expected. But then more surprisingly was that the secondary effects are also much, much bigger in our conditional knockout. So there's 370 at only embryonic day 13.5. And these actually seem to really affect cell cycle, DNA replication, and these processes critical for neurogenesis. Now, when we do network analysis, we can connect direct targets to this cloud of secondary effects. And in fact, it's very specific for the conditional knockout. So for instance, this gene CDKN2A is only upregulated in the conditional knockout, but not in the mosaic and not in the wild type. So we believe that in this cloud of deregulated genes from a small set of direct target genes, we are affecting major pathways that control neurogenesis. So this really shows that there is an important role of EED, PSC2 complex in lineage progression here, and the deregulation of a relatively small number of direct targets leads then to massive secondary effects, and those actually include cell cycle and apoptosis deregulated genes, but there's no rescue if we also lose P53. Now, this shows really a dramatic function of PSC2 activity in lineage progression and neurogenesis as such, but there has been in the literature evidence or suggestions that it may also affect production of astrocytes. However, the evidence is conflicting. Some say it does, others say it wouldn't, and so we took this as an opportunity to analyze astrocyte production in the absence of PSC2 complex. The data is shown here. So we have a control madam, all cells are wild type, where we have about 20 astrocytes per square centimeter. Then if we're losing EED in the green cells, we have a dramatic reduction of astrocytes here, as you can also see from the overview. So that shows that EOC seems to be essential for astrocyte production. We also looked in a time course, and there's no rescue later on during development, so it's not a delay. So it's really, while a certain level of astrocytes are produced, this level is constantly low. So there seems to be an astrocyte production problem. We also measured at the clonal level using, again, our clonal induction paradigm. Also here we see there's a significant drop in astrocyte numbers per clone when you're lacking EED. Now, we also looked at morphology of these cortical astrocytes, and it's actually quite dramatic. So if you look in a time course, these, especially at 3 weeks of age, it's quite significant. So these astrocytes lacking EED are much smaller, and also the branching pattern is a very different one. So we wanted to also see a bit further with transcriptome analysis what goes wrong here. So we isolated astrocytes, again using a FACS paradigm, and then measured, again, gene expression differences of wild type versus mutant. And also here we see quite a dramatic deregulation of genes. There's not that many as in cortical neurogenesis, as you can see, but there is a significant amount of genes up and down regulated. What we think is particularly interesting is that actually genes related to synaptic signaling are ectopically upregulated here. in the mutant cells, and that may be interesting in regard to the morphological phenotype as well. So this is something that I think we should look at further. Now, let me summarize this part here of the presentation. So I showed you that we have a global tissue-wide function for EAD or PSC2 for neurogenesis, and I showed you that actually it's a secondary effects due to deregulated gene expression which seem to be responsible for that phenotype. It's p53 independent on top of it, and cell autonomously, PSC2 seems to have no function for neuron production. In this slides before, I showed you that also once glial cells are being produced, there's a cell autonomous function for astrocyte production and maturation. So there's a different function very clearly that is also required. I didn't show you the data, but there is also a function in adult neurogenesis which we don't understand completely, so we are still looking at this. Now, I want to really focus also on the second part, which is way shorter, so it's don't be afraid. So it's not as long as this one, but it's an intriguing new finding actually that relates to asymmetric cell division. And as you can see here, during lineage progression of these radial glial progenitors, by asymmetric cell division, they produce neurons and or later astrocytes. And the field is actually quite mature in that sense. There's ways how to generate asymmetry in a neurostem cell division. So either you have the secreted signaling molecules which may pattern the plane of division. Alternatively, also the whole tissue may be patterned and with some polarity cues that may instruct asymmetric cell division. Or there's also intrinsic components which actually drive the fate of the two daughter cells. Now, for the last few minutes, I really want to focus on an alternative theory, actually, which is the selective chromatid segregation model. So what do I mean with this? During cell cycle, we know that chromosomes or DNA is replicated, and there is always two sister chromatids. Now, this selective chromatid segregation model actually proposed that the two sister chromatids, although identical in DNA sequence during replication, they would acquire some kind of tag. So the hypothesis is that it's also epigenetic to distinguish themselves, and even further that this selective segregation of these two sisters to either the stem cell or the postmitotic cell would actually instruct the fate of these cells. So in other words, because there's epigenetic tags, potentially they may produce these non-equivalent sister cells that then would correlate with the fate of the daughter cells produced. Now, this sounds a bit spooky, but there is actually real evidence that this may really happen. In particular, in Drosophila, it was found in vivo that this non-random sister chromatid segregation happens during germ cell division, but also from cultured mouse embryonic stem cells in vitro. And what people have used to actually measure the segregation of sister chromatids is an assay involving interchromosomal recombination that can mark the two sister chromatids selectively and thereby enable the tracing of chromatid segregation in stem cell division. So again, we would have a cell in G1, then DNA synthesis happens, so you duplicate your genetic material, but then this X marks actually a recombination event. This happens, this can be actually induced artificially, but it also happens naturally every now and then. But that would produce these so-called recombinant chromosomes. So you can see the red and blue split as opposed to the parental blue and red chromosomes here. Now, depending how these recombinant chromosomes that can be traced now segregate, it can be that they go to the same cell and the non-recombinant one go to the other one. This is called set segregation. And again, this happens also in Drosophila and other organisms. However, if these recombinant chromosomes go to different cells, actually this leads to a loss of heterozygosity, especially in these distal parts. You see both parts here are red and here it's blue. Now, in principle, if chromatid segregation is completely random, you would expect a 50% segregation in that direction and a 50% segregation in that direction. However, as I already mentioned, if people use this in the embryonic stem cells, mouse embryonic stem cells, and trace chromosome 7, for instance, what they found is in neuroectodermal stem cell lineages, there's actually no G2X, so no segregation in that direction, but that in endodermal lineages, there's only G2X, which produces a number of organs here. So that really clearly shows that there's non-random segregation. But this is in vitro and only for one chromosome. So we thought we're going to address this question and look how sister chromatid segregation behaves in our neocortical EMX1 positive lineage that are produced by radioglia cells. Now we benefit from the MADAM technology, and without going into detail, but the aficionados know that MADAM relies on interchromosomal segregation here to reconstitute marker genes, GFP and tomato. That's why we have red and green cells and yellow cells. Now what happens in a MADAM event is that we're producing these recombinant chromosomes here, and depending how they segregate, it's against X segregation. So if they go to different cells, we get a red cell and a green cell, or if they go together, we get the yellow cells. Now this enables us to in a highly quantitative manner to actually distinguish X versus Z segregation, and to this extent, we define a so-called yellow-green-red index, or YAGRI. And this is really the number of yellow cells divided to the average of green and red cells. And now if sister chromatid segregation is completely random, you expect a YAGRI of 1. In other words, it can be 50-50 chance to either go that route or that route. So in other words, 50X or 50Z. Now when we look at projection neurons, cortical pyramidal cells, we were actually surprised that only for 2 chromosomes, chromosome 2 and 17, you get a YAGRI of about 1. So only here you get about 50-50 segregation, but for all the others, we actually see a biased segregation pattern that can be up to an order of magnitude higher here towards the Z segregation. In other words, we have way more yellow cells than red and green cells. Now you could argue, okay, this may have to do something with the efficiency of recombination maybe, but this seems not to be the case, because if we look at the chromosomes that show higher recombination frequency, or the yellow cells, and establish a ranking plot, there is no correlation of the non-random segregation bias with recombination efficiency. Now, lastly, we also looked in other cells, so cortical astrocytes in particular, and we see there the bias is much less. So it's only within a factor 2 or 3, actually, for the chromosomes that we are testing. But we also looked at CA1 pyramidal neurons. They are also positive for EMX1, actually, although different lineage, and cerebellar Purkinje cells. And we do not see any pattern. So in other words, for chromosome 7, we have a YAGRI of 2 for astrocytes, about 4 for pyramidal cells in hippocampus and Purkinje cells, and almost 9 or a bit more for cortical pyramidal cells. So we really believe there's chromosome specificity in how you segregate your sister chromatids, but also cell type specificity. This is summarized here. So basically, what I just said, for different chromosomes in a given cell type, there's a variable probability how you segregate your 2 sisters or sister chromatids, which can be 50-50, but there's also mostly a bias for most of the chromosomes into the other direction. And we looked at different neurotypes, so also they differ from each other. And finally, we have also analyzed peripheral cells, T and B cells. They also show this phenomenon of non-random segregation, but it's also very different from the neurons here. So what we are proposing is that there is really a non-random sister chromatid segregation in the production of projection neurons, but also glia cells. And we're proposing that this could be a mechanism for asymmetric neurostem cell division. Again, the mechanism would be unclear, but there is evidence that chromatid-specific epigenetic modification, in other words, the green would be epigenetically modified differently than the blue, that this may correlate with the instructiveness of whether you're getting to a post-mitotic cell or a stem cell. So this is still work in progress. Part of it is actually put on a bioarchive if you're interested to read a bit more. Now, with this, I would like to thank the people involved here. So I mainly talked about the work of Nicole in the lab and also Florian in the first part with the PLC2 complex. Actually, Nicole is going on the job market right now, and also Robert Beatty is on the job market. So if you're interested in people who can master the madam technology, these are two excellent candidates. I also want to highlight a talk by Andy Hansen on Thursday in this Neuromatch forum at 6 p.m. Central European time. And acknowledge a few collaborators, in particular Denis Jambordon, and also funding sources. And then I would like to thank again Guillermina and all of you for having me today.
So thank you, Simon, for this wonderful talk and this exciting data, and thank you for sharing this. So if you go to the question and ask and answer button there at the bottom of your screen, you can read 3 questions that some of the audience have raised. And if you go fast, I think we will have time to answer the 3 of them if you can. Yeah. So can you actually read the questions so people, yeah?
Sure. So I'm reading a question from Maria Pompeiano. Sometimes you seem to compare somatosensory to visual cortex. Are the effects of EEG knockout the same in different neocortical areas? The short answer is we don't know. We haven't distinguished between functional areas, but it seems very important because what we do know is for the neurostem cell lineage progression and the clonal units that we saw, the number of 8 to 9 neurons from a radial glia cell produced, that seems to be invariant for different functional areas, but how the neurons distribute in the different layers seems not. So together with actually Oscar Marin, we found independently and in a collaboration that there seems to be a region specificity of how you assemble the neurons. So it would be very interesting indeed to look at this. Yeah, definitely. Second question is by Pranjal Garg. Do you think it will be possible to induce neurostem cells in situ so as to generate neural parts? How can we solve the issue of plastic variation of newly generated part and required levels of computation? Well, not sure if I understand completely the question, but I think this is very interesting. So there's a lot of data out there that imply that there is some remained plasticity, not only in stem cells as they pass in the lineage, but also for post-mitotic cells. So in particular, work from Magdalena Goetz shows that you can, and from many others like Benedict Berenger, you can actually reprogram post-mitotic cells to again produce neurons, in particular astrocytes. I mean, you can with a set of ectopic expression of genes, you can induce to produce neurons. So there is plasticity later. I'm not sure we understand completely all the mechanisms. We know that a few factors, a very small number of factors are sufficient actually. But also recently, Denis Jambordon showed that even stem cells remain plastic, in particular the radial glia cells, as time goes. So I think the underlying mechanisms of having that is unclear. Likely as epigenetic mechanisms.
Yeah.
Yeah. There's another question by Ella Mercer, and I'm trying to read. Hello. Oh yeah. Cortical astrocytes appear to have overall a lower YAGRI when compared to other cell types. Do you know why this is the case? Short answer, no, again, not. So this is very recent data. So basically the question is, the selective chromosome segregation bias is different for astrocytes than for projection neurons. Yes, we noticed that as well. We don't know. So what it could mean is that the level of bias is less important for astrocyte production. In other words, it could be other mechanisms that are much more important than selective chromosome segregation. Whereas for neurons, maybe it is. Again, we don't know the mechanisms, maybe epigenetic modification that could be involved here.
Yeah. I have a question for you. If we have like 2 minutes, I see no other question from the audience. So there is a role that you have found in EEG in the astrocyte, in the production of the astrocytes. Yes. Do you think that the effect is at a certain time point during development on the switch from the neurogenesis to gliogenesis on the progenitors that occurs something that makes the role of this EEG in the production of the astrocytes, or do you think that is something set from the beginning of the progenitor's life that somehow epigenetically might be switched on a certain time point, like a clock that is, you know, switch on? I guess it's difficult to answer this. I mean, maybe you don't know, but it could be both scenarios, no? Or what do you think?
Yeah, in fact, actually, this was our preferred hypothesis that you just said. So in principle, when you look at Denis' paper, we did stainings, and we saw there's a sharp drop after embryonic day 12 in histone methylation. So in other words, the antibody doesn't recognize. But clearly, we see expression is still there. So there may be an ambient level of histone methylation still there. Mm-hmm. But what we think, what we thought is that maybe you have a priming mechanism that exactly sets the stage at the very early stage, but it's only translated later. Given we have also a cell autonomous function in the astrocytes later on, we believe that there's an active role for repression, and if you remove that, it's a separate function then for lineage progression. The problem is if we can separate the function of priming for lineage progression from the other function. At this moment, we believe it's relatively difficult because we also saw that the neurogenesis problem during the early lineage is really, if you knock out a gene in the entire neuroepithelium, not in single cells. So it may be that in the sparse knockout, the surrounding cells rescue these cells, and that's why they don't show a problem. But in the global knockout, there's so much deregulated gene expression if you remove that block that it may actually hide the priming concept that you were referring to. So it would be really nice.
Sure. Yeah, absolutely. Okay, I don't see any other questions so far. So thank you very much, Simon, as we have a very tight, slot. So thank you for your talk, and I hope you guys enjoy, everybody. So thank you for this. And see
Thank you very much, Guillermina. I really appreciate again the opportunity. I'm looking forward to all the other presentations, in fact.
Okay.
So, thank you.
Looking forward to seeing you soon. Bye.
My screen share controls are more, they're in the same, I've got two monitors. So.
Yeah. Hello, Chang Chang.
Hi.
Was that the correct pronunciation of your name?
Yeah, it's Cheng Huang. Yeah.
Cheng Huang. Got it. Okay. Do you want to try your screen share?
Okay.
And then, Dimitra, did you get the two links to the articles that?
Yeah. I have read what I will write and your Twitter.
Okay. Just make. I'm just making sure.
Yeah. Thank you.
Okay, it's good. You can see my cursor.
Looks perfect. Yeah.
Okay. Good. Okay. Great. Okay, cool.
Great. And Noga, you can try yours.
Okay. Let's see. Do you see my presentation and my cursor?
Great.
Yeah, is it on? Okay. Should I leave it on or?
Yeah, you can leave it on. I think, if I'm not mistaken, we're actually visible. So at the point at which people start, you've actually started the webinar. Is that correct?
Yeah. I started that.
Yep. So once people,
I'll start broadcasting at in 5 minutes or 6 minutes.
Oh, okay. I thought that maybe it already was broadcasting because it says we're live on the streaming service.
Yeah, we're live, but I think it shows, actually, I don't know, because I checked on YouTube and it's still the previous. That's strange. We are. Oh, fuck.
Yeah, I think I'm pretty. As people start to come in, they will be seeing us. So if you would prefer not to be on screen, Alex and Chang, you can turn your videos off if you want.
Okay. Yeah, turn off for now.
Yeah, I'll also mute myself because I've got cats over here that might.
Yeah, likewise. Once we get started, I'll be muting myself because of my dog.
So,
Trying to set my background to this, the one that the Neuromatch is the Neuromatch that the one that they sent, but it's not allowing me to add another to set a background, which is weird.
okay.
I think I missed
Maybe
it.
I just have too many. Let's try again.
Should I add a background or?
No,
No,
you're
it's fine.
good.
Okay. Do you understand how to pronounce my name?
Noga. Yeah. Oh, your last name. I'll give that a try. Can you pronounce it
So
for
it's
me?
Mosheov.
Mosheov.
Yeah.
Mosheov.
Yeah.
Nice. Thank you for that. Actually, I totally. Yeah, I'd meant to go through everybody, but I'd forgotten. Have you presented this work before, or is this all new?
In the department seminar, but not in any conference.
Oh, great.
And not that short talk. So it's
So it'll be the first go at the short talk format.
yeah. And the first outside of the university as well.
Yeah. That's good.
Yeah.
It's nice to present fresh work.
Yeah.
What year when did you publish the paper that you had sent me? 2019.
Was,
so that's
2019, but it's on different subjects. It's
yeah.
from my PhD, and this one's from the post.
I see. Yes, they're also a very relatively new paper though. That's good.
Mm.
One minute. I don't know. Okay, I will mute myself. No, I will give you we go on and then I will mute myself. Oh, we go on.
All right, so we'll get started. So welcome everyone to, let me get my tab here. I apologize for this. So hey everyone, I'm Adam Steele, a postdoc at Dartmouth College in Hanover, New Hampshire. We've got a great lineup of speakers in this session who are doing some really interesting work modeling neural systems with recurrent neural networks. As a reminder, we have the chat where you can post messages and clarification questions. Just make sure that you have the message set to panelists and attendees. If you'd like to introduce yourself, now is a great time to do that. We'll also have the Q&A tab for questions. So you can post questions at any time and upvote your favorites. And for those on Twitter, please tweet about the session using hashtag Neuromatch3. So with that, I'll introduce our first speaker, Noga Mosheoff, a postdoc at the University of Pittsburgh. She'll be presenting her work looking at the properties of population activity in artificial neural networks, titled Chaotic Dynamics in a Spatially Distributed Network Generate Population-Wide Shared Variability.
Thank you, Adam, for the introduction. And thank you for the opportunity to present my work. This work was done with my advisor, Chen-Chang Wang, and with our collaborator, Bart Ehrmantraut. So let me start with a quick introduction about population, about shared variability. Here we can see different neurons and their spikes over time. And N would be the spike counts over time in time window T. And you can see that the neurons are highly variable in this example, where a shared variability is defined as the correlation coefficient between the spike counts of a pair of neurons. In this figure from Smith and Somers, you can see the shared variability or the correlation between spike counts as a function of the distance, or the cortical distance between neurons. And you can see that this correlation decreases with the distance. This is called population-wide shared variability. More evidence for this phenomenon can be found in these papers over here. Okay. It was previously shown by Wang et al. That spatiotemporal dynamics in spiky neural networks generate population-wide shared variability. And this is a figure from the paper where the correlation as a function of the distance between neurons is wide. We noticed that the covariance between spike counts is equal to the covariance of the firing rates, at least for Poisson neurons. So if this is true, so a Rett model might exhibit shared variability as well. Our hypothesis is indeed that shared variability might be explained by the firing rate dynamics without any noise. In order to show this, we're going to analyze a model of two populations, excitatory E and inhibitory I, each distributed on a two-dimensional sheet with periodic boundary conditions. Each has its own time scale, tau E and tau I, and the connectivity is spatial with two different spatial scales, sigma E and sigma I. The dynamics of the E population can be described by minus the rates plus some nonlinear transfer function of the total input, which is the connectivity, G is a Gaussian, convolved with the rates, plus the connectivity of the I population convolved with the I rates, plus some constant input. The I population have a very similar dynamics, and phi is our transfer function. So in this talk, I'm going to keep all the parameters constant, except for two, which are sigma I and tau I. Because of the symmetry of this model, we know that there is a uniform solution, which means that there is a solution where all the neurons in the same population are identical. In order to understand the behavior of the model, we want to compute the stability of such a uniform solution. So first we assume that we have a uniform solution. We assume that there is a uniform solution that all rates in the E or I populations are identical. And if this is the case, our model reduced to just two neurons model. There is no space. And we have one excitatory and one inhibitory neurons. And the behavior of such a model is pretty simple. There are two stable solutions. It depends on tau I. For a range of low tau i's, we have stable fixed point. For a range of higher tau i's, we have a limit cycle, stable limit cycle, which means that the rates oscillate between these two values of R. And we have a small range of taus where both solutions are stable, both fixed point and limit cycle. So now we wish to compute the stability of the uniform solution for the full network and not just for the simplification of two neurons. In order to do so, we first compute the stability of the fixed point. I'm not going to elaborate on the math, but just to explain the main idea. So we linearize our full dynamics around the fixed point. We're using a Fourier transform and we compute the Jacobian's eigenvalues. If all the eigenvalues smaller than 0, then the fixed point would be stable. By doing so, we obtain a range or a regime of stable fixed point, which is the purple regime here, as a function of psi I in the x-axis and sigma I in the y-axis. Okay, so that's nice, but still it's not enough. As we also want to compute the bulk oscillation stability. Bulk oscillation is a uniform solution where all the neurons in the same population are identical, but can oscillate over time. In order to understand this, I'll show you a movie with a small example. So the x-axis here would be the neuron location, the y-axis is the firing rates, where the blue and red curves are for the E and I populations. And we can see that the neurons are identical to each other in the same population, but oscillates over time. The stability of such a solution can be computed using the method that was presented in Ali Harris and Ehrmantraut 2016. What they do and what we did is to linearize the equations around the limit cycle this time, using a Fourier transform and compute the eigenvalues of the monodromy matrix this time. More details about this can be found in their paper. Anyway, using this method, we obtain a range of parameters of stable bulk oscillation, which is the blue area here. You can see also that there is an area that both fixed point and bulk oscillation are stable, as was in the simplification case. There is another area, the yellow area here, for eigenvalues that are smaller than -1. In this area, at least close to the stability border, we expect to see some alternating special patterns. And this will be a bit more clear in the next slide. And we also have another area, the green one, in which we expect to see non-alternating special pattern. So next I just want to show you some examples of the behavior of the model. I'm going to focus on the yellow area, as the fixed point is just a solution where nothing is going to change over time. The bulk oscillation is similar to what I showed before. And now let's look on the parameters that are signed in the square here. So in this movie, the axes are the neurons locations, and the colored map are the rates of the E population. And here we can see a solution of traveling waves, totally periodic and regular. We understand what's going on here. Okay. Next, we in different parameters closer to the border of stability, we can see a solution of alternating bumps. So this is what we see here. You can see we have bumps, but they alternate in location. And it's still very periodic. Another solution would be for alternating stripes. So there are stripes and alternate their location. And lastly, I want to show a solution of irregular pattern. So we don't see any regular pattern in this solution. And I already wrote here that this is actually a chaos. I didn't show you yet. But we can be convinced that our previous analysis of stability of bulk oscillation is not enough in order to understand what's going on in this yellow area. So next, we compute the maximal Lyapunov exponent, which is a measure for chaos, MLE. So a positive Lyapunov exponent would mean chaos. This is in yellow over here. A negative MLE is a fixed point, and zero MLE would indicate periodic solutions from different types. We can see that we do have a chaos in our model, a range or a belt of chaos in the area of sigma I close to 0.1, which is also sigma E. And now we can look on the behavior of this chaos in the model and compute its autocorrelation. So in the middle figure, I plotted the autocorrelation of the parameters signed in square here. You can see that this autocorrelation is pretty wide. It's almost isotropic. If we look on the autocorrelations of different examples of chaos, we see that they are all wide. And this might explain population-wide shared variability as we wish. The next thing I want to talk about is adding noise to the system. So here we use the same model, but driven by noisy input. Each population is now receiving two types of noise. One is correlated in red, which is identical to all of the neurons in the same population, and independent noise in blue, which is differ between each of the neurons in the population. Sigma n would be the standard deviation of the total noise, where C is the correlation factor. So for C equals 1, we have only correlated noise. For C equals 0, we have only independent noise. Let's see the effect of this noise on our system. For small noise and for the parameters of traveling waves, we can see that we can clearly see still traveling waves. For the same parameters and the same initial conditions, but for larger noise, larger standard deviation of noise, we don't see any regular pattern. It could be chaotic. In order to be sure what's going on there, we compute the MLE, and we see in the left one the map of MLE without any noise that we already saw before. If we add some noise in the middle, the range of chaos extends a bit. And if we add more noise, the chaotic regime is much larger. Even larger noise would just give us a yellow map in all of the parameters. Okay, so the last thing I want to talk about is analyze this noise even a bit more. When separating the two parameters of noise, the correlation parameter C and the standard deviation sigma n. Let's look again on the parameters of traveling waves. We can see here a sharp transition between traveling waves in blue to chaos in yellow. This is the MLE, of course. If we look on the same simulation, but as a function of sigma n times square root of C, which is the standard deviation of the correlated noise, we can see the same transition, and this transition is only depend on the correlated noise, actually. We did the same for different parameters as well, alternating bumps, alternating stripes, and bulk oscillation. We got pretty similar results. So just to summarize, we proposed a firing rate model that exhibits population-wide shared variability without any noise through chaotic dynamics. Correlated noise expands the parameter regime of chaos. And I didn't have time to show, but we also found that the chaotic dynamics contain low frequency power and low dynamical structure in correlation. I want to thank again my collaborator and advisors, Chenshang Wang and Bart Ermentrout, and thank you. I would like to answer any questions if you have.
All right, thank you so much, Noga. Unfortunately, we won't have time to do any questions live, but over right now, but you can post your questions in the chat and Noga will be around to answer questions there. And so now we'll move on to our second talk. This talk is being given by Alex Kunin from Baylor College of Medicine, who is doing some fantastic work modeling the vertebrate retina. His talk is titled Receptive Field Geometry Shapes Information Content of the Neural Code.
Okay, I think I have to.
Noga, you just have to stop screen sharing.
All right, so desktop 2. All right, people can see my slides. Great. Okay, so I'll try to. There will be some details that are skipped, and this is like a sampler of work that is being written up right now. But basically, I'm relating sort of individual neural activity, so receptive field properties, properties of their tuning curves and things like that, to a population level statement about information content of the code. So first, just a real quick retinal ganglion cells. Retinal ganglion cells are a spiking neuron in the retina. They are generally very weakly correlated, even in the face of like naturalistic stimuli where nearby pixels of the image might be very highly correlated, things like that. But they are not independent, and I'll come back to this in a moment. So this is a population level statement, weak pairwise correlations. Okay. On the individual neural activity level, we can think of neurons as just responding to external stimuli, where there's some external stimulus S, there's some transfer function, and this gives you the firing rate, and noise is anything I'm not talking about right now, anything that I'm not accounting for in my model or what have you. These are gross oversimplifications, but here, for example, is a spatiotemporal receptive field of a retinal ganglion cell, a mouse retinal ganglion cell. I'm doing a little bit of a sleight of hand here. I say the transfer function is, I'm actually monotone functions where the support of, there's going to be a convolution inside here and the support of that convolution is convex. But, see my paper when it gets posted to probably the archive in like a few months. That basically this receptive field here, and can you see my cursor? Is that? Okay, good, yeah. This actual physical receptive field as a subset of the visual field is a convex set. So that's the kind of convexity I'm interested in right now. Okay, so there's this paper from 2006 that you ignore those weak pairwise correlations, you ignore them at your peril. You need them to properly describe the sort of global population state of the network, but you don't need more than that, which is surprising. So I just noted that these receptive fields are convex, and so for mathematical reasons, I believe I hypothesize that this should be enough to produce such codes. So I should be able to use, if I have neurons that are purely driven by their stimulus, purely driven by the interaction of the stimulus with the receptive field, and the receptive field has certain geometric properties, that should produce codes with these things. So I need to explain all of the parts of the statement, which is what I will do now. So we're going to test this with some simulations and compare it to some mouse retinal ganglion cells. And so first I'll talk about these simulations, and then I'll talk about the analysis that we did to explain this idea of information content. So I'm going to model cells as basically linear nonlinear models. So this is a Poisson rate, a Poisson spiking process, where I'm convolving with this F. So this is, S is a video. So this is the pixel value. This is the pixel location. This is the frame, and this is the value, the intensity of that pixel. And again, noise is sort of everything that's not being accounted for. And in my simulated neurons, this term just disappears. So for example, I just compute the spike-triggered average receptive field from a population of actual neurons, and then I'm going to feed the same stimulus back to this type of a neuron, where all I'm doing is I'm just making sure that I match the sort of overall firing rates. So each simulated neuron should match the overall firing rate of the real neuron that it is modeled after. Okay, so that's the simulation. I'm feeding a stimulus through this convolution, some exponential, or just actually I think just a ReLU nonlinearity, and I get a firing rate, and then I have a spiking process, and I look at the code. So what is this information content that I keep talking about? Information content in this case is, we're looking at this from a combinatorial neural coding perspective. So we're going to bin each spike or no spike in bins of width delta t. We're using delta t of like 10 milliseconds for most of this work. And we think of the state of the population in one time bin as a sample drawn from some probability distribution on n-bit vectors. So here, even though this neuron spiked twice within this bin, we still only record a 1 in that position, for example. And the point is, 2 to the n, which is the number of possible binary vectors, is a very large number. That's too many. So we're going to approximate the true distribution using the max entropy principle. So the max entropy principle, if I want to describe a probability distribution on n-bit vectors, I will maximize entropy, where entropy is this measure of randomness. I think of it as the sort of the opposite of information in some sense. People typically think of as entropy as the information contained in the code. And the point is, we're going to look at a sequence of distributions where the probability distribution that maximizes entropy subject to the firing rates match, the mean value of each bit matching the mean value of each bit in the data, the mean value of product of bits matching the mean value of product of bits in the data, and so on up to the kth order. This will be our distribution PK. This is the distribution which matches these features of the data and makes no other implicit assumptions. That's the way to think about this. So if we just match the firing rates, then we get independent neurons. And what I mentioned earlier, that retinal ganglion cells are not independently spiking, what the precise statement is that P1 variable approximation of the data distribution of spiking code words, of these binary code words from the data. Incorporate the second moments, so these pairwise products, then the distribution of this form, where J is a symmetric matrix, so this is a quadratic form symmetric matrix. The compactness of the notation is just a convenience because we're using binary vectors. Oftentimes you'll see people use plus and minus 1 for spike or no spike, in which case this looks a little bit different. The point is, these parameters, there's roughly n squared of them, which is a lot less than the n, and we estimate them using maximum likelihood estimation or minimum probability flow. There's lots of algorithms for estimating these parameters, again, to solve this optimization problem. If we kept going and incorporated all of the, sort of all of the correlations of the data, we'd get the data distribution back. So that's something to keep in mind. So there's more than this in the write-up, but for brevity of this talk, I'm going to be focusing on this quantity I2, which is the numerator is how much information do you get by incorporating the second moments, by incorporating the pairwise correlations. And the denominator is what is the total information content beyond the mean firing rates in the data. So the change in entropy from here to here is the total information beyond mean firing rates, and the drop from here to here is the information just in the pairwise correlations. And so that 2006 paper, one of their results was that this quantity is typically very high in all of these retinal cells, and across even some simulations. So across stimuli, across species, this I2 hovers near 80 to 90%. So there's lots of work about this phenomenon of sort of low order correlations shaping the population level state. So this was just one of the first ones that we found. And so the point is that our model neurons sort of reproduce this result. So here, these 3 bars are a population of mouse retinal ganglion cells. This data is available on CRCNS. They have a high I2 quantity. This is the mean I2 across subsets of 10 neurons. The simulated cells are a little bit lower, and there's explanations. I believe that this makes sense for for at least for like this V-bar stimulus, it makes sense. And so this drop sort of represents the difference between the real cells and the simulated cells. But also we presented some stimuli to the simulated cells that were not presented to the mouse, and we also get this high information content. So this sort of confirms what we thought, that these are cells driven purely by the receptive field. They have convex visual receptive fields, this F hat. There's some transfer function to make this a positive firing rate, and we get the result we expected, that I2 is fairly high. I2, the pairwise correlations capture most of the information content of the code. So why should this be? And here I will have to give I will actually skip ahead to the other end. Basically, correlation between cells goes up when the receptive fields are overlapping, or the receptive field centers are close together. There's a mathematical result called Helly's theorem, which basically says if you place convex sets in the plane, so 2-dimensional convex sets like visual receptive fields, they're constrained in how they overlap. And specifically, once you know the 3rd order overlaps, all higher order overlaps are completely determined. They either exist or don't exist. So this is, you can think of this as, once we know the 3rd order correlations, within very tight bounds, we know all higher order correlations. And then this last piece is that it's likely, this is another sort of quantitative statement, that it's very likely that knowing the pairwise correlations is very likely to give you the 3rd order correlations, okay? So once you know how pairs of receptive fields overlap, you're likely to know with like 65% probability, you know how the triples overlap. And then once you know how all of the triplets of receptive fields overlap, within tight bounds, you know how all of the higher order correlations must be. And therefore, once you know the correlations, once you know the pairwise correlations, you essentially within tight bounds know all of the higher order correlations. So most of the information is captured by those pairwise correlations. So the bold statement I will make at the end is that the dominance of low order correlations in these populations may be an epiphenomenon of receptive field geometry, as opposed to some recurrent interactions or things like that. So with that, thank you. I'll take any questions. I think I might have a minute.
Okay, thank you very much, Alex. That was a great talk. If anyone has time, or if anyone has a question, feel free to post it in the Q&A. I guess my question would just be if you could unpack the what I guess is a pretty bold final statement there, what some testable implications might be for the experimentalists. Yeah, that would be a.
So this is the dirty little secret of this work. One of the testable implications is that you should see a similar phenomenon with hippocampal place cells for similar reasons, as long as they're receptive, as long as the place field is fairly convex. And there's been at least one paper that I've seen where they tested this at least they tested the implications of the geometry, and they found that it maybe wasn't as strong as we're claiming it should be. These are things that we're going to address in the write-up, because we've been working on this for a while. Things got in the way, and so new papers have come out that now we have to go back and say, well, okay, are we actually right? And the core idea should still work, which is that correlation should die off with distance between receptive field centers.
Right.
Which I think people sort of accept because it just should be true and is true in a lot of situations. But the dominance of low order correlations, that's something that we're making more precise, I feel like. But yeah.
It's really, really interesting. Thank you. So with that, we'll move on to the next talk. If you have any further questions for Alex, you should find him on Twitter at StinkyNeuro, Neural, which is in the chat. And we'll move now on to our final talk of the session, which is from Cheng Huang from the University of Pittsburgh, talking about propagation and modulation of information in the visual pathway.
Okay, thank you. Thanks, Adam, for hosting this session. I'm very happy to be here to share my work. Okay, let's get into it. We know that visual signals passes through multiple stages in the visual pathway. In visual cortex, the information is processed in several cortical areas, such as V1, V2, V3, and etc., in a hierarchical manner. We want to study how information propagates across cortical areas, and secondly, how cognitive factors such as attention modulates information flow. Okay, so here we consider only very simple stimulus parameter. So that is the orientation of a Gabor image. And here I show you trial average population frame rates as a function of the preferred orientation of the neuron. We know that it's well known that neurons in the visual cortex are selective to orientations. So the population frame rates would pick at the neurons that are most selective to the orientation of the image. However, we also know that cortical neurons are highly variable. So for one trial, you may get a population frame rate like the red dot, while for another trial, you may get a frame rate like the blue dot. Okay, so we want to measure the information content in the population activity with respect to the image. So here we use linear feature information as a measurement to measure the information content. Here the stimulus parameter theta is the orientation of the image, and linear feature information is defined in this way. So F theta depends on the derivative of the tuning curve function of the population, F prime, and also the inverse of the covariance matrix of the trial-to-trial variability. Okay, so this feature information measures the discriminability of the population activity with respect to small changes in the stimulus parameter, which is orientation in this case. Okay. So from the definition, we can see that the linear feature information depends on two components, the tuning curve function and the population covariance. So there has been a long list of work in the literature that studies this problem of feature information in neural networks. And most of the models are parametric, meaning that they make assumptions on the function forms of tuning curve functions and as well as the population covariance matrix. And depending on their different assumptions, they often lead to different conclusions. So in some models, a correlation can be important for information coding, where in other models, correlation can hurt information. So it has been a meandering path in the literature and still not clear whether correlation is good or bad. So instead, we want to use a different approach. We want to study this information flow in a mechanistic spiking neural network, where both the tuning curve function and the population covariance are shaped by the circuit wiring. Okay, so specifically, we study a two-layer network. For the first layer, the neurons are modeled as Poisson units with Gabor filters, and so that gives them orientation selectivity. And the orientation preference are arranged according to a pinwheel map. And the second layer are modeled as a recurrent network of excitatory and inhibitory neurons. And the connectivities are ordered, has spatial ordering, meaning that the connection probability between a pair of neurons decreases with the distance. Okay. Okay, so here we can measure the information of the second layer, the information contents of the neurons in the second layer, and look at how the information depends on the different spatial scales of the connections in the network. Okay. Okay, so let's first look at the effects of feed-forward projection width. So here I plot the fish information as a function of the number of neurons we read out from the network. So firstly, for a small number of neurons, we can see that for networks with very narrow feed-forward width, so that's the purple case, we'll have much larger fish information than the networks with very broad feed-forward projection width. And in the extreme case, when the feed-forward projection width is infinity, means that there's no spatial dependence on the connections. Okay. However, as we can see, as the number of neurons increases, then surprisingly, all the information curves converge to the same level. So it means that the total amount of fish information is independent of the feed-forward projection width. And we also can see that they converge to a level that's very close to the input information bound. So it also shows that the network can be very efficient at transmitting almost all of the information. Okay, so that's kind of surprising. Then next we can look at what is the effect of recurrent connections. So for this case, we fix the excitatory projections and the inhibitory projection to be the same. So it's just spatially balanced excitation and inhibition. Then for this case, we can vary the recurrent connection width. So it's from the light color to darker color. And you can see that although there's a small changes in information for a small number of neurons, as the number of neurons increases, the total amount of information also is independent of the spatial scale of recurrent connections. And they all converge to a level that's very close to the input information. Okay, so why is that? So it turns out that for the parameters regime that I just showed you, the network is in the stable regime. So it means that the frame rate dynamics of the network is uniform, and it's without, with constant input current, it will generate asynchronous uniform dynamics. So in this stable dynamic regime, the network response are approximately linear in response to the input noise. And we know that linear fish information is conserved under a linear transformation. So therefore, it means that in this stable regime, then the total amount of information is independent of the circuit's varying parameters, and it's very close to the total input information. Okay, so then that raises the question that how do we modulate the total amount of information in the network? So for example, how does attention modulate information? So our stability analysis of the neural network suggests that we need to go outside of this stable region. So in the unstable region where the network can generate pattern forming dynamics. So for example, we know that when the inputs to inhibitor neurons are weak, the network transitions through a Hall bifurcation and generates wave propagating dynamics. So here shows you an example of the rust activity of the network. So each dot is a spike at that location. So this nonlinear wave propagating dynamics can potentially lower the information in the network. Okay. And previously we have used this wave dynamics regime to model the unattended states of the V4 population activity, V4 data. In this case, in the unattended states, the wave dynamics can generate low dimensional shared variability, and that also produces positive correlations among the neurons. And that's very consistent with the population response in the V4 area during the unattended states. And in this previous work, we model attention as a top-down depolarization of inhibitor neurons. So by just giving more input drive to the inhibitor neurons, we stabilize the network, and that quenches the first mode of the dominance mode of the shared variability, and also reduces the average noise correlations. Okay, then how does this affect the information flow? Okay, so we can see that indeed depolarizing the inhibitor neurons can improve information flow. So in this case, when mu I equals 0, the network is in a wave propagating dynamics regime, and then the internal wave dynamics reduces the information content in the network. And as we increase the drive to the inhibitor neurons, we can stabilize the network. Then we transition into the stable regime where the network is the most efficient as transmitting information. Okay. Then next, one may say that this modulation in information seems very small in percentage. So what about we propagate the information across multiple layers? So here is just a result of a 2-layer network. So next, we also look at a network of 4 layers, and each layer is basically layer 2 to layer 4 are modeled with the same spatial connectivity. They are all recurrently connected excitatory and inhibitor neurons. And then we can look at what is the information across layers. Okay, so first we can look at the unattended states when the network exhibits wave dynamics. So you can see that the information quickly deteriorated across layers. So this is layer 2, layer 3, layer 4. On layer 4, the total amount of information is already only a small percentage compared to the input information. Okay. Where in the attended states, the network is more stable, and we can see that much more information is returned in the last layer. And then next, we can look at the ratio of the information across attentional states at the last endpoint. And we can see that the improvements in information also increases exponentially across layers. Okay, so finally, let's summarize. I showed you that in the stable dynamic region, a network can transmit almost all of the feature information across layers. When the network loses stability due to either border or weaker inhibition, then the internal dynamics of a recurrence network reduce information flow. And lastly, I showed you that attention can improve information flow by suppressing the internal dynamics of the recurrence network. Okay, so lastly, I want to thank my collaborators. So this is joint work with Brent Storm and Alex Puget. I did the work when I was a postdoc with Brent. I also want to thank our other collaborators and members from the Doran group, and also our funding sources. And thank you for your attention.
All right, thank you so much for that talk. That was really, really interesting. I love the, how you showed that it matters more as you add more layers. I thought that was a really beautiful analysis. So you have a question from Matthias Hennig. He says, maybe I missed it, but why would a network then go into an unstable regime in the first place? Yeah, that's a very good question. So it's a kind of interesting, we don't have a clear answer. So why is in the unattended states, we lose more information? Yeah, so it's possible we need some wave dynamics for other functional reasons. So for example, one hypothesis is that maybe the wave dynamics is important for strengthening the connections across long distance. So maybe it's important for plasticity. So we know that during the developmental stage, the retinal waves are important for shaping the recurrent connections. So that's a possibility, but it's still not clear yet. Yeah, that makes a lot of sense. Thank you. David Damaris asks, can you clarify the input? Is it the response of a single oriented receptive field or an array of oriented fields?
The input is just a one Gabor image. Oh, I guess I just stopped the sharing. Yeah, so
yeah.
Sorry. Yeah, the input is just a one Gabor image. And then the neuron on the first layer has Gabor filters. So basically, it's, the response is only just a one orientation. The input is just a one orientation. if it's clear.
I think that's clear. You can post a follow-up if it's still unclear. And we'll one more quick answer. Can you use this approach to understand what kinds of computations a recurrent neural network might be performing? In this context, the recurrent network seems to merely lose information.
Yes. So for this work, basically we don't have complicated computation. So we are looking at the representation information of the stimulus. So just what is the estimation capability of the inputs. So we are not trying to extract more complicated information. Yeah, so that should be very important to study if we consider specific computation to do to change the image, the information of the image. Then we may lose some information that's irrelevant, but return more important information. Yeah, so that's in future direction. Yeah.
Great. So we'll wrap up there. Thank you very much to all of our speakers for this session. It was fantastic, and we look forward to seeing you guys at the university. Internet's probably better and perhaps quieter. So we'll just give a few more minutes, or 1 more minute for people to come in before we get live. So there's the question in the chat is, can you see us? I'm guessing from Mohammed, I'm guessing that means, or you are asking whether we can see you on your webcam, and we cannot see you. But for this session, because it's interactive, you can pose a question and ask to be brought on stage. And at that point, you'll be able to enable your webcam. But we can't see you. That being said, there have been Zoom issues earlier in this week in the news. So thank you for checking. So I'll get started now. So hey everyone, I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. I've been told that we have a few moments as people are trickling in. So I'll use that time just to say that everyone in the US, please make a plan to vote next week. It's super important. So if you've got a plan, that's great. If you don't have a plan, make one. And if you do have a plan, get in touch with people that you know to make sure that they have a plan. So we have a great lineup of speakers today who are doing some really interesting work on the neural coding of motor behavior. This is an interactive talk. So our speakers are going to give a 5-minute talk. If you'd like to come on stage and ask a question, there's going to be time for that. So post your question and whether or not you'd like to be on stage. Your panelists and attendees. So that way we can all see it. Also, because this is an interactive session, if you'd like to introduce yourself in the chat, now is a great time to do that. So post your name and where you're from in there, and we'll all get to see who's among us today. Also, for those on Twitter, please tweet about this session using the hashtag NMC3. So with that, I'll introduce our first speaker, Ruihan Wei, a postdoc at the Johns Hopkins University. They'll be presenting their work looking at the ability to alter locomotor control in the behaving primate, titled Modulation of Deep Mesencephalic Nuclei to Locomotor Control in the Walking Monkey.
Good afternoon, everyone. I'm Rehan Wei from Johns Hopkins University. I'm in Colin's lab. Today my topic is modulation of deep mesocephalic nuclear to locomotor control in walking monkey. The deep mesocephalic nuclear is a large midbrain region. It is thought to play a key role in integrating information for sensory motor processing like sensory, cognitive, and motor. Previous studies have shown that the electronic and the chemical stimulate of DPME will elicit the posture changes and the locomotion, but the single neuron unit active to limb dynamics has not been characteristic. To modulate locomotion in response to self-motion, integration of vascular information is required. DPME cells also shows vascular sensitive like rotation and translation, which is not moment related. And it remains unknown that whether if those same DPME neurons would integrate vascular and locomotion related signals. And also, how do these vascular sensitive DPME neurons modulate during locomotion without vascular input is still not clear. And to address those questions, we did single unit recording of DPME neurons. First, we applied passive translation and the rotation to characteristic the vascular response of neurons in DPME. And second, we recorded the same DPME neurons during treadmill walking in absence of the vascular cues. Four high-speed cameras were used for motion recording, and markerless motion capture deep lab cut were used for extract the animal's posture. And for the result, first we identified 16 vascular sensitive cells in DPME. Most of them are sensitive to both rotation and translation. And here we show 3 examples from these 3 different categories. Like the first one is for the rotational cells. It is rotation. It is have high sensitive to the rotation stimulate, but during translation stimulate, it doesn't show modulate of this cell active. And next, we want to show that the DPI neurons active during the head-fixed treadmill walking. For the first figure, what we showed is from the rest to treadmill walking, what does the kinematics and the neuroactives like? And we show the ankle horizontal position and the knee joint angles during this paradigm, and also the corresponding firing rate. It is very clear to show that the DPME neurons are modulated with the gait cycle. And second, we compared the mean firing rate between rest and the treadmill walking, head-fixed treadmill walking, and it shows that DPME neurons shows an increase during locomotion. And next, we want to show that the phase modulation during the DPM neurons phase modulation during the head-fixed walking. And we presented the mean frequency distribution histogram of two types neurons during head-fixed walking. Also, the corresponding phase distribution of discharge frequency of neurons in 30 gait cycles. Modulation patterns varies between DPME cells. Some cells demonstrate peak active during their stance duration, and some are peak active during a swing duration. Conclusion. We established that the same DPME neurons that are sensitive to vascular stimulation also modulate during head-fixed walking. During head-fixed walking, treadmill walking, DPM neurons show sustained phase-dependent active changes and increase the mean firing rate. Furthermore, DPM neurons show different in the phase dependence during head-fixed walking. And for DPME neurons, it integrates vascular and locomotion-related signals. It is important for the generation of volunteer everyday activities such as walking. And our finding regarding the neural circuits that coordinate gait and balance have important implications for the development of the treatment and the interventions. And with that, I want to thank all the people in Colin's lab. Thanks for listening.
All right, that was really great. Super interesting. So you've got a few questions that are coming in over both the chat and the Q&A. I'm just going to remind people when they post to please ask whether you would, please also include whether or not you'd like to be asked to come on screen. I'm going to ask Omid Zobieri's question, which is, how did you extract the gait cycles? And then after that, we can begin bringing people on screen.
Okay. How I can extract the gait cycle? For the method, I use the camera to recording the walking for recording the video of during the monkey is working, and then I use Deep Lab Cut. This is open source software. It could extract the markless motion capture, and for divided the gait cycle, I have the key point position from a monkey. So I extract like ankle position. So I just define the use left ankle angle to define the gait cycle.
Great.
So the start of the gait cycle is left ankle of the start of swing duration.
Great. So we have one question from somebody who's on stage. So Sean, why don't you ask your question?
Hello, everyone. Thank you for your nice work. And my question is the following. Since when you stimulate this region, you can initiate locomotion, then would you say that the neurons that you are recording now are like sensory neurons that receive information about locomotion, or do you think they are more like motor neurons that are actually controlling locomotion?
Yeah, for now we still don't know whether it is the for this DPM neurons which responds during the head fixed working. It is the motor information is input, or the DPM neurons send the motor command to working. We're still not clear with that.
Thank you.
I'll ask a question from Falanto. Have you studied the response of neurons to active vestibular stimulation? If not, would you speculate about the neuronal responses in that case?
Actually, we already started. We are studying the neuroactives, the DPME neurons active during the natural walking, like walking on the ground. It also have free. The head is free. We learned the active motion, active active vestibular input, and it shows like during when it walking on the ground, it will also increase the active of DPME cells. The active is also increased and higher than the head fixed treadmill walking.
Great. And now we have Robin, who's on stage to ask her question. So you can unmute yourself and turn on your video if you'd like, Robin.
Okay. Hello, great presentation. I was wondering if you think that these neurons play an important role in maybe integrating different signals in order to modulate stance to swing transition, maybe as well as initiating locomotion, or maybe maintaining balance and like catching the center of mass with stepping.
Mm-hmm. Can you say it again? Sorry. Sorry, what is your question?
So given that the two example cells that you show have these different like stance and swing activity, do you think they play maybe some sort of role in modulating that stance to swing transition by integrating different sensory and motor signals?
Yeah. For this cells, DPMEs, vestibular sensitive cells, some of them, I think, some of them have different functions, or they may have different sensitive for the inputs of the like proprioception from working, and also like the motor control from like some cells are related to the extender, some muscles, some are related to the flexor muscles. It all have these possibilities.
Great. And we have time for one final question from Farouche, who asks, what are the alternate motion capture methods? Why did you choose deep lab cut over other methods?
For the monkey, we used we didn't use mark on the monkey because sometimes the monkey is like 4 limbs working. If we attach markers, the monkey will grab the markers, so it won't be stable. And also there are hair on the monkey, so if we attach the marks, it will like move, the marker will have movement, so it would be not that good. So we change it for the markless motion capture deep lab cut. And for the deep lab cut, we can have our own. We can build our own networks for training to capture the specific model of our own. So we choose deep lab cut for analyze.
Great. So that is all the time we have for Ruihan Wei. Thank you again for that great talk. We're going to move on to our second talk, which is going to be given by Vanessa Chang from McGill University, who's doing some fantastic work understanding eye movement coordination. So her talk is titled Mouse Nucleus Prepositus Neurons Encode Eye Movement During Passive and Active Head Movement. So I'll just remind people to use the Q&A and specify whether or not you'd like to be on screen during the Q&A time.
Okay, I'll start. Thanks for the introduction, Adam. So I'll share some preliminary results on mouse nuclear prepositus recording. To briefly start with some background, prior anatomical and lesion studies in rodents suggest that nuclear prepositor neuron in brainstem contribute to head direction stability by conveying vestibular information to head direction cell network, ultimately contributing to navigation process. But this is interesting because prepositor neuron is well known for integrating the eye velocity commands into tonic signal that holds the eye in the new position after horizontal saccade. This functional role has been demonstrated in various animal models like goldfish, cats, rats, and non-human primates. And the recent study in non-human primates in particular look at the output of prepositor neuron during passive and voluntary head movements and demonstrated that prepositor neuron predominantly encode eye movements. So then what about mouse prepositor neurons? So given the vast majority of studies that suggest prepositor's role in head direction cell stability, were all done in rodents, it is necessary to investigate whether mouse preprocessor neuron also output eye-related signal. So to investigate what preprocessor neuron encode, we recorded preprocessor neuron in different paradigms. So first, I record neurons during paradigm that evoke only eye movements in absence of head movement, like static eye position and optokinetic reflex. And from that, I can compute eye position and velocity sensitivity using this equation shown here. And then I can build a fine rate estimation model based on purely eye movements. Then I move on to the paradigm that involves both head and eye movements, the vestibular ocular reflex. And then same thing again, I can compute eye position and velocity sensitivity. So you can see the fine rate estimation based on eye position and velocity in red here. Then I can compare the sensitivity across paradigms. So if preprocessor neuron encode eye movement, then the sensitivity from this eye movement only paradigms should be similar to the sensitivity from vestibular ocular reflex paradigm. So when we initially look at the preprocessor response, the first thing we found that was the eye position and velocity sensitivity were not comparable, as you can see here, compared to other paradigms. Eye position and eye velocity sensitivity are significantly higher compared to other paradigms. And so, so we wonder, is this because preprocessor neuron encode head velocity, or are there any other explanation? And we first checked whether behavior are same during vestibular ocular and optokinetic reflex paradigms. So here you can kind of see that optokinetic reflex makes smaller eye movement, but if I put the eye traces in the same scale, you can see they're compared to vestibular ocular reflex, the optokinetic reflex response is very small, but the modulation is considerably high. So we then test whether if the amplitude or match, how does eye sensitivity compare between two paradigms. So we changed our protocol so to maximize OKN response and decrease VOR response to match those two eye amplitude. And then we start to see different results. So here in red, you can see when the amplitude were matched, eye position and eye velocity sensitivity were comparable. Is compared to the very high significant coefficient in previous slide I showed when the amplitude was smaller. So this suggests perhaps the response of preprocessor neuron is nonlinear. It's not too surprising because the downstream target neuron of preprocessors, abducent nuclei, previously showed nonlinear response to the eye amplitude. So, this may dynamic of mice preprocessors could be similar to abducent nuclei in mice. And finally, we look at the response during active paradigm. Since navigation involves voluntary movement, this is a crucial step to test if the preprocessor neuron contribute to navigation. And eye movement during active paradigm is compared to the eye movement during VOR, that is not amplitude matched with OKN. And we compare eye sensitivity between two paradigms and saw that they're comparable. This again suggests the preprocessor neural response in the nonlinear phase of the curve here, when the eye amplitude is bigger, is similar. So in summary, our preliminary results suggest eye sensitivity during VOR and OK are different when the amplitude eye movement are not within the same range, and eye movement response was nonlinear. But when the eye amplitude was matched, the eye sensitivity during OKN can explain your response during VOR. So overall, our preliminary results suggest that the preprocessor neuron in mice mainly function to stabilize gaze, as previously demonstrated in other animal models. So with that, I'd like to thank everyone in our lab, and I'm ready to take questions.
Great, thank you, Vanessa. That was a really interesting talk. So this kind of reminds me of some of Reza Shadmir's recent work on the neural mechanisms about holding the limb still. So I guess, do you have any, as an opening question, like, can you talk about this in this, is this a similar circuit mechanism to the mechanisms involved in holding the limb still?
It's a little different. I can show the circuit again. So vestibular system encode head velocity. So, and then the limb movement is not really relevant in this circuit.
I guess, right, but the overall structure of the circuit is similar, or not necessarily? As in, like the components of the circuitry. If it's not a good, it's okay to move on. We have other questions as well. We'll move on to, well, Richard, do you have a question?
I do, and I'm also going to pull the next person up on stage, but in the meantime, I'll ask a question. So you have the two conditions, one with the only visual stimulus and one with the only vestibular stimulus, each of which must generate some amount of cue conflict for the animal. And I'm wondering if the nonlinearity that you see in the firing rate has anything to do with the kind of causal inference, like Bayesian cue combination models that people have studied before in, like, I don't know, the ventriloquist effect with audiovisual stimuli or something like that.
That's a really good question, but I'm not very familiar with the Bayesian inference.
Yeah, sorry, it was a little jargony. Allow me to make a slightly reword my question. So is it possible that, let me start one step back. Do you have a sense of whether the vestibular or the visual signal is noisier, like which one's the more reliable cue, and how they get weighted when they're both like in conflict in different amounts?
So optokinetic and VOR system, they work together. So VOR is better with the high frequency stimulus, and then opposite is true for optokinetic, the visual stimulus. So when you see the traces of the gain for VOR is increased with the amplitude, and then decrease is for the OKN. So they're more like, they're not really conflicting, but they're more complementary to each other.
Great. So we have Jean, who's going to ask another question here.
Yes, thank you for your work. And having myself recorded OKR in mice, I know this is very hard. So how exactly did you do to get to match the VOR and the OKR?
So I had to use smaller amplitude stimuli, and also decrease the frequency of the stimulus too, to maximize. Decreasing the VOR gain was not gain, amplitude was easy, but the maximizing OKN, because mouse doesn't really generate a big OKN response. So reducing the frequency was the big component of matching.
Yes, it makes sense. Just what you said in answer to the previous question, that the OKR would be better at a lower frequency.
Yeah.
But so in the end, did you still use the same velocity for the stimulus? So the rotation during VOR and the motion of the visual surround during OKR, did they have the same velocity?
Yeah, they're same velocity.
Okay, thank you.
Okay, great. We have two other questions that have come in. Omid has asked that his question be asked by the panelists. So here it is. Has the nonlinearity in these neurons been studied in non-human primates?
So there, I can think of like two main studies who look at the response of prepositors in non-human primates. One is looking at the prepository response during like passive and voluntary head movement, just like my paradigms. They didn't report any nonlinearity in the papers, and also similar to abducin.
Interesting. Okay. So if he has a follow-up, he can post or if Omid has a follow-up, they can post it in the Q&A. So we have another question from Poom Wabun Sasuki. I'm sorry I'm not going to be able to pronounce your last name. But this person's asking, if the NPH is not sending the vestibular signals, do you have other potential circuits that might be accomplishing this for the head direction network?
So this circuit is very simplified, simplified ones. So the Legion study also mentioned that vestibular nuclei project to another brainstem nuclei for supergranule nuclei, and that was also shown to disrupt the head direction cell signal when it was lesion, and it is connected to vestibular nuclei. That could be another potential circuit, or head direction network. They might use other signal, weight them more than vestibular signal to for the further processing.
Great. If there are any other questions, feel free to post them in the Q&A. Otherwise, at this point, we're going to move on to our final talk. This is going to be given by Olivia Levitt. Apologize, I've lost my. Her talk is titled "Prosthetic Stimulation in the Axis Corresponding to Support Surface Perturbation Improves Postural Control in a BVL Primate."
Good afternoon, everyone. I'm Olivia Levitt. I'm from the Cullen Lab at Johns Hopkins University, and I'm going to be presenting some preliminary data on prosthetic stimulation in a primate model with vestibular loss. The vestibular system consists of 5 organs in the inner ear that sense the angular velocity and linear acceleration of the head. Signals from the vestibular system are carried on the vestibular nerve by regular and irregular afferents to the vestibular nuclei, which contribute to visual stability, self-motion perception, and posture and balance. In the absence of vestibular signals due to vestibular loss, all of these functions are impaired. An emerging approach to treating vestibular loss is the vestibular prosthesis, which uses a head-mounted gyroscope and a microprocessor that uses a mapping function to translate the angular velocity of the head into a stimulation pulse train that can be delivered to any or all of the semicircular canals in the vestibular labyrinth. Importantly, the electrodes primarily activate irregular afferents, which also happen to be the primary contributors to posture and balance downstream. One such prosthesis is currently in clinical trials in humans at Johns Hopkins, and it's been successful in restoring patients' confidence moving through the world, but some aspects of their performance require further development. So here we're using a rhesus macaque model, which allows us to not only probe externally measurable factors like posture, as we do here, but also probe vestibular pathways in the brain and better understand how the prosthesis may be interacting differently with the brain than the native periphery.
In humans, balance is often measured by applying perturbations to the support surface. When the frequency of perturbations is low, the head and center of mass remain stationary relative to the platform, while at high frequencies, the head tends to remain fixed in space. The strategy switch from what we call platform fixed to a head fixed strategy results in high gain at low frequencies, followed by a switch to low gain at high frequencies. In humans, this strategy shift occurs around 0.3 hertz. In monkeys, we expect to see a similar strategy shift, although the frequency the strategy shift occurs at may change depending on the animal's biomechanics. For this study, I delivered sine wave perturbations in the roll axis on a hexapod motion platform while the animal stood in a chamber. The monkey's motions were measured with a force plate with an IMU, as well as joint tracking using markerless pose estimation with deep lab cut. Here's an example of the traces that come out of the data once it's synchronized in post-processing. As you can see, the data is very multimodal. There are 12 analog type inputs as well as up to 20 joints being tracked. Here I'm focusing on just the head roll because it's easy to connect to postural sway. Here is the sway response in a normal animal. In a monkey with an intact vestibular system, the gain curve for postural sway mirrors that of humans, although the strategy shift here seems to be occurring at around 1 hertz rather than at 0.3 hertz as it does in humans. In the case of vestibular loss, a monkey had higher gain over all of the frequencies, which means that there was more postural sway at all frequencies, which we would consider to be poorer posture and poor postural compensation. With stimulation in the posterior canal, which senses movement in the roll and pitch axes, there is a slight decrease in gain from bilateral vestibular loss in most frequencies, which indicates improved posture. This is particularly true at high frequencies. This makes sense because the posterior canal is sensing movement in the roll axis, so any movement that is sensed would help contribute to improving postural compensation. In contrast, when the horizontal canal is stimulated and the horizontal canal senses movement in the yaw axis, there was much less postural improvement overall, and particularly at low frequencies, the animal tended to sort of tip over as the perturbation reached its peaks. In summary, the vestibular prostheses are an emerging treatment for vestibular loss, but there's opportunity for improvement. Rhesus monkey postural responses resemble human postural responses, but with a higher strategy shift frequency, which is as expected. Bilateral loss does cause increased sway across platform perturbation frequencies, and vestibular stimulation appears to at least partially restore posture, particularly at low frequencies. And with that, I'd like to thank my colleagues in the Common Lab.
Great, thank you, Olivia. That was really nice. It's amazing how, like you said, multimodal the data is. So seems like really interesting stuff. So I guess I'll ask one question that was kind of sticking out in my mind. Is the translation between how you think that the postural stability should work, as in, are the monkeys going to be doing the same thing that a human would be doing, because monkeys aren't bipedal and they have a tail?
So one of the things that was really interesting doing literature review for this work is that most human, as this image shows back here, most human work is done in the anterior posterior tilt, so pitch tilt. And because our base of support is sort of horizontal relative to our shoulders, that makes sense. I decided to change to roll because that would be the primary unstable axis in a monkey model because they're quadrupeds.
Cool, that makes a lot of sense. So we've got two questions. I believe both are one is definitely panelist asked, so I'll ask that one first. What insights do you gain from your results that could be incorporated into the pre-existing vestibular prostheses?
In this case, I think the biggest thing to think about in terms of prosthesis development is to ensure that all of the canals are being stimulated, because early prostheses and early work primarily focused on the yaw axis, because that's where a lot of the research has been done, and also it was thought that head turns would be a primary perturbation for someone with vestibular loss. But given these results about posture, it's important for maintaining posture and probably gait as well to have stimulation in all of the semicircular canals, if not also the otoliths.
Great. So Robin also has a question, so you can unmute yourself and turn on your video on.
Okay. I'm wondering if you've tried to relate that difference in the transition frequency between humans and monkeys to the height of the center of mass. Any way quantitatively?
I haven't yet, but I'm assuming that because it's a shorter pendulum, it would be because of the lower center of mass and the shorter limbs.
Great. And Omid has a question as well. Do you expect the performance of unilateral vestibular loss animals to be closer to normal or bilateral with vestibular prostheses? So you go ahead.
Because the prostheses are generally implanted unilaterally, I would guess that with a perfect prosthesis, a bilateral loss animal with a unilateral prosthesis would perform closer to a unilateral animal. But a unilateral animal would likely not have quite as good posture as a bilaterally native or a normal animal.
Great. And then Richard's going to ask the next question.
So we have another question from Yvette, who asks, do you think regular afferent stimulation would be useful and or integrated with irregular afferents in the future? And while you're at it, I wanted to ask this question because my question was, could you tell us a little bit more about the hardware of how you actually conducted the stimulation and could potentially produce regular?
The irregular afferent stimulation, I believe, is due to the hardware. I'm not entirely sure. And it's also has to do with the just higher excitability of irregular afferents in contrast to regular afferents. Because regular afferents are more involved in velocity storage and visual stabilization, I think it would be helpful to also figure out a way to stimulate regular afferents at some point. But right now, the way that the vestibular prosthesis works, it's unlikely that we'd be able to do that.
Okay, John had a question. Did you check VOR gains? How long did the monkeys get to habituate to the prostheses?
So in this study, I didn't personally check VOR gains, but the monkey that I used had relatively low VOR gains relative to normal. I'm not entirely sure of the exact numbers, but VOR gains were checked.
Great. So we have time for a few more questions. If anybody has those, they can post. Yeah, I'm really interested in that regular versus irregular afferent stimulation. That's very interesting. Oh, here comes John has another question. What lesion was it? Canal plugging? Question mark.
The lesion was introduced in the process of implanting the animal, so the electrodes themselves plug the canals.
Robin says, have you tried to estimate COM with deep lab cut or IMU? Would the results be different from the head in monkeys?
I'm currently working on trying to get either an IMU or the center of mass measurements with deep lab cut. One way that I have tried with one of our more cooperative monkeys was having a vest with an IMU on the back of the monkey in addition to on the head. I would anticipate that it would be a slightly more stable signal just because the monkey does shake his head and stuff during the experiments, but I think the head is a decent analog for now, and it's just much easier to fit an IMU to the head.
Okay, with that, I think there's a few more questions, but I think we'll just wrap up. So I want to thank the panelists for this session. It was a bunch of really great talks, and thank you to the audience for being involved in the discussion. And the panelists, if you, or Olivia, if you can take note of the questions that are in the Q&A right now, you might consider following up with some people afterwards. Otherwise, thanks, and we'll see you guys at some later sessions. Okay, so hey everyone, I'm Adam Steele. I'm a postdoc at Dartmouth College in Hanover, New Hampshire, USA. I've been told that we have a few moments as people trickle in. So I'd just like to use that time to tell all of anyone who's in the US right now to make a plan to vote. Election day is next Tuesday. And it's really, really important. If you've already got a plan to vote, or if you please contact a person that you know and make sure they have a plan to vote. And if you're not in the US and you have US colleagues, get in touch with them and make sure they have a plan to vote, because it's super important. So with that said, we have 3 great thought-provoking talks in this session about the intersection of neuroscience and society. And I'm really looking forward to hearing them. As a reminder, we have the chat where you can post messages and clarification questions. Just make sure that you have the messages set to panelists and attendees so that way we can all see. If you'd like to introduce yourself now, you can use the chat to do that. So just introduce yourself if you're there. Also, we have a Q&A tab for questions. You can post at any time and upvote your favorites. For those of you on Twitter, you can tweet about this session. And if you do, use the hashtag NMC3. So I'll get started here by introducing our first speaker. That is Katie O'Neill. She's a master's student working with Matthew Apps at the University of Oxford. And she's going to be giving a talk titled Science Fiction and Folk Knowledge in the Brain, Insights for Science Communicators.
Cool. Thank you for that lovely introduction, Adam. So to kick this discussion off, I'm going to start with a brief discussion of the general goals of neuroscience communication for non-expert audiences. So we have a couple different reasons we might want to communicate scientific findings in general. could be to correct misconceptions, both in science, so that persistent myth that we only use 10% of our brains, might want to get rid of that one. Also in the process of science, how it's done. So the misconception that scientists are all old white men who exclusively wear lab coats and have very big frizzy hair. That's a misconception I'd like to get rid of, though I am working on my own frizzy hair. It'll be there soon. We also want to encourage participation in the scientific process, be this through participant recruitment, encouraging students to study neuroscience and psychology. We also want to encourage people to think critically about neuroscience justifications for policies and product design in the world, something along the lines of EEG attention tracking headsets in elementary schools to make sure that kids are always focused on their work. And one of the reasons I'm particularly interested in science communication is to provide additional context for questions that people have about the mind and brain. These questions could be personal, so whether memory training works, whether if they have a genetic predisposition to Alzheimer's, they should start memorizing decks of cards. These could be professional questions. So, oh, I've read that interviews introduce bias. Should I use brain scans to recruit the smartest employees? And I'm just going to come out and say, no, you should not do that. These could also be socio-political questions. So if people, especially in the contemporary American political landscape, have been hearing more and more about prison abolition, but are worried about what might happen with psychopaths, understanding the contemporary scientific knowledge of what psychopathy is and how it operates would be particularly useful. And in order to answer these questions and promote these goals, we need to understand how people come to understand the brain. And understandings of the brain come from a lot of different places. Some come from science classes. I don't know about the rest of you, but in elementary through high school science classes basically just told me that the brain does stuff. People learn a lot about what brains do by having a brain, noticing the things that it does, talking to others. I know that sounds like somewhat of an obvious one. Apologies for my bed back there. This is the world of Zoom. But talking to others, there have been a lot of conversations on Twitter recently about people discovering that they have aphantasia, which is the inability to produce mental imagery, by realizing that others actually could imagine an apple or a beach or something in their head. And this is true for a lot of non-neurotypical communities, especially on the internet. And then a third major way that we learn about the mind and the brain is through media representations. And this is where science fiction comes in, particularly stories about artificial intelligence, aliens, advanced animals, minds that are like ours, but are different in some fundamental way. So let's talk about what kind of conceptions of the mind and brain that science fiction books and movies promote. So since this is a talk that involves science fiction, I'm contractually obliged to talk about Frankenstein, as it is considered the first novel that could be labeled science fiction. I'm also going to note that there are previous science fiction novels before this. If we want to take a look at Francis Bacon's New Atlantis in the utopian tradition, Margaret Cavendish's description of a new world, got some lovely Hollow Earth stuff from the 1600s and 1700s. Let's talk about Frankenstein. And I'm actually not going to talk about the creature first. First, I'm going to talk about in the very beginning where we have Robert Walton discovering Victor Frankenstein, the scientist's frozen body, where he is thawed out and prepares to tell his story. And the central conceit here, with the idea of cryogenics, the ability to freeze and thaw people, that is related to the way the creature is created, is that there's some animating force that can bring a brain from a hibernating state to a live state, that frozen brains can be restarted as long as the starting position is maintained. And this promotes the idea that the brain is, on a certain scale, a finite state machine, that there are limited deterministic biological processes of the brain that are inherently plausible and restartable as long as some central state is maintained. And today, we would see that as an implication that the brain shares fundamental properties with a computer. However, if we look at Frankenstein in historical context, it came out 20 years before Charles Babbage's original mechanical computer, many years further before Ada Lovelace's famous first computer program. So what you see in the time surrounding the publication of Frankenstein is descriptions of the mind and body represented in terms of some sort of contemporary technology with wide popular recognition. So you can see Charlotte Bronte in Shirley with her narrator talking about herself as a human mill with a heart that is a boiler. And you don't see comparisons to technologies that share the same mathematical underpinnings of this finite state machine. So you don't see the brain described as a train semaphore, you don't see the brain described as a turnstile. But as the computer has gained cultural primacy, and the von Neumann computer specifically has gained cultural primacy, its metaphorical tendrils have had a greater impact on the ways we think about the brain. So the metaphors of the von Neumann computer have affected our conceptualizations of the brain in three main ways: our understandings of how the brain works, what the brain is for, and how we as ourselves relate to the brain. So with relation to how the brain works, the von Neumann computer promotes an idea of modularity. Don't worry, we're not going to get super into photo here, but we see things that promote an idea that separate individual parts of the brain do individual things, are replaceable by each other. So we see things like in the Minority Report, which is originally a book and also a movie, we see something where someone's eyeballs are removed and replaced. A part of the central nervous system, admittedly an outside one, that is easily replaceable. We also see a form of modularity with the brain in response to, or in relation to the body. So we see this in examples like brain uploading to computers, brain replacements, brain transplants. So in movies like The Matrix, Avatar, Captain America, Winter Soldier. And the important thing here is that the brain is considered independent from the body. And what's particularly interesting about this to me is that this metaphor has become more and more popular as our ideas of work and self have become further abstracted from the physical world. So most people can understand and explain to you how a hammer works technologically. By the time you get to a bicycle or a toilet, most people struggle. And when you get to neural nets, these are very hard to explain in terms of physical phenomena, which means it's easy to separate the functions of the mind, the functions of cognition, from the embodiment in a human physical body. One of the other things that this metaphorical framework has told us about how the brain works is that language is a computer program. So you get a lot of classic science fiction works steeped in the tradition of Sapir-Whorf, of linguistic relativity and linguistic determinism. You get Samuel Delaney's Babel 17 and Languages of Pow, both of which have language as a force in war, as a way of changing people's way of thinking to bring them towards a certain ideological position. Of course, you have Snow Crash, which introduces ancient Sumerian as the operating system of the human brainstem. You have books like 1984 and Sophia Ray's lovely short story collection, Everything is Made of Letters, that talk about how language degradation can be used as a tool of authoritarianism by making people think differently, thinking in line with the state. One of the other ways this metaphor is constructed is in understanding the brain as readable, writable, and erasable. And it's worth noting that this isn't necessarily false, it's just overextended, as metaphors often are. So with definitions of brain reading, we can take a look at fMRI, EEG, dream and visual stream decoding. Like, there are definitely ways we can tell generally what's happening in the mind, but it's certainly not as pronounced as it would be as going into a computer address and retrieving a piece of memory. Because humans use generally content addressable memory rather than address addressable memory, like computers do, it's a different retrieval process. With writing, there's actually a talk, I think tomorrow or the next day, about sculpting visual concepts in the mind that I'm very interested to see, related to this idea that writing information into the brain, as would be true for a computer, is possible. There's also a long tradition of erasing memories in science fiction. My favorite example is Men in Black, being able to flash a bright light and having very specific sets of memory erased from people's minds. While certain forms of memory erasure, like blocking certain PKM molecules in snails, is possible, it's definitely limited to a recent range of memory. It's definitely not as specific as science fiction teaches us that it can be, in line with this computational metaphor. So in terms of thinking what the brain is for, This is a big one for taking a look at stories that center on artificial intelligence. So we often see AI as calculators and administrators. Again, in Minority Report, these artificially all-knowing minds can predict crime before they happen. In the Fantastic Four movie, an absolute classic from 2007, Mr. Fantastic promotes the Superhero Registration Act because he's perfected the art of Asimovian psychohistory and is able to predict that billions of people will die unless this happens. Yeah. We also view artificial intelligence as an existential threat, which tells us something about the way we conceptualize the mind as a device for dividing self and other and protecting self over other. That's fodder for a whole nother talk, so I won't get into that today. One of the other things this computational metaphor tells us is the way we should relate to our brains. So one of the things we learn from Asimovian psychohistory, from the Foundation novels, is that brains are individually unpredictable, but predictable in large numbers. We also learn that brains are something that we have and use rather than something that we are. So if we take a look at Lucy, which is one of the big promoters of this 10% metaphor, if we can just unlock the rest of our brain, it will allow us to be all that we are. However, on the flip side, we also get messages steeped in biological determinism, like in The Fifth Element, where character is recreated from a scrap of DNA, fully aware of her culture, able to speak a language, ready to go. We also have the idea that—
you have one.
Yes.
I just want to let you know you have 1 minute.
Okay, cool. We also have the idea that brains control our behavior, implies a causal direction. We can talk about that more in questions. How should this guide our science communication methods? It introduces specific places to check where in your audience's assumptions about the brain and the language we use to describe it intersect. Woo, done. There we go.
Great. So I will give you one quick question and then we'll move on. So the question is, I am wondering how you think science fiction can be made more accessible to more people. While I agree that it's a good way to learn about the brain and technology in general, it seems like it's too niche to attract people who would benefit from learning these concepts. And you can give a short answer on screen, and if you want to do a longer answer, you can post in the chat.
My short answer is changing the trope that science fiction is only understood by the same nerdy old men we think are scientists, because that's absolutely not true. I can talk more about that in the comments, though.
It's a great answer. Great. So now we'll move on. That was awesome. Thank you so much for that talk. Our second speaker is Alexei Guzy, who is an unaffiliated independent researcher working on metascience. He's most interested in the organization of life science and the ways people get selected in and out of academia. He writes on his own website and blog, and I'll post a link to that in the chat. His writing has been mentioned in the popular media, including BBC and Wired. And today he's going to be giving a talk titled The Structure of Life Science Academia.
Let me start sharing the screen. So I'm Alexei, and I'm going to be talking about the structure of life sciences academia. And I think that there are a lot of problems in how academia is structured, and I feel like I'm a bit less idealistic than, for example, the organizers of this conference, in that there are certain, a lot of issues, for example, of editorial control, as was pointed out in the introductory session. And the organizers of the conference, for example, decided that the way to deal away with editorial control would be to just anonymize all of the talks and let the algorithm decide what talks to suggest to us. But it seems to me that you basically outsource the editorial control to algorithm in this way, and people just rely on different ways to source what they're going to be looking at. But like the editorial control still is going to be there, and you still will have to make decisions about how to design it. And it's a question of whether it's going to be people or algorithms or some mixture of it. But this is a more difficult problem that it appears to be, and like this is one of the reasons why you like can't really just go and destroy all of the journals, for example, and substitute it by archive. But anyway, so my talk is I'm going to be focusing on three like big points in my talk. And the first one that I'm going to cover is the structural rigidity of academia. And one of the ways in which I think academia is very surprising in that like science is built on us proving like past scientists or demonstrating them to be wrong, and us discovering that we were wrong about things. And at the same time, the only way to advance in academia is via getting approved by people who are more senior than you are. If you want to get into grad school, you need to get recommendation letters from professors. In order to get a postdoc and a faculty position, you need recommendation letters, and you need to get published in journals and get approved by people who are more senior than you are. And in order to get a tenure, you need to get even more recommendation letters, so that people would confirm that you're doing interesting work at the frontier of your field. And this contrasts very strongly with how like world outside of academia works. And like if you think about industry, and if you think that some business is functioning poorly, or there's some people are wrong about something, then there is you can go and like raise in some initial amount of capital from a really large selection of venture capitalists or banks who can give that to you, and then you're going to start serving customers. And once you start to serve customers, they're the only people you're dependent on, while academics are like, well, first of all, it's not even clear who like the final customers are, whether it's other scientists or whether it's the public or whether it's the funding bodies. And academics are forever dependent on people approving their work essentially in order to continue to do their work, which I think is interesting. And yeah, as one physicist I was talking to recently told me that to get a faculty job, you need two things to happen. You need someone who is lobbying for you specifically, and you need everyone else to find you unobjectionable. And this is also interesting because like people who are advancing science the most often are people who are objectionable, it feels like. Then there are like a lot of other ways in which the structure of academia is surprisingly rigid. For example, companies are often founded by several people, like maybe someone is more technical, someone is more social, and people team up and found a company and do the things they're stronger in. And in science, like you are a PI and you're the one head of the lab, you have to fundraise, you have to do science, you have to manage people, you have to hire people, you have to be good at everything. And I would guess that this ends up selecting out a lot of people who would probably be like really good scientists, but who would not be very good at managing people or hiring people or teaching. And yeah, and if we think about the scientists in the past, it seems that quite a few of the greatest scientists were like maybe a little bit autistic or just like autistic, and they would probably like struggle a lot if they were forced to become a PI. And this is also related to the next point, which is that like because everything is set up, all of the funding structures are set up to make hiring grad students and postdocs, being it very cheap to hire grad students and postdocs, there is really no incentive to ever hire research scientists. And so people who would just make really good research scientists, but not very good PIs, end up having to leave academia effectively. So the next point that I think is very misunderstood by people outside of academia is the risk aversion. So NIH is the primary funder of neuroscience and of life sciences in the US. And naively, it seems that NIH is this giant bureaucratic organization that is really risk averse and really likes to fund incremental research and really doesn't like anything too innovative. But looking closer, like at this, it seems that like even though this is kind of true, in reality, like people find ways around this. And for example, I guess the biggest way is that everyone just lies to NIH, and like people just like say they are going to do one thing, and they are like, maybe this is part of what they're going to do, but then they're actually going to do the projects they really want to do. Sometimes people like you do the experiment and you have your results, and then you're about to publish them, and you write a grant like as if you're going to perform the experiments, and then you use that grant to do something interesting. Or sometimes like, so related to this meme, if you want to do something really exciting in some basic research project, I think neuroscience is especially guilty here, because like there are a lot of brain diseases, for example, and we haven't really solved any of them, but you like kind of have to say that, or I'm going to work on this project of how brain circuits in mice models of autism work, and like the project probably likely doesn't really have much to do with autism or with depression, at least in the near to medium term, but you still have to like make it seem as if it's not just a basic science project. And then of course, like risk taking is actually encouraged, because like if you're a student, you really need to get publications at Cell and Nature and Science and other top journals in order to get faculty positions. And these journals like really like flashy results, which often require you to work on something that is risky. And of course, PIs also have some portfolio projects, and have some projects that are more risky, some projects that are more likely to work out. But this is also, especially the point about lying to NIH, also I guess continues the point about selecting some people out, because like lying, like they need to hype up the research and to like invent ways to explain it in order and to make it fundable. Does also like just select people out who find this unpleasant or who are not very good at this, which also probably introduces some structural differences in how science is made versus how like it should be done. Then there is a question of like cooperation in science, and the thing that I think I was most surprised to by like how often people mention this to me, because like again, naively, you would have people usually have this idea that industry is like cutthroat and there are businesses competing and everyone is individualistic. And then when I like started talking to scientists were like, well, and then like I thought that, and then I went to grad school, and it turned out that like grad school is usually a really lonely experience, because if you're a grad student, like and you want to become an academic, then like the only currency that you have are first author publications. And if the only real currency is first author publications, then like it's like really difficult to cooperate on projects. And even if someone has a project that is maybe as interesting as your project, or maybe more interesting, but you're not going to be a first author in it, like cooperating on it and like working it, you're still probably not going to give that project as much time as in some utopic system you would. And in contrast to industry, where like, especially if you're working in a startup, like, and everyone has equity, everyone has like, it really is like bound by this in a very tangible way, and in a way that is kind of impossible. This is kind of impossible to do in science. Yeah, and the way I think to try to pull this all together in some synthetic example of we can try to imagine some like a grad student who like is interested in Alzheimer's disease and who decides that, well, right now other hypotheses are more accepted, but it seems that like 20 years ago, beta amyloid hypothesis was so dominant that you really couldn't work on other hypotheses. And if you were a grad student 20 years ago who's interested in Alzheimer's, and somehow you got, you decided that this is not the right way to think about this problem, there really wasn't that much to do. There, you couldn't just like go around the field the way and just like work directly on this. You would have to work with, like, be a grad student of someone who thinks that this is the right hypothesis. Probably you would like somehow have to figure out how to do that for many years, and like to work on projects that you think are probably wrong. And if you just like somehow graduate and try to get funding, then it's going to be really hard, because like people, senior scientists, think that like this is the right way to approach this problem, and they're going to be sitting on NIH panels. And this is also just like really, really hard to do. And if you try to do something differently, you're like you're essentially trying to destroy their careers if all they spent the last 20 years doing is publishing on this hypothesis. And
Alexei, you have 1 minute left.
Yeah, and as a and as a result, it seems like in a lot of ways science like needs to be like yes and like in order to succeed you have to be like original but not heretical. And yeah, there is a quote about like people trying to work on the field that is captured by other people in the and succeed, unfortunately. But yeah, I think I'm basically done. And I have a lot of more things on this topic on my site, and you can continue reading in my essay. And there's a bunch of memes, but we don't have time for them. Yeah.
All right, that was a great talk, Alexei. Thank you very much for that. So I guess I will ask a question. Short answer question. You can post more in the chat or on Twitter, however you would prefer to respond. In general, I agree with you on most of the points you're making, but I guess, do you think that that science right now is structured based on like the Kuhnian kind of revolution perspective, whereby most of us are toiling at a question or like, and not going to make any progress or make very incremental progress until one person makes, or there is one major scientific discovery that precipitates a revolution that changes how we're all thinking about things. And then everyone will begin toiling away at that level of understanding until we reach the next revolution. Not to say that that's like a good thing, but
so the question is whether I believe that this is how science works.
Whether, yeah, I guess it is kind of whether or not that's how science is working. Whether there's an alternative model.
I suspect that this is at least partly how science works, and like that it's like at least it's discontinuous, and like we discover some things that change our perspective more, and we discover some things that change our perspective less. And sometimes we discover things that like demonstrate a lot of other things we believe to be true to be false, and people start to building the new thing out. But yeah.
Yeah, unfortunately, like the structure of science sometimes makes it seem like whose revolution we follow is who's most popular.
Yeah,
that maybe is the problem. Okay, so we have to move on, but this is a very fun session. I'm really enjoying your talks, and I think the last talk is going to be great as well. It's going to be given by Josef Mészáros, a postdoctoral fellow at Columbia University. He can be found on Twitter at NeuroJojo. He would like it to be mentioned that he's an active member in the postdoctoral union at Columbia University, and was a member of the negotiating team between the postdocs there at Columbia and the university. So please contact him if you're interested in anything related to that work, which I think is a really great idea and that you guys are pursuing. So with that, I'll let Josef take the stage.
Hi, everybody. Thanks a lot for the great talks, Catherine and Alexi. I think my talk will follow up nicely with a lot of the themes you raised about the stories we tell about each other and the stories that other people tell about each other and their brains. So here I'll begin my talk. It's about violence, neuroscience, and the legal system. It is a little bit of a Halloween themed talk. So for the international audience, Halloween is like this holiday in the United States where people dress up and try to be very scary. As it turns out, every day in America is pretty scary, and it's kind of like getting scarier. So as I begin, I'm going to talk about kind of two stories. One of those stories is that we have this legal system in the United States and much of the rest of the world where we have sticky concepts. And I don't think that neuroscience is going to challenge a lot of those concepts. So some of those concepts include vengeance, blame, responsibility, harm, and the idea that there's a culprit and the culprit is the one who did the bad thing. So I'm going to talk about that theme first, and then I want to talk about another theme where neuroscience might possibly influence the legal system by working with and not against these concepts. So first of all, a question for us, especially like younger people in this group who are, shaping their careers, should we use neuroscience? And I'm not saying results, but intuitions. Results can be very hard to obtain, but we want to put very rigorous standards on our science, but we develop a lot of intuitions doing neuroscience. Should we use these intuitions to influence the legal system? And I think some of us may feel timid or modest about our contributions and will say, no, that's like different. We're doing science, politics is separate. But I think it's worth considering that fields like economics have had an outsized influence on the way that we think about the law. And no one really said, hey, economists, please change the law. They just went in there and did that. So, don't feel too shy about using neuroscience as well to influence the legal system. So where does this story that I'm going to tell begin? Well, it starts around 2004 when very influential article of the Philosophical Transactions of the Royal Society B published an entire issue about law and the brain. And in that issue, Robert Sapolsky, who's a very prominent neuroscientist, authored an article, The Frontal Cortex and the Criminal Justice System, and said, neuroscience could and should do away with criminal law as we know it. Very big pronouncement, right? The Economist a few years earlier said, genetics may threaten privacy, kill autonomy, do all these things, but neuroscience could do all of that first. And yeah, it is The Economist, and they do say really weird things sometimes, like that autistic brains are wasted. So I'm not sure how much we trust The Economist to really predict the future. And in any case, let's take a look at whether either Sapolsky or The Economist or the tons of other people who went down this line of reasoning were right. So there's this meme, I like it, how it started and how it's going. Okay, so how it started. Criminal law revolution. Oh, publish tons and tons of paper in law and neuroscience because we're going to figure it out. And then throw, of course, tons of money in it, right? Because no one's going to do this work. Alexi kind of hinted at this unless we fund it, right? So the MacArthur Project, because this isn't really an NIH thing, it's kind of like an intersection of two fields. So it has to be kind of a quirky thing. And the MacArthur Foundation is this big foundation in the United States. You may have heard of the Genius grants and stuff like that. That's the MacArthur Foundation that gives that out. So how has the, at least United States legal system interpreted some of the neuroscience contributions? Well, we used to have a thing called the insanity defense. If you were judged to be legally insane, you could get basically not guilty verdict, right? You were insane when you did the thing you did, so you're not guilty. Well, actually, Kansas was like, no, insanity doesn't exist, whatever. So if somebody has a schizophrenic delusion that, you know, God told them to kill their neighbor, that doesn't get heard in trial. That doesn't get heard by the jury. That's not really relevant. The relevant thing is that this person killed their neighbor. Okay, so whether you have schizophrenia or whatever, it doesn't matter. The point is that you killed someone, so you go to jail for life, or you get the death penalty. It's also possible. And if you think that's just a weird Kansas thing, the Supreme Court heard a case about whether this was a constitutional law, and they decided it's perfectly okay. 6 members of the court, including some of the very conservative members that we like to always bash on, and also Elena Kagan, rolled this out. So now any state within the United States can just eliminate their insanity defense. So we went from thinking that we were going to use neuroscience and all these lovely, elegant results that we have to change the way that people think about guilt and responsibility and culpability. And the Supreme Court took that football and just spiked it and said, you know what, actually, like, we have other things that we care way more about than your neuroscience, or whether schizophrenia involves overactivation of D2 and whatnot. We actually just, you know, if you're guilty, you're guilty. All right. So that was kind of the first part. There's these incompatible notions that make us feel maybe uncomfortable as neuroscientists. So could neuroscience influence the legal system by working with some of these concepts? All right, so the way that it goes is, you know, neuroscientists, they focus on the brain of the offending individual. So if you committed a crime, the neuroscientists are like, oh, well, there's no free will, or, you know, you had this gene variant of MAO, and it happens to correlate with violence. Or, you know, addicts, they have these reward pathways, they get hijacked, et cetera, et cetera. Well, okay, but you could also do this other thing where you looked at the brain of the punisher, not the brain of the person that is on trial, but the brain of the person deciding. And that's what Oliver Goodenough proposed also in that Philosophical Transactions article. And that article, you could look it up, it's called Responsibility and Punishment, Whose Mind a Response? Okay, so neuroscientists, you know, they're saying we're a product of like molecules and circuits, but I'm a jury member, right? You know, I have to think about vengeance, blame, responsibility, harm, who's the culprit, et cetera, right? So what do I do? Okay, well, the accused brain, it has like, you know, molecules, neural circuits, it has like pathways and stuff. The punisher's brain, the punisher, who's the jury, they want to think about like who did the harm, who is the culprit, who's responsible, et cetera, right? How do these two brains interface? Like, how can you think about them, you know, thinking of each other? That's basically what we have in this situation, right? So I want to think of a hypothetical, right? We do a lot of like opto hacking in all of our neuroscience experiments. You know, you get this pathway activated and then the animal goes left or bites a cookie or whatever. Okay, let's say that you have a situation where you have an evil neuroscientist who pushes a button and it activates a part of the brain that causes an individual's neural pathway, maybe it's in the hypothalamus where the aggression circuits are thought to be located, to become overactivated, right? And then that person's hypothalamic aggression circuit becomes really excited, and they do some kind of behavior, violates norms. Maybe they hit someone or they kill someone. Okay, you know, I'll put that to the audience right now. Like, who would be guilty, right? I mean, I think we all say that it's like the scientist who started this thing, right? It's not the person, like, but nevertheless, right? Like, these cases can come up. This is not made up, right? So if you are interested, there was a clinical report in an issue of Neurology. I'm not going to go into all the details, but basically they used a stimulation. They didn't expect this to happen, but they stimulated in this hypothalamic area. And what happened was that within a few seconds of the stimulation, the subject who had his brain being stimulated became very aggressive and violent, and it wasn't confused. It wasn't like a seizure. It was like very, intentional or, just like directed aggressive behavior. So let's say that that individual hadn't been restrained in time and he assaulted someone in the operating room. Do we want to find him guilty of assault? I mean, it really doesn't seem like we do, right? Okay, so how can we make the legal system sort of compatible, or in what cases does this even come up? I think in this situation where, and I said this is kind of scary, there are these third parties, right? There could be a third party, and the involvement of a third party could allow neuroscience to come into the legal system. And when you have a third party, you have a culprit, you have an evil act, the shocking the brain or whatever it was, and you can punish that third party. So a lot of the aims of the legal system can be achieved by sort of thinking of this in this neuroscience context. And you can rehabilitate, take the third party away, take the opto or whatever, take the stimulation away, and maybe the person will recover their normal, pro-social behavior. So there are some kinds of neuroscience work where you do.
Joseph, I just want to let you know you have one minute left.
Perfect. So there are neuroscience kinds of neuroscience work where you do see that you can shift blame to a third party in these kind of criminal contexts, right? So there's Deborah Deno, and she's looked at how lead poisoning very, very strongly predicts whether someone goes on to be a violent offender. There's other studies that look at CTE and how that increases the chances that people commit violent acts. And I also have a study, a very long law journal article looking at how people who are experiencing intermittent partner violence will commit crimes. And maybe we should think about the third party who is involved in that. So thank you. These are just acknowledgements and further reading.
Great. That was really, that was another great talk. This is one of those sessions where I wish we were important so we could all go to the pub after and chat. So I appreciate that. If anyone's got any questions for Joseph, please post them up. We've got a couple, we've got one question in the Q&A that's pretty relevant, Joseph, I think, which is how can we make the legal system comprehend neuroscience concepts, which I think is really at the heart of the issue. It's one of the hearts of the issue, maybe.
Yeah, I think you have to realize that the legal system is us, right? And no matter how much neuroscience you read or absorb, it's really hard for you as an individual to be completely okay with someone who stabbed your best friend or, did something really horrible to you. So you have somehow to take the neuroscience that you know and integrate it with the kind of moral concepts that you have always had and that civilization and societies often have, been created around. So, neuroscience that says, oh, there's no free will, everyone is actually just like this molecular machine and so on, that's just not going to really it's not going to land well. It wouldn't land well with you when you're, considering your friend, and it won't land well with trials and jurors. So I think, just making a better connection between the legal norms and the neuroscience studies would help.
Yeah, I really liked the visualization that you had of the Punisher and the, I can't remember the two phrases that you used, but the accused and the Punisher, because it really drives home the point that actually we are talking on two very different levels, and integrating across those levels is hard when you're trying to write a talk to get people interested in your research, but it's way it's way more difficult, and the stakes are much, much higher when you try to make that integration at that level, which is jumping not only to oh, I need to create human interest up to societal implications. And that's just a really, it's just a really tough nut to crack there. I think that's a real, an illustration that really drives home the point. Unfortunately, we have to wrap up. We are already over time, but I think it's because these were just such great talks, and I'm wish we could all give you a round of applause. But thank you all for listening, and we'll look forward to seeing you guys at some talks later in the future.
Go ahead.
Yeah, and for the back end, it's just, as long as everything is on schedule, everything is going to go okay. Now I, have made the connection to the live stream as well. And by the way, this is going to be recorded on the cloud if you want to make local recordings for your on your computer for your purposes, for your lab website, etc. It's also your choice, totally fine. Yeah, I think it should be pretty okay. And maybe I can launch it and share the screen with the splash screen that says the event's gonna start in a few seconds.
All right. So I'll just introduce the session briefly, and then Kirthi, I'll have you take it away.
Sounds great. Hi, Deborah.
Hi, I was trying to say yes. I consider you one of my predecessors for the for the project, even though we've never met in person.
This is Billy. You've emailed him multiple times before, I think.
Yes, I remember.
Yes. Oh, wow.
You guys.
Very good. Hope everything worked out with whatever I told you.
I hope the data update isn't disappointing today. Hopefully, I took it somewhere. I tried, guys.
It's all good.
Looking forward to your talk.
Thank you.
All right, should we count down?
Yeah, I guess we could.
Okay, great. And we'll let the people roll in.
Yes.
Should I start sharing now or wait?
I'll let you know.
Okay.
I'll give you the cue to start, and then you can share at that moment.
Sounds good.
Yep, I will start the webinar now.
Great. All right, do we see, so we've started, right? Live on YouTube, and the webinar started, and I see participants rolling in. Great, so hello, welcome, good morning, and good night to everyone. Welcome to the first day of NeuroMatch 3. We have a very exciting session today. First, I'll do introductions. I'm Medina Sarvastani. I'm a postdoc at Max Planck, Florida, and I'm your front end host. My back end technical host is Taha Morshidzadeh, who's helping with the technical side of things. And just a few reminders before we get started. Please add your questions for the speakers in the Q&A. And importantly, please upvote each other's questions so we can just sort of use the popular questions if time is limited. And feel free to use the chat for any other relevant comments, discussions, if there are papers and references you want to post. And finally, so much of this conference has happened on Twitter, so please help us get the word out. Take screenshots of cool slides of the talks, post those on Twitter, post your thoughts and comments on Twitter, and help us get the word out. And the hashtag for that, I'll put it in the chat, is NMC3. Okay, great, and let me put that in the chat. All right, so this session is focused on understanding Rett syndrome across scales ranging from movement behavior to circuit plasticity. We have 3 talks, each a total of 15 minutes, including Q&A. Our first speaker is Kirthi Krishnan. She's an assistant professor at the University of Tennessee, Knoxville. We'll put her lab website and Twitter handle in chat in a minute if you'd like to learn more about her work. So Kirthi, please take it away.
Great, thank you. I am. Are you able to see my screen now?
Yes, great.
All right. Okay, so thank you for the opportunity and for the introduction. All of our insights related to this title are due to our work on Rett syndrome. The next 2 speakers in the session will also be discussing Rett syndrome and mouse models in general. So I'm going to start with that as an introduction. Rett syndrome is diagnosed as a neurodevelopmental disorder that affects 1 in 10,000 girls and women worldwide. Many girls with Rett syndrome exhibit typical normal early development and then regress with atypical speech, gross, and fine motor issues. The hallmark of this disorder is this hand-wringing phenotype that you see over here. Many Rett syndrome patients live well into their middle age. It's currently unknown how the syndromic trajectory changes as the girls go through these physiological changes, such as puberty and menopause. Due to these studies in animal models for this disorder over the past 2 years, past 2 decades, from many groups, including ours, detection and diagnosis of early sensory processing is being used as a clinical diagnosis from now. Mutations in MECP2, an X-linked gene, causes Rett syndrome. It is predominantly found in the nucleus in this beautiful punctate pattern that you see here in green. It is found in most cell types, including our favorite, the paravalbumin positive GABAergic neurons, shown here in blue, which are surrounded by these extracellular matrix structures called perineural nets, shown here in red. Ms. Dana Leo, our next speaker, will be talking about these structures next. MECP2 is thought to be a master gene regulator that could affect chromatin compaction and or affect gene expression by associating with repressor or activator compounds. The community as a whole is trying to work on this puzzle of understanding how MECP2 function could relate to the Rett syndrome pathogenesis and symptomology throughout life. So one hypothesis for this connection is this presented here. Based on the studies from many groups, including ours, MECP2 could be considered as an epigenetic regulator of critical period plasticity, not just during early development, but perhaps into adulthood. This is illustrated nicely in this graph over here, where we see the expression of MECP2 increase, which is shown here in red, in relation to the known distinct critical periods for sensory processing, such as hearing, vision, and motor learning. Here, I'll focus on this hypothesis by introducing maternal behavior as a paradigm to study adult experience-dependent plasticity in the female heterozygous mouse model. Parental behavior is a great model to study adult experience-dependent plasticity, as the early developmental critical periods have long been established. With pregnancy and the need to take care of the young, there are new demands on these already established circuitry and established behavioral patterns. We use the alloparental behavior as a model here, where parental care is provided by other females besides the dam in order to focus on adult experience-dependent plasticity mechanisms over changes induced by the pregnancy hormones. Here's a schema depicting our alloparental behavior paradigm. A pregnant female is co-housed with 2 naive virgin females, a wild type and an MECP2 heterozygous female. These are later mates. 3 to 5 days prior to the birth of the pups. From the day the pups are born, which is day 0, till day 5, we perform the pup retrieval assay. During the actual retrieval assay, we start by removing the 2 other adults and any remaining extra pups out of the home cage and habituate the 1 adult and the pups for this particular assay for about 5 minutes in the home cage with the recording setup. Then we remove the pups and prime the adult in the isolation stage. Next, we scatter the pups in the different corners of the cage to initiate this retrieval process. After about 10 minutes of retrieval and recording, we start the process again for the other adults. After day 5, we go through and perfuse the wild type and the heterozygous surrogates, extract the brains, and perform immunostaining and microscopy for PNN structural analysis, which you'll hear about a lot afterwards. So mice perform this alloparental care of the pups very well. Here's a video showing that assay. In this, a wild type female is retrieving the pups that are shown in the different corners here. This is done in a dark box. I'll let you watch for a minute. You can see that she's pretty good at retrieving the pups, finding them and retrieving them to their nest. These are not her own pups. And here's the MECP2 surrogate het, the heterozygous female. I'll let you watch for a second. As you can see very clearly, the het goes from pup to pup, but doesn't actually retrieve the pups to the nest. okay? From a Rett syndrome perspective, this is a really nice assay to look at primary sensory processing deficits, but also multisensory integration, which is important for social communication and higher order cognition. Okay, when we quantify these behaviors, which was published in this work in 2017, we look at latency and errors. Latency calculates the time it takes each adult to retrieve the pups back to the nest. Each circle here is an average latency of an individual mouse over the 3 different days. Though there is significant variability between the mothers and the surrogate wild type, it's clear that the surrogate het has significantly worse performance in this index. And now when we look at the errors, which are metrics for where the pups are being interacted by the mother or surrogate wild type or the het, but then actually not retrieved back to the nest. Again, the 2 other mothers and the surrogate wild type do show some variability in this, but the surrogate het has a significantly worse performance in this metric as well. So in the rest of this paper here, we focused on the auditory component, and I suspect that the last speaker will be talking a lot more about this later on. So when I started my own lab at the University of Tennessee at Knoxville, I decided to follow up on these error metrics and focus on this proximal tactile contributions. The idea was that they are able to find the pups and interact with the pups, but they're not actually bringing them back into the nest. But before we needed to focus on that, we needed to actually make sure that the assay works at the University of Tennessee. And so we decided to redo these assays here with a minor change where we did the behaviors on every single day rather than on day 0, day 3, and day 5, as was published previously. So what we see, first of all, is that the surrogate het do perform worse compared to the wild type animals. So reproducibility crisis has been averted. But we do see this nice behavioral rescue in some areas from day 2 to day 4, which then gets worse again on day 5. This is something that we noticed previously, but we didn't actually hit significance in those data analysis, but perhaps doing this assay on every single day is making this a little bit more clearer for us. Due to these dynamic changes in the latency and the errors, and also that the surrogate wild type is actually getting better throughout these days, we decided to do a systematic frame-by-frame analysis of the retrieval behavior using data view software, which links videos to an extensive coding spreadsheet. The coding was performed by a tenacious undergraduate student, Ms. Parker Stevenson, in my lab, with extensive guidance and mentorship by Dr. Devin Kassenheiser, our collaborator at the University of Tennessee Health Sciences Center. Devin also performed all the statistical analysis from this coding information. I'm going to focus on 2 key data visualizations here. We plotted the sequences of these goal-related movements. We identified about 9 of them, which seem to be very important for this process. The surrogate wild type is shown here on the top. This is for day 0 of retrieval, and surrogate het over here. These are individual animals performing the behaviors. And the x-axis here is the time. So it's about 10 minutes of the retrieval time. And each block here is about an 800 millisecond time frame. So what we can immediately see is that each block, each behavior actually doesn't look the same as another one. So there's a lot of individual variability across different animals. The second part is that the gray, which is the search phenotype, and the retrieval, the full good retrieval, is the pink phenotype. You start seeing those emerge in the surrogate wild type pretty well. Whereas in the surrogate het, as we had previously shown, they don't actually do that process very well. The other parts that we notice is that there's a lot of yellow, which is the pup approach phenotype, and light blue, which is nesting phenotype. So still remember that the pups are scattered in the different corners. They're not actually in the nest. This is the hets that are hanging out in the nets. And there's also abnormal behavior shown here in black, which is mostly jumping phenotype. When we look at these behaviors on day 3 and day 5, we notice that the surrogate het actually improves. Many of them improve in this behavior. But by day 5, they actually get worse again. So the abnormal behaviors are not as much as it was on day 0, but you can still see the pup approach, nesting phenotypes, and grooming phenotypes emerge in these hets on day 5. The wild types by now have completely crystallized this behavior, where you only see the gray and the pink, which is the searching and the good retrieval process. Then we calculated transition probabilities to determine what the probability of seeing a particular goal-related movement was, given that the adult was in a search mode. The main takeaway here is that the big arrows and the thicker lines that you see are the higher probabilities. And you can see that the surrogate wild type is crystallizing this behavior by seeing this gray to the pink transition quite well, with lesser arrows going towards these other 9 behaviors. Whereas the surrogate het, as you might have expected, has arrows going all over, lines going all over, suggesting that the transition probability between these different behaviors are quite variable in these animals. Okay, so in conclusion, it's important to take the individual behavioral responses over time into account. Individual animals, definitely, but per day is also very important for this. And according to the sequence analysis, we would actually say that's true for time as well during the assay. Tactile stimulation by whiskers, face, and somatosensation is very important for this behavior. This has been shown in wild-type animals, mostly rats, in the 1960s and 1990s. And we've also started performing some deep lab cut analysis so that we could start taking these assays with this gold standard that we have from Parker's data set to now start looking at supervised machine learning approaches for automating this purpose further. This is a study example here by Mike Mikins, a graduate student in the lab. So in addition to the people I've already mentioned in my talk, none of this work would be possible without my partner at home and at work, Dr. Billy Lau, shown here, and the rest of the Interpret undergraduates in the lab. I'm happy to take any questions. Thank you.
That was really interesting. Thank you for that wonderful talk. And thank you so much for keeping on time. I know that it's not easy to give your research review in such a short amount. I have a question. It seemed that over the multiple days where you had the color codes of all the different behavioral activity, pup approach maintained. So it's as if they're not learning the futility of their approach, so that they keep trying, like the effort persists. Is that true?
Right. So yeah, that's one way of interpreting this. We classify pup approach as when the adult actually directs their body and their movement, their snout and their facial areas towards the pup. So that's what counts as pup approach, versus search is like a random movement behavior. So we think they are interacting with them. We can see a little bit of whisker and interactions there. But so we think it's actually physical tactile interactions, which doesn't result in this. So that's why we think tactile perception is very important for this process, and that the het animals are somehow being aversive to this condition because of that pup approach phenotype. So they keep going back, but they don't actually finish it because maybe they find the pups aversive or something like that at this point. And actually, Dana would tell you a little bit more about this in the next step.
Okay, that's really interesting. Okay, great, thank you. Yeah, that's perfect timing. If we can, if Dana, we can switch to you and you can share your screen. Thanks again, Kirthi. Great, yeah, we're still very good on time. Our next speaker is Dana Leo. As you might have noticed by now, she's a graduate student in Dr. Krishnan's lab at the University of Tennessee in Knoxville. And you'll see that these two talks will perfectly, actually all three talks, perfectly complement each other. She just entered the postdoctoral job market since she's late in her graduate work now, and she's looking for the right fit. So get in touch with her if you're hiring, and we'll post her contact info and a link to her recent publication in the chat. Dana, the stage is all yours.
Thank you. Hi, so my name is Dana. I'm excited to be presenting parts of my thesis work here today on the systematic and nuanced analysis of perineural net expression in the adult female mouse somatosensory cortex within the context of maternal behavior. So I'm going to be reiterating a couple of key points about pup retrieval in our female mouse model of Rett syndrome that Dr. Krishnan just elaborated on. So one of these is adult surrogate het mice, which are heterozygous for this MECP2 protein expression, are inefficient at the pup retrieval task. So each circle here is an average of the 3 days of behavior for each individual mouse. And each circle, so what we see is the surrogate hets on average take a lot longer to retrieve the scattered pups, which is how we measure this through latency. And then also when they go and interact with the pup, as shown here in errors, they fail to bring them back to the nest. as Kirthi just elaborated on a lot of that information. So in this behavior, rodents are thought to use olfactory and auditory and tactile sensations to identify and interact with the pups during maternal behavior. My specific thesis project focuses on the tactile components of this behavior. So in addition to using the ventral surfaces to provide milk and warmth to the pups where they get tactile input there, they're also using their whiskers and facial areas to engage with the pups. We're now more aware that goal-related movements such as pup approach and pup grooming are atypical in our surrogate hets. This is a surrogate het here. And what you can see is that the mouse is going to the pup and then leaving the pup to go explore the rest of her environment and not really interacting with them. Again, this is in a dark box, so they're not relying on vision for this process. We also know that rodents sense their environment with active and directed whisker movements, as shown in this video here, where you see the protraction, retraction of the whiskers. So we are interested overall in determining kind of the underlying neural circuitry in this aberrant tactile sensory processing and perception within these animals. So the primary somatosensory cortex is a very well-established model for this, where there's a structural relationship between individual whiskers, as you can see in this model, as well as this one, and a specific part of the cortex called the barrel field cortex, which is part of the somatosensory cortex, where each whisker aligns to a very specific barrel within the brain. A lot of work has also been done in terms of looking at the structural and functional connectivity, as well as input and output layers and local circuitry, where we know where input goes and how that input connects to other regions of the brain, whether it be local within a barrel or long-range connections to other sensory regions. So in the previous work published in 2017 by Dr. Krishnan and colleagues, they show that extracellular matrix structures called perineural nets, or PNNs, are important neural substrates for auditory cortex, which hindered pup retrieval behavior within our surrogate hets. So perineural nets, PNNs is how I'll be referring to them because they're a mouthful, are surrounding paravalbumin positive GABAergic interneurons within the cortex. So in this image, the PNN is in green, stained with WFA, and the PV neuron is in red. Here, the WFA staining is in pink, and we have the PV in blue. So the high intensity nature of a PNN correlates to the more mature network. So the more of these proteins that make up the structure, the more mature it is. And these PNNs are thought to be more stable structures as we go through adulthood. They're also thought to inhibit synaptic plasticity by inhibiting axon growth, by being physical barriers to synaptic plasticity, as well as regulating synaptic proteins. So on the picture on the right, what you see is you have the PNN and WFA in pink, but you also have this green, which is standing for synaptotagmin, which is a vesicle protein, which is involved in neurotransmitter release. And you're seeing that they're overlapped with each other. So this further adds to the hypothesis that PNNs can retain and also solidify synaptic connections. So in order to investigate this tactile contribution of pup retrieval behavior, pup retrieval behavior, we decided to look at PNN expression within the primary somatosensory cortex. So how we did that was through whole brain immunohistochemistry and manual PNN counting. So here you have sagittal sections on a medial to lateral axis from the Franklin-Paxnos Atlas, which is what I'll be referring to from here on out. You also have a lateral section, so further out, and a more medial midline section here, showing PNN expression kind of in a whole brain surface here. So when we were going through and we were doing the immunostaining for this and imaging, we were starting to notice that the more lateral sections seemed to have more PNNs than the medial sections did. And so we decided to take a whole brain approach so we didn't miss that nuanced piece of our data. And what we did briefly, this is an IO protocol. It's available online. I can throw it in the chat later if we want to. We took our brain sections, we figured out kind of which brain section matched which map number from Franklin-Paxnos Atlas. We then overlaid that Atlas number onto our section, fit it correctly using ImageJ, and then mapped out each individual section for the whole somatosensory as well as the somatosensory functional regions. And then we did whole brain immunohistochemistry for each section. So for our conditions, we have 4 mice, as Dr. Krishnan just explained. We have our naive controls, so our wild type and naive het, and our naives have never seen the pup retrieval behavior, have not been through it, have not touched a pup. And then we have our surrogates, which are wild type and het. These are the animals that get housed with a pregnant female and go through the behavior. All of these animals are adult females, so they're between 10 and 12 weeks old. So here's an example of the output from these analyses. What I'm showing here is PNN density within one brain. And this is the left hemisphere and the right hemisphere of that brain. And this is naive Het, showing that each one of these dots is an individual brain section that we counted PNNs for. So when we were going through this, we started to realize that some had really low numbers and some had really high numbers. And we were wondering where that came from. And what we noticed was these medial sections had much lower numbers than the lateral sections did. And we can elaborate more on that from the publication that we most recently came out of our lab. So we took this, we looked at 5 different cohorts. We have the wild type Het and then our surrogates wild type and Het here. And what you can see is that in the Hets have significantly increased PNN densities as composed to the wild type, regardless of condition. So for the surrogate Het, we have the increased PNN expression as opposed to surrogate wild type. This is very similar to what was previously published in the auditory cortex using the same behavior, same aged mice. However, in the auditory cortex, we did not see this naive Het difference where the naive Het is increased over the naive wild type. So what we're seeing here is just 2 representative epifluorescent images of these PNNs shown in green. The arrows are pointing to the high intensity PNNs, those more mature structures, which is what we quantify when we're doing our analysis. And this just shows that, there's more high intensity PNNs within the naive Het over the wild type, naive wild type in these brains. The cool part about how we do the analysis is we're also able to look at different layers and see how many, that intensity within the layer. And we're also able to look at functional subregions within the primary somatosensory cortex in PNN alterations within those. So this is just a summary of the data. So in summary, total, the PNNs in the primary somatosensory cortex of the adult females seem to have this hemisphere-specific and subregion-specific expression. So when we compare the naive mice, we see that regions that are related to facial tactile input as well as Worcester tactile input are increased over naive wild type, which is what I just showed. However, when we look at surrogate Het, we see that there's an increase in the right hemisphere when compared to naive Het, an increase in the S1ULP, as well as a decrease in the S1J, specifically in the left hemisphere. Another interesting thing when we see this hemispheric bias within one brain is we see that the S1FL and S1DZ have higher expression of PNN density specifically within the right hemisphere of the brain. And what's particularly interesting about this, wrong way, is we see that same increase within surrogate wild type brains when we analyze these as well in comparison to the naive wild types. So this could suggest that there could be a maternal experience specific changes happening in the brain regardless of genotype, so more circuit specific, which is very interesting and cool. Another thing that we were able to look at is since these animals have extreme individual variability within our behavior, we were able to plot mean fold differences in PNN expression for all 20 mice. And in the wild types, what you notice is that there's a slight left hemisphere bias within the naive and the surrogate. But in the naive Het, you can see there's a lot of variability and there's a much larger left hemisphere bias here, which almost disappears within the surrogate Het, suggesting that possibly is this increased PNN density of the naive Hets maybe interfering with the mouse's ability to learn the maternal experience, resulting in this inefficient pup retrieval behavior. So what we decided to do was do a time course analysis. So like I said before, those are adult animals at 12 weeks old, between 10 and 12 weeks. So we did time course analysis looking at different subregions of the primary somatosensory cortex as well as the whole to kind of figure where is this naive Het wild type difference disappearing. And what we see looking at 12, 10, 8, and 6 is once we get to 6 weeks old, there is no significance in primary somatosensory cortex as well as these different functional subregions we were looking at. So from here we went, okay, let's perform the maternal behavior at 6-week-old animals and see if the surrogate Het can perform the behavior better. And they can't. Very simply, the surrogate Hets are inefficient at pup gathering behavior in both error and latency. These are very similar to the graphs you saw before, where this is individual animals' behavior. And then this is the average on each of the days, very similar to what Dr. Christian showed before. They're bad at it still. So we were like, all right, let's go look at the PNN expression in these brains. And this is, recently unpublished data. So I'm just showing a summary of it, so we don't have to go through all of it. is that when we did, again, whole brain analysis of PNN expression, we found that most of the changes that we're seeing, all of the changes we're seeing, are just specific to the surrogate het. So not as many changes as we saw before with the other animals, just surrogate het. What we're seeing is in the barrel field within both hemispheres is significantly higher in PNN density when compared to naive wild type in surrogate het, as well as S1DZ, which is the dysgranular zone in the left hemisphere of the surrogate het as well. So kind of all together, these results suggest that PNN expression is dynamic in spatial location with hemisphere and subregion specificity, but also that PNN density is being abnormally regulated within our surrogate het animals in a context and age dependent manner. Though it's happening at both ages, it's happening differently at both ages. We're currently in the process of understanding what that really means to the animals. So looking at the different subregions and how that pertains to maternal behavior, as well as determining the neural circuitry effect on these PV GABAergic interneurons. I'd like to thank my lab. I know I'm over time, so like 16 seconds.
No, that's perfect timing. Thanks a lot, Dana. That was exciting. And, you know, it's always nice when you can tell that the speaker is really engaged in the detective work of the science. and it sounds like you're still in it. You're still sort of chasing some tails. And I guess
very much
what real science is about. It never ends, right? Yeah, I have a question. So this asymmetry in the left and right hemispheres, that's really interesting. Has that been, you mentioned that the perineural net differences have been observed in auditory cortex as well. Has this bias been seen in olfactory, auditory, or any other cortices?
So from the analysis that was done in the 2017 paper, no. But what we're doing is kind of, there have been some reports of hemisphere bias in these animals, mostly with electrophysiological studies. But we're going in now that we've seen this, because before what we weren't doing whole brain, or Kirthi wasn't doing whole brain. So now that we're doing it, that's where I think we're starting to see these nuances in the data. So we're going back now with the brains that we already have. And now we're trying to do that. Okay, let's look at the left, right hemisphere. And we're also taking a lot more care to look at it, because it seemed, especially with that medial to lateral difference within each hemisphere too, there seems to be, it's hinting, we think, at more of a circuit level of what's going on. And in those circuits, things could be going wrong in a specific hemisphere. So, you know, studying it, making sure you're studying it in the right hemisphere or the left hemisphere, and kind of going from there. I think it gives us more of an idea of where to go next, for sure.
Okay, that's great. Let's see, that's perfect timing. We can move on. Thank you, Dana. And Deborah, if you want to take over the stage and share your screen.
Yeah, give me 2 seconds here.
Yeah, take your time. And a note to our attendees, I know that Zoom webinar is strange, but this is really a better experience for everyone, especially speakers talking to a blank room. If you participate in the chat, if you post questions, so it can feel a little bit more like the real deal, like we're in a bit of a conversation, that would be great. Okay, great. Our next speaker is Deborah Rupert, who's starting her 4th year of graduate school in an MD PhD program at Stony Brook University. She's doing her PhD work in the lab of Dr. Stephen Shea at Cold Spring Harbor, so you can start to see the relations with the 2 previous speakers that we've had. You can, yeah, again, we'll put her contact info in the chat. I'll put her Twitter handle, so if you want to get in touch with her, you can do it that way. All right, Deborah, the stage is yours.
Okay, thank you so much for that introduction, and thanks for everyone for tuning in. So today I'm discussing our work examining the contribution of methyl CpG binding protein 2, or MECP2, in parvalbumin inhibitory interneurons in the auditory cortex for processing socially relevant sounds, namely ultrasonic vocalizations. And I know that that title is a mouthful, so let me back up and give a bit of context here. As we've heard from the other presenters, mutation of the MECP2 gene is responsible for Rett syndrome. And because MECP2 is a ubiquitously expressed X-linked gene, patients are typically female and they're typically heterozygous for the mutation, which means that their cells form a sort of mosaic pattern of those that have the functional protein and those that fail to express the protein. And it's currently unknown whether some cells within that mosaic are more dependent on having the functional protein than others. So our aim is to understand how lack of MECP2 in certain cell types contributes to aberrant audition. And to do that, we study the mouse auditory cortex, which is a structure that's not only critical for localizing and parsing sound features such as frequency, but it also processes more complicated sounds such as ultrasonic vocalization and their associated features like contour and social salience. Ultrasonic vocalizations, or USVs, are very high-pitched calls above the detection range of the human ear. And I'm going to try and play this, and hopefully you can hear it on your end. But this recording is being emitted by an isolated mouse pup. And it sounds sort of like a baby call, and it has been slowed down so that you can hear it. Normally you wouldn't be able to. So as I mentioned, my project focuses on the role of auditory cortex inhibitory interneurons for processing those USVs. And this class of cells is especially important for the temporal organization of the sound processing circuitries. For example, in humans, EEG gamma waves, which are driven by inhibitory cell activity, are time locked to the onset and offset of speech cues, and those have shown to be disrupted in patients with autism. And in mouse models, inhibitory cell plasticity is really crucial, as we've heard, during sensory critical periods across modalities, including the visual and auditory systems. And again, that plasticity is disrupted in MECP2 mouse models. So the overarching goal of the project is to determine whether in a cell type restricted mutation, we can recapitulate deficits in vocalization processing that we've seen in non-conditional knockouts, and we do that using a Krelox genetic line. And a lot of the data I'm presenting today compares across these mouse lines. So I think that this visualization is very helpful. So on the left is the restricted mutation to PV cells, and on the right is sort of a non-conditional, non-cell type restricted symbol. So to examine sound processing in a socially relevant context like others, our lab uses the pup retrieval assay. And as we've seen and heard, in the paradigm, female test subjects are co-housed prepartum through postnatal day 5, and we call that the surrogacy learning experience. And as you can see here, they are tested on retrieval across multiple days. So to quantify the efficiency of those retrievals, we score with a latency index, where the higher the latency index, the more inefficient the gather is. And previously, Dr. Krishnan's work here shows that animals that are MECP2 heterozygous perform significantly worse compared to wild type littermates, and also compared to the dam across days of testing. And they also found that this is inversely related to the percentage of cells that are expressing normal MECP2 protein in the auditory cortex. So another way to say that is the more normal MECP2 protein expressed, the more normal the phenotype. So to return to our cell type specific hypothesis, when we restricted the manipulation of MECP2 to parvalbumin cells, you can see that surrogates performed significantly worse on postnatal day 0, and there's also a trend-like difference on postnatal day 1. And since we're interested in the relative contributions of certain inhibitory cell populations, for comparison, this is a restriction to the somatostatin conditional line. So these data really taken together to us seem to argue that parvalbumin cells really at these early time points are playing a critical role and need MECP2 in order to learn that maternal retrieval behavior. So now I'll turn to our single unit loose patch extracellular recordings, and these are taken from the left auditory cortex of awake head fixed mice. And the data I'm showing today are all from putatively determined inhibitory cells. This is an example of firing rate data from a single patched unit. And on the top, you can see our raster plots, and on the bottom are the corresponding peristimulus time histogram for 8 unique ultrasonic vocalizations that are being played back to these animals. And these red lines here just denote cutoffs for what we would consider to be a response to the stimuli being played, an auditory response. So in this plot on the left, we have heat maps of cell call pairs. So each one of these rows on the heat maps is one of those PSTHs that you saw in the slide before, of the mean response of a single unit to a single call. And these rows have been sorted by z-score magnitude from those that are dampening or decreasing their firing rate to those that are increasing their firing rate in response to the call. And here we're comparing a cohort of animals that are wild type naive to those that have undergone the surrogacy experience. If we take the mean score of all of these recordings taken together, this trace right here in red is the average trace for experienced wild types compared to in gray would be the average of the naive animals. And you can see that, again, these are all PV cell recordings. There's really a dampening in the response of PV activity. And to further quantify that, we can take the area or integrate under this curve, and indeed there is a significant drop in PV activity. So try to keep this in mind for what you would expect the wild type auditory cortex to be doing in response to surrogacy. These are the same plots, but for recordings from the cell type non-specific mutant. And these non-conditional mutants, you can see right away, really fail to show the dampening. And in fact, there's actually an increase in the firing rate in response to calls following surrogacy. As I mentioned, though, these are data taken from the cell type non-specific model, which means that 50% of all cells, regardless of the type of cell, are being affected here. So we can't really attribute that to one cell type or another, which really brings us to our cell type specific knockout model. And here you can see that we've recapitulated our findings in the control. These animals do show a drop in PV activity in response to surrogacy. And then in the conditional knockout, very similar to the cell type non-specific, lack of that dampening. So I know that's a lot of similar plots. So just to summarize the last 3 slides, these are the mean z-score traces from the wild type, the non-conditional, and then the conditional knockout. And I think that putting these side by side really drives home the point that restricting the mutation to the PV cells is almost sufficient for recapitulating the lack of functional plasticity here. The last bit of data I wanted to touch on uses fiber photometry to examine PV selectivity. And briefly, the idea here is to selectively record PV firing, but at a population level by using Cree-dependent GCAMP. And that allows us to monitor fluorescent signal linked to calcium activity in those cells. And of course, we expect that these recordings would have similar outcomes to our single unit recordings. But in addition, this approach lets us look over the learning time course itself in the same individual subjects, whereas in the EPHIS cohort, these are separate naive and experienced cohorts. They're not the same animals. So these data are from a wild type control subject. And here, again, we're just plotting z-score traces, but of the delta F over F instead of the firing rate changes, again, to each of our stimuli. This top row is the naive time point and the bottom, the postnatal day 5. Personally, I think it's a bit easier to see in the heat maps where, again, each line here is a specific stimulus. And my takeaway from this is that we are able to recapitulate that PV cell at a population level, that activity is dampening over this learning time point in the wild types. To get a better look at that, we can also take a mean z-score across all the different stimuli instead of dividing it out among the 8. And you can see here that there's a drop from this first exposure to the end of the learning time point at postnatal day 5. And then in comparison, here are the corresponding plots from a PV mutant animal. And you can see our early learning time points here show a strengthening of the PV activity. So this is a direction we're actively following up on right now. So just to summarize, the phenotypic data suggests to us that PV cells are particularly important and compared to other cell types, and that the single unit activity of PV cells in the cell type specific mutants recapitulates the cell type non-specific. And we're hoping to further drive that home with some population level calcium signaling, which parallels those single unit findings. I want to thank you all for listening, and especially my mentor, Stephen Shea, and my sources of funding. So happy to take any questions.
Thank you, Deborah. First of all, I want to say kudos on well, getting a couple of very different measurement modalities under your belt and your shortened PhD, and also extracellular recordings in a moving awake animal. I've been there. That ages you, so congratulations. I have some questions, but so does the audience, so let's jump right to those. Do you have, Billy Lau is asking if you have cortical layer information for the in vivo recorded cells.
That's a really good question. Yeah, so the paper that Dr. Lau put out in 2020 suggests that some of those changes are depth specific for where you are in the auditory cortex. And that is an analysis that I am actively working on.
Excellent.
So stay tuned.
Okay, so you'll give him the answer later offline. And Dr. Christian is asking, when were the PV conditional knockout recordings? P0 to like early on, or P5 or later?
Right. So the EPHIS recordings, I think if I'm understanding correctly, for the surrogacy experience, they're all within a week after the surrogacy experience. But yeah, definitely like when those recordings are happening could certainly affect what you're seeing. It's I think that's also definitely one of the advantages of using the fiber photometry, is like really getting a tighter grasp on the timeline of changes going on. Although it's not single unit recording, so pluses and minuses.
Okay, great. And I have probably what's a naive question, because I'm not quite in this field, but it seems that you've sort of identified a potential therapeutic target here. Like, would optogenetic stimulation of the PV population rescue some of this experience-dependent plasticity? Can you sort of speculate on the applications here?
Wow, that's a really long-term question. And I would actually argue that the other speakers probably identified a more easy target than I did, just because.
Great, let's unmute everyone's mic and do a few sentences of a last round. That's perfect.
Just suggesting that perineuronal nets, with the molecular end of things in terms of pharmacology. But your idea is also really cool too.
Yeah, I think one of the big problems with all of this, it's really cool to know the underlying mechanisms, but PNNs and PV do so many different things. The idea, unless you have like really precise timing that you're affecting with this process, I think it's going to have huge consequences. That's the best way to say it. So I guess with optogenetic methods, if you are going to try to do that, then it would have to be timed exactly right. And I think we don't know that precision at this point, I would say.
Yeah.
I would also say that, a lot of this stuff in terms of translating from the basic to the clinical science is like, it's all well and good to say, oh, this is really a cell type specific issue. But in patients, we don't even have that capacity to like target whether, optogenetically or pharmacologically. It'd be pretty hard to specify the target there.
Okay, great. Okay, we're right on time. So thank you, all three of you, for a really exciting session. As I said, I'm a little bit outside this field because I'm a volunteer for Neuromatch, but I learned a lot. And I'm happy to have both seen for the first time a puck retrieval movie and also to hear the slowed down vocalizations. So thank you all. Thanks for keeping time. And we hope to see you for other talks the rest of the week. And thanks to everyone in the audience and to Taha. Bye, guys.
Thank
Great
you.
job hosting. Great job hosting.
Honored.
Cool. I guess then without further ado, I'll begin. So today I'll be presenting some of my PhD work. So as you're all probably aware, the brain is a highly complex network. Its arrangement of neuronal elements facilitates cognition and behavior. So understanding this arrangement of connections is important. Here, I'm investigating whole brain macro scale connectivity. This type of connectivity refers to the white matter pathways that connect different regions of the brain. We can understand this connectivity by representing it as a network of nodes and edges. So with the humans, we can do this with MRI, so we can use diffusion-weighted imaging and run a process known as tractography to reconstruct potential white matter pathways using streamlines. We can also parcellate the brain, in other words, divide it up into different regions. Then to make a network representation, we can have the brain regions become our nodes and the streamlines become the edges and connections between those nodes. And when you've done this, you have a network representation of the brain, and you can begin to ask questions about its organization, its topology, how its connections are arranged. So there are many such topologic properties that you might investigate. The one of interest for us today is this idea of node degree. So node degree is the number of connections a node has. So nodes with lots of connections we call hubs, and they tend to be known to be very important for brain communication. We know that in many disorders and diseases that hubs seem to be preferentially targeted. So it's very important to understand hubs' role in the network. So in the field of network neuroscience, we've gotten very good at describing all these different kinds of topological properties and what kind of function they may have. But the question now we're beginning to ask is, how do these properties emerge? What factors constrain the development of the brain's connectivity? And one of the key ideas is this notion of wiring costs. So forming and maintaining a connection in the brain requires some expense of metabolism matter. And so in turn, connections are constrained by these properties. And this gives rise to the notion that connections have a wiring cost. Indeed, we can see in a variety of networks evidence for this wiring cost by this distance decay. So what we see is the probability of any two connections, any two regions being connected, the probability of this varies as a function of the distance, and it follows this characteristic exponential decay. And we see this across numerous different species, such as the mouse, the macaque, and the human. And so one question is are these wiring costs key in shaping connectivity? And we can investigate this with generative network modeling. So a generative network is quite simple. It basically tries to form connections according to some rule. In this case, we're saying the probability of connection forming between nodes I and J depends on some exponential function of the distance between them. And this function is controlled by this eta parameter. And so when this eta parameter is set to this value, what we'll get is, if we generate a network, is we'll get mostly short-range connections, but some long-range connections as well. And as we increase the value of eta, so increasing the strength of the scale, we get more and more short-range connections. And as we relax it, we get less and we get more and more. We get more and more long-range connections. And so basically, we want to see if this model can reproduce features of the brain. And so to do this, we basically do model comparison, where basically we take our model, we take our data, and we compute various topological properties, such as the degree distribution or the edge length distribution. And then we compare these fits using the Kolmogorov-Smirnov statistic, which involves comparing their exponential distribution functions. And basically what we're looking for is the maximal difference between these curves. And you look at this maximal difference across all the properties you're looking at, and that becomes your indication of model fit, which we term max KS, but some people also term energy. And then you do some kind of optimization scheme, searching through all these different values of eta, trying to find a combination that gives you a network that most closely resembles your data. So these wiring cost models can produce properties such as a degree distribution. So shown here in red is what the degree distribution of what the model reproduced, and the black dots are the data. So it's a reasonably close correspondence. However, the brain seemingly did not evolve just to conserve wiring costs, but also to favor forming functionally valuable connections, even if those connections have some kind of high cost. And this isn't captured by the kind of model I just showed. Instead, we require so-called trade-off models. In these trade-off models, the probability of forming connections is not dictated just by distance, but also some kind of topological term, which represents some kind of functional value. And examples of potential topological values are shown in the table on the left. And what we see is that these wiring cost models, represented here by this geometric model, have the worst fits, and instead you require trade-off models in order to produce a network that closely resembles the brain. And so this is all very well and good, and this is currently where the field is, but there's a few issues which sort of need resolving. The first regards model formulation. So as I've just shown, this model is formulated, this wiring law is formulated as this multiplication of two terms, a distance term and a topology term. And these terms are nonlinear. And so when you multiply these terms together, you get nonlinear effects. So for instance, as we're increasing our value of topology, what we see as distance increases, that this topology has less and less of effect. And this kind of is at odds with this idea of a trade-off, where the idea that a functionally valuable connection can overcome its wiring cost in order to form. Under this multiplicative formulation, it's very difficult for this to ever to happen. So we reformulated this as an additive form. So now we're adding on a topology value, and this makes our model more linear. And so now that when we're adding more and more topology in, that long-range connections can stand to benefit from these values. Another issue concerns developmental processes. So this, as I've said, this notion of wiring cost is very key. And usually we approximate this notion of a wiring cost by just measuring the distances between two nodes. The problem is we measure these distances in the adult brain, and the adult brain is a lot larger than the fetal brain when connections are actually forming. And so it stands to reason that during development, when distances in the brain are much, much smaller, there is a reduced wiring cost. And so to investigate if this had any effect, we developed a model of growth throughout development, where basically we took MRI scans from fetuses from 21 weeks gestational age on a week-by-week basis up to 38 weeks gestational age. And basically what we wanted to be able to do is measure changes in the distance between nodes over time. So to do this, first we need to identify correspondence between these time points. So we use something known as multimodal surface matching, which basically allows us to identify where a position on one brain surface is on another. And with this information, we can then take our adult parcellation data and then project it throughout all our time points. And then we can measure basically how distances are changing between these nodes at every time point. The final thing that we wanted to look at was this idea of spatial properties. So currently I've talked about topological properties as just being distributions, but these distributions are spatially embedded. So if you look at something like the degree distribution, shown here, we can see that it has very distinct spatial positioning. And we know that this positioning is very important for the brain's overall functioning. And the spatial embedding of properties is a key property of brain networks. And so it's unclear whether generative models can capture these kind of distributions. Some evidence says potentially they can, other evidence says they can't. So we're throwing our hat in the ring and seeing if this is possible. So basically, what we ended up building is we built a growth model and a static model. In our growth model, we basically have our distances at one time point, we form connections, move to the next time point, distances, form connections, and so on and so forth. And in our static model, we're just using the adult distances, and the static models is what most people use. So we ran the model for 100 participants and optimized this to identify the best parameters. We used a leave-one-out cross-validation procedure to evaluate model performance. And then finally, we assessed the spatial embedding by simply correlating the degree of the empirical networks with our networks produced by the model. So for our results, there were three main things we were looking at. The first thing was looking at the multiplicative formulation versus the additive formulation. So in our additive formulation, what we see is that it can produce networks that have better fits than our multiplicative formulation, shown here with the black violin plots. But again, this is only for select models, but we only really worry about the fact that it's only our best performing model, which it's this matching model which is performing the best, and under the additive formulation is getting the most advantage. Next, we're looking at static versus growth. And what we see is that even though there might be some significant differences, in actual fact, these differences are very, very small. So growth isn't really having that much of an effect. In some cases, it's even detrimental. And the reason for this is if we look at our eta parameter, what we see is that there's an increased decay for the growth models. And this makes sense because on average, the brains are much smaller. And so basically the model is trying to compensate this for the smaller size by just increasing the distance penalty. And the final thing we looked at was the spatial distribution of nodal degree. So what we see is that if we look at our best performing, best fitting models, what we find is that on average, these correlations are around 0. So they're not reproducing the spatial patterning of degree much at all. And if we look a little more closely, we can sort of see why. In our empirical networks, the hubs are more sort of distributed across the brain, but in the models, they tend to be very tightly clustered in geometrically central regions. There's no way for the model to be able to escape this distance constraint. So I guess there's some quick subtleties to my results. So firstly, an additive formulation can improve fits, but only for select models. The generative models, including growth, don't really appear to be able to better replicate statistical topological features of the brain than non-growth models, and generative models seem to fail in reproducing the exact, say, spatial positioning of hubs. And so on that note, I'll quickly end. I'd just like to thank my supervisors, Alex, Ben, and Kevin, and also my colleagues, Arena and Rosa, for all their technical assistance. So thank you very much.
Thank you very much, Stuart. Was really great presentation. If anyone has questions, feel free to use the Q&A button again. And while we wait for people to type in, I actually have one question, which is, so you mentioned that you evaluated your model, the fit on the model using the degree. Did you use any other metrics, like for example, strength in nodes?
So
and what is the reason why you chose degree, let's say also?
So basically, the way we optimized it was based on 4 parameters, which are showing here. So we optimized it on the degree distribution, the clustering distribution, betweens distribution, and edge length distribution. And so basically, we were trying to look for, based on these properties, whatever one was causing the worst fit, and that was our indication of model fit. The reason we chose these properties is historically that's being done. So in order to allow comparison with previous work, we chose these. These are all binary measures because currently it's unclear how to easily extend these models to weighted networks. I mean, it's very tricky because you can get what we might call runaway effects. So basically, when one connection forms and you're trying to infer weights, you might try to add more connections, but what the models sort of end up doing is that one connection that initially forms has such an advantage over everything else that it just attracts all the weight. So it's an ongoing problem. We know that the brain is a weighted network and those weights are very important. And currently we're only assessing it in a binary form. So that's something we need to extend these models to.
Yeah, okay, thank you. And we have another question from Larissa. What interface did you use to create the connectomes?
What interface?
Yeah.
So I was using diffusion weighted imaging. So I would use tractography on that. And then I'd combine that with a parcellation, if that's what you mean.
Okay. I also have another follow-up question from my previous one, which is that I guess is not only like the fact that the brain is a weighted network, but the next step is also to be able to extend these models, I guess, also for directed networks from undirected to directed ones, especially for taking into account inhibition and excitatory like activities in the brain. So what are your thoughts about that?
Well, in human, it's hard to infer directionality. So obviously it'd be something I'd love to do, but we just don't have the data on that. I believe people have done it with C. elegans, where you can potentially infer directionality based on these networks. I mean, these networks, it's very easily to extend the model to directed networks. But I mean, potentially with a macaque, I just don't know if everyone's really tried. I think it's a very sort of new field, this idea of generative network modeling for network neuroscience, at least. So I guess we're trying to just work through a lot of the kinks and underlying sort of mathematics first.
Yeah, so the follow-up question from Larissa was like, what program like MR-Trix
what program?
did you use?
So the diffusion imaging was done with MR-Trix, and the surface processing and everything was done through FreeSurfer, and then everything else was basically done in MATLAB.
All right. Yeah. Another question, like I would say this is the last one. So from John, would you consider the overlap of my Cartesian coordinate system for the human forebrain in your system? And as the link on forebrain.org. I don't know if you are familiar with that.
No, sorry. Could you just repeat the question? Because it dropped out for like a second.
Yeah, sorry. So would you consider the overlap of his Cartesian coordinate system for the human forebrain in your system. I think he has put in the chat box also a link to the forebrain.org.
I'll have
But
to
yeah,
I have to look at it a bit more closely to sort of figure it out.
Yeah. Okay. Yeah. We'll take it that offline, I guess. Well, thank you very much for presentation. Really great work. We're looking forward to seeing it available to everyone.
Looking forward to publishing it.
Thank you, Stuart. Now we invite Peter. So unfortunately, we had a dropout. So we are gonna proceed with Peter Nicholas. Hello. Okay.
Hello there.
Yes, I can hear you now. So Peter is gonna present O8T Labs, an automated machine learning based approach to personalized brain mapping and functional connectomics.
It is a bit of a mouthful, isn't it?
Yeah.
Would you be able to confirm that my screen is coming through okay?
Yes, it's coming through okay.
Awesome. I can launch into it now if you'd like.
What, sorry?
I can get started now if you'd like.
Yeah. Go ahead.
Okay. Good morning, everyone, and thank you for your time. Thanks for the introduction. My name is Peter Nicholas, and I'm a senior data scientist at Omniscient Neurotechnology, or O8T. I think it'll be a bit easier for people to remember and say that. Today, I'm going to be presenting on our workflow, actually, and that is a automated machine learning-based approach to personalized brain mapping and functional connectomics. And broadly, what this aims to do is generate functional connectomics in a kind of easy, seamless way and enable researchers to spend more time researching. So to begin with, a brief introduction to functional connectomics. I'm very glad that I get to follow up Stuart because there's a lot of overlap with the structural connectomics that he just mentioned. We begin with a brain parcellation. And then for each of those parcellations, we're kind of extracting out the resting state fMRI signal. We're looking at the correlation between each of those signals, and we're kind of constructing an adjacency matrix or a functional connectome that I've heard quite a useful analogy. It's how the cities and highways of the brain kind of fire together. So basically what we're producing here is a map of what areas fire together, and we're using bold signal change to quantify and generate that map. Why is this valuable? Well, functional connectomics have enormous potential. They offer kind of unparalleled insight into brain disease because it enables you to kind of trace brain activity to functional patterns. Once you start putting a label on these functional connectomics, Alzheimer's, depression, what have you, you can start doing some really interesting analysis. And it starts giving you an opportunity to leverage big data. It's much easier to work with 1,000 functional connectomes than it is 1,000 resting state MRI scans. Unfortunately, this does come with some barriers to entry. As we might all know, it's quite a time-demanding workflow, not easily scalable, unfortunately. And there's a lot of expertise required in the pre-processing. That is to say, just general data cleaning and processing. It's not the kind of sexy stuff, it's not the elegant stuff, but it is incredibly important. And with this comes a lack of standardization. The variance, the necessary variance in all these pre-processing pipelines does raise some concerns for replicability and kind of lack of standardization across studies. So how have we approached this kind of problem? Well, the first thing I'd like to mention is the personalized atlasing approach. So this starts with a diffusion MRI scan and involves the generation of CST tractography, as you can see in the middle diagram on the bottom row. We use this CST tractography to update the standard atlas and generate a machine learning adjusted atlas, which is what we're seeing in the middle diagram on the top row. Then the combination of that parcellation scheme with resting state fMRI enables us to generate structural and functional adjacency matrices, or connectomes rather, that are more accurate than a traditional kind of 3D registration of atlas. We do a lot of work with people with tumors and cavities who have had tumors resected. And so this methodology allows us to kind of work with deformities like that. The other part of making this workflow automated and effortless is in achieving kind of computational scale. So a lot of the pre-processing steps are quite complicated and time-consuming. And what we do to obfuscate and abstract that away is what I've just highlighted in red to the right. Send that all off to the cloud for processing. So we kind of run all this processing in parallel in the cloud, and that enables us to kind of launch many, many jobs in parallel and not have to iterate through all of them sequentially, as I've seen people do on their local laptop, and that is just the biggest headache. We actually happen to do a lot of work with clinical researchers. So as a necessity, we've kind of built in, an interface to typical hospital infrastructure and the ability to work with DICOMs. A lot of the concern they have, rightly so, is in patient security, patient data sensitivity, and matters like that. So because of that, things like de-identifying the data, stripping DICOM headers, etc., face removal, all of these are very much desired by those kind of researchers who want to take data concerns very seriously. And then I'll just mention it, but not so relevant for the researchers, but for actual clinicians that want to use this data, once we've de-anonymized it and they want to use it in the clinical practice, we can actually reattach the face, reattach those data points that we've stripped away for privacy. So at this point, I'm just going to kind of give a quick demonstration of the workflow itself, not a demonstration of research and results. This is very much kind of work in progress, but I thought I'd just kind of ground this in reality and put some kind of context around this workflow, which I'm espousing. So recently we worked with a group in China who wanted to use our workflow to generate functional connectomics on a set of schizophrenia subjects that they had. They came to us with a very, I'd say, ambitious turnaround time, requesting that we generate the data for 125 schizophrenic subjects before and after treatment, as well as 100 matched controls. Concretely, what this meant was that there were 250 fMRI cases that we needed to process, as well as all the CST tractography, motion realignment, all of that happens in the background. And by launching these in parallel in a swarm, up in the cloud in our automated workflow, it was a less than 24 hours runtime until we could get the results back to that team and they could start doing the research that they wanted to spend their time on, kind of saving them the time to fiddle around with different pre-processing pipelines and trying to link together the computing power to get the job done. So I've been talking about this workflow so far, but I'd just like to touch on where it might be going in the future. So unfortunately, some of this slide is already outdated. I have on the left here structural and functional fingerprint of Alzheimer's as an area of research. We've already been working with some teams to get started on that. And what that looks like is very similar to what I previously outlined, but kind of enabling our workflow on someone else's data to get them the data that they require to start looking at what kind of functional connectomes and structural connectomes are unique to Alzheimer's. There's also a large undertaking in quantifying the impact of brain surgery. As I mentioned before, we do a lot of work with clinicians that have patients with tumors and want to see what the impact of resecting those tumors are. And it would be incredibly insightful to put a number on the impact of a brain surgery and empower the surgeon and the patient that way. And an ongoing research area that we're looking to get into is into responsiveness to treatment in depression. Excuse me, depression. But in the long term, this is going to necessitate some upgrades to the workflow itself. So a big area of focus is in multi-source data homogeneity. And what I mean by that is that if you want to leverage big data, you need a lot of it, and it's going to, out of necessity, be collected across multiple sites, different scanners, different protocols. You cannot just use that willy-nilly. You need to take some sort of standardization practices into account to make that comparison fair and valid across multiple sites. We're going to be extending our analytical use cases. So right now I've shown you really only functional connectomics and a little bit of structural connectomics, but there's so many other things that would be good to automate into our online workflow. There's some machine learning smarts behind all of this, and it is, as you'd want to see, it's good to iterate that and continually improve and improve that over time. And then in the medium to long term, our kind of goal is to shorten the bridge from academia to clinical practice and bring some of the great ideas into the hands of clinicians as soon as possible. So before I run out of time, and so that I don't get yelled at by the people upstairs, I will quickly touch on exactly what I've been talking about this whole time, the offering in question, and that is O8T Labs. It's a fairly available and unlimited tier offering for generating these structural and functional connectomics. It lets you investigate at the subject level. I mean, the user on the right is navigating the viewer, and that's completely in Google Chrome. So, no software needed to install, can work on anyone's laptop. And the use case here is to help people organize their huge data sets, identify particular patients of interest, and export kind of academic research insights as they desire, things like connectomes, but very much use it how you desire. So with that, I'll open to questions. Thank you for letting me present, and yeah, it's been an honor to present here.
Thank you very much, Peter. It was a wonderful presentation. We have one question already in the Q&A chat, and I'll read that for you. So John asks, I noticed your parcellation of cortex derives from one economo and not Brodmann. What was the reason?
Yeah, that's a really good pickup, given how quickly I blasted through all those slides. So we actually went with the HCP MMP Atlas that came out of the Glasser research study. We felt like at the time that was, I guess, kind of the cutting edge, and so we just went with it. There is definite need to kind of evaluate the whole world of parcellation schemas, and I think a lot of them actually, I don't even think that there's a one size fits all or there's a best. I think different schemas offer different advantages and disadvantages. So in response to your question, we happened to go with HCP MMP because it was detailed, granular, which is exactly what we wanted at the time. But I'm sure there's a lot of value in the other schemas that you've outlined and the ones that are out there. I notice now that he's linked to forbrain.org.
Yeah.
That's something I'll check out.
Yeah, thank you. And I also have a follow-up question, which is, so you mentioned you want to deploy this, especially for clinicians or for researchers in the area. And I was wondering, like, if you also had a plan for making like task fMRI and the task connectomics like supported by your platform, especially considering like the variability that there is there whenever you design a task and you design an fMRI study, and how you plan to cope with that.
That's a really fascinating question. We're in an interesting space where we work with researchers and clinicians, and we've actually found that clinicians much prefer resting state because it's a bit more hands-off. But there's been enormous interest in having something, similar for task-based, where you can see the impact of a stimuli. A lot of clinicians have expressed an interest in kind of being able to do a task-based fMRI, but without the task. So that is to say, seeing where vision would fire if you were to do a vision task, or where motor system would fire if you were to do a motor system task, but getting as much of that as possible out of a resting state scan. And that's, that's not a small question. Would be a dream, honestly. But so there's been a lot of demand there, and something we need to look into.
No, sorry for the difficult question. I know it was like much broader than the time that we have available.
No, it's a good question.
But yeah, I was curious about like your take on that. We also have a kind of sort of a question, if you can share your contact information in the chat. It was like, I don't know if you can, I think you can see the chat. Larissa was asking if you have like a interest in connecting with her for discussing the OAT program further.
Absolutely. Thank you for your interest. I'll share the details in the chat and reach out.
So I'm just waiting for another maybe 1 minute or so if anyone has any other question for Peter. In the meantime, I wanted to ask you, like, what are your plans from the machine learning perspective? Like, if you're planning to implement some algorithms in your framework, or just leverage on other platforms for machine learning?
That's a great question. I think there are so much great work out there that we should definitely leverage as much as possible. Specifically, a lot of the programs we have on the back burner are machine learning analysis of functional and structural connectomics, and people have had enormous success in that. It's astounding what they've been able to achieve. As I touched on, a huge concern for us is multi-site, multi-scanner kind of site-specific issues and those kind of confounds. So I think that's the big challenge to get over first. We'd want to have some assurance that whatever machine learning we're applying to our connectomics or whatever it may be will work across the board as much as possible. But yeah, there's a lot of great research out there.
Thank you very much. So with that, I think we have no further questions. Let's thank Peter again for his presentation, and also Stuart, our previous speaker, and all of the people that attended these talks. Thank you very much for being here, and I'm hoping you're going to enjoy the rest of NeuroMatch. Thank you. Bye, all.
Thank you.
I think we will get started because we are short on time. We want to make sure that this moves quickly. So hi, everybody. Welcome to this session. Just by way of background, all the speakers have 12 minutes to introduce your topic, and then we'll discuss for 3 minutes. So I will give you guys, if you guys leave my video up in your feed, I'll give you a warning at 5 minutes, 2 minutes, and 0 minutes. For those of you watching, please post your questions in the question and answer panel. Ask anything you like. There are no dumb questions. This is a fairly new topic for me as well, so I'll maybe start out the discussion and just vote for your favorite questions in the Q&A panel. Those I will make sure to ask those first, and you can chat to panelists or everyone You feel free to share any links to papers, links to Twitter handles, links to relevant work. You're welcome to do all of that in the chat. So I'll stop the share here, and Adriano, why don't you start? So we're going to start with Adriano Bellotti, who's a PhD student at the University of Cambridge in Timothy O'Leary's lab, and he's going to be sharing his exciting work today on synaptic scaling of excitatory postsynaptic firings. Actually, that's not what you is of hippocampal neurons, right?
Yeah,
no,
yeah.
Synaptic scaling, requiring a balance between stability, efficiency, and accuracy in realistic neuron morphologies.
Yep, that's good. Perfect. Thank you. Well, hello, everyone. My name is Adriano. I'm a graduate student at University of Cambridge. Today, I'll be talking to you about this project. Again, the title is Synaptic Scaling Requires a Balance Between Stability, Efficiency, and Accuracy in Realistic Neuron Morphologies. I'll be showing you some of the theory, the model, and the simulations from this project. So let's get started. Can everybody see my screen? Can you all see? Give me a thumbs up. Perfect. Okay, so I have a little pointer here as well. Okay, so we're going to begin with some background in neurobiology, specifically the mechanism that forms the foundation of learning and memory. So we're going to start by defining what a synapse is. A synapse is a junction between two neurons. It allows a presynaptic neuron, this bulb-like structure here, to pass an electrical signal to the postsynaptic neuron, which is this long blue projection. So synaptic potentiation is when a synapse becomes stronger. So the strength of this connection gets stronger. There's a theory in neuroscience called Hebb's postulate that says neurons that fire together wire together. And this holds true here. Increased activity in the pre or postsynaptic terminals strengthens the activity, strengthens the synapse. And I'm representing that here with the thickness of these borders. So this happens at the level of the individual synapses, but in the context of cells and neural circuits, this is what enables us to learn and to retain memories. This kind of feedback is dangerous, though. It can firstly result in saturation of input signals, where the overall strength of the cell is saturated. It can also result in a sort of vicious cycle. Correlated activity between neurons leads to synaptic potentiation, and synaptic potentiation then leads to more activity. So what keeps this in check? What kind of prevents this from strengthening forever? What balances the books and prevents oversaturation? Let's take a look at this in the context of a larger cell. So in this example, we have a larger dendritic tree and a single branch is being potentiated. Just below this neuron, we're going to have a trace of neuron activity, which is essentially the number of times that the neuron fires an action potential. And that's experimentally correlated with calcium concentration, which is this curve at the bottom. Now, when a branch is potentiated, we're potentiating this branch here, we have more firing in the middle plot, and the calcium concentration on the bottom increases as expected. How does the cell prevent this from continuously increasing over time when more branches are potentiated? If left unchecked, this could result in excitotoxicity at the cellular level, and at the level of the brain, this might manifest as seizures. So the neuron's solution is to globally decrease its total excitability, to decrease the strength of all its synapses, and this is called synaptic scaling. Synaptic scaling has been proposed to do a lot of things, and I'm listing them up here. It's thought that synaptic scaling prevents the vicious cycle that leads to unconstrained excitation. It also maintains the relative strength of the synapses. And after synaptic scaling, this last step, notice that the potentiated synapse is still stronger than the rest. That change was preserved. And this is why it's integral to learning and memory. Stable, reliable changes at the molecular or cellular level, like this, result in stable, reliable memories. So this is what synaptic scaling does and why it's important for our cognitive functioning. But this begs the question, how does it work? We'll show that next. So here's a figure from a prominent review that provides an answer. This figure shows that a neuron expresses some AMPA receptors, shown at the bottom here, these little red markers there. And these receptors shape the electrical inputs to the cell, shown here, which changes the rate of neuron firing, which again changes the calcium concentrations, and that kind of feeds back into the nucleus where they regulate scaling factors that then go out and regulate AMPA expression. So there's kind of a feedback loop here. Now, the takeaway here is that synaptic scaling requires global regulation of AMPA receptors. This is kind of nice. It's a difficult problem to regulate throughout an entire dendritic tree, much more complex than any diagram I've shown so far, and the cell needs to normalize all synapses by the same factor. The way to do that is with global control. But this brings up a bunch of new questions. How does the global signal work? How does it get transported everywhere? Why is it so slow? Does it matter that it's slow? We've observed 1 or 2 day settling times for this. What influence does cell morphology have on this? And are there inherent fundamental constraints because of this? To answer these questions, we've built a model, and our model contains realistic AMPA production. Our model also has AMPA transport, insertion into synapses, as well as global and local feedback based on calcium concentration, all of which are consistent with experiments and the leading hypotheses for synaptic scaling. So our model has produced some nice findings. The global control required for synaptic scaling turns out to be a limitation for a few reasons. Neurons are big and transport can take quite a while, especially in dendrites that are up to 1 millimeter in length. This results in a phase lag or a lack of synchronization between what's happening in the center of the cell here and what's happening with the AMPA receptors at the ends of the cell. And this asynchronization can be a source of instability. So this explains why synaptic scaling is slow. You need to transport a scaling factor long distances over the dendrite. And it can take a day or two because of this phase lag. This limitation also imposes a trade-off. Synaptic scaling of a neuron happens to be a balance between efficiency, accuracy, and stability. And this is kind of the meat of the results of our project. But before we get into the results, let's dive a little deeper into the model, get an idea of how it works. So this is our model. I'm going to move this neuron up and break down all the individual parts. We're interested in neurons with a soma and a large branching dendritic arbor, because remember, the larger the dendrites, the more phase lag there's going to be, the harder it is to globally regulate. If we zoom into the soma, we can see the site of cargo production and global feedback. Our cargo of interest is the AMPA receptors, which are these dark blue dots, and we're using AMPA receptors themselves as our scaling factor. They're produced in the nucleus and transported on these microtubules, these light red tracks. They're then released for spread throughout the cell. Now, global regulation occurs based on the total number of AMPA receptors on the surface of the dendrites. That's this computation here. And if neuron activity is too low, more AMPA receptors are produced. If neuron activity is too high, AMPA receptor production slows down. If we zoom into the dendritic branch here, we can see that inactive AMPA receptors are trafficked inside the cell, and they're labeled as M. Active AMPA receptors are labeled as G, and synaptic sites are labeled with C. Now, the important thing about C here is that there must be an available synaptic site on the membrane for activation of an AMPA receptor. So in a sense, C is the capacity of the synapse. Now, lastly, if we zoom into the membrane itself, we can see what cargo insertion looks like. Cargo is inserted with some rate SI, and it's removed with some inverse rate S minus I. Now, it's worth noting that this model isn't just conceptual. We want to make it data-driven. So these quantities are broadly quantified from experiments like the velocities, the insertion speeds, and the degradation rates. And using these numbers, we made a coarse-grained assumption with as few free parameters as possible. So this is kind of a visual abstract for a model. And these are the kind of, this is the basis for how our model works. Now, in this first simulation, it's kind of a proof of concept to show how our model works and to show that it actually replicates synaptic scaling. So we have a pretty big pyramidal cell, approximately 1,000 microns or 1 millimeter in length. And we're going to simulate production, distribution, and regulation of AMPA receptors. Now, 5 hours into this time course, we simulate synaptic potentiation, where the red compartments are going to be potentiated. And let's take a look at that here. So this potentiation of distal compartments in neurobiology might be similar to the perforant pathway. Now, when we run this simulation, we could see that when we potentiate the synapses, the distal synapses actually increase in density, so that works. And what's nice here is that the unpotentiated synapses in black, they actually decrease in synaptic strength. So that's synaptic scaling in action. It actually happens to work here, which is good. Now, to navigate this trade-off, we've said that synaptic scaling is a balance between these three parameters: efficiency, accuracy, and stability. Navigating these requires tuning some parts of the model. And the neuron ends up tuning its global regulation, local regulation, and pool size. So I'm going to kind of refer back to the model to show you how the neuron might balance between these. So let's start with defining what these attributes actually look like. Stability is kind of defined by the overshoot and the oscillations that you see here. accuracy is the synaptic ratios, which is kind of how well it maintains the synaptic ratios after potentiation. And efficiency is the speed or the time constant, how quickly it adjusts, how quickly it reaches that steady state. Now, for that first end of the triangle, we're going to show that the neuron can regulate its sensitivity of global feedback. And here's kind of the gist of it. With weak global regulation, the neuron has a slow and stable response. With strong global regulation, shown on this side, the neuron has a fast and unstable response. So this first parameter, global control on the side here, kind of regulates between efficiency of synaptic scaling and stability of synaptic scaling. Now, this was global control, which of course was all compartments feeding back into the soma. The cell can also regulate itself locally, and that's what we're going to show next. If you regulate local feedback, you can then kind of tune between, and this is the local feedback mechanisms that we're kind of lumping together. If you regulate local feedback, you can kind of tune between this unstable accurate regime with weak local regulation and a stable inaccurate regime with strong local regulation. And that's what these three simulations show as well. So local feedback can kind of tune between this side of the triangle, between stable and accurate synaptic scaling. Lastly, we regulate this internal pool size on the top here that kind of regulates between efficiency and accuracy. So this internal pool size is essentially the amount of inactive AMPA receptors that are present locally, that are present internally. And that's modulated by this S-I parameter. Let me show you the simulations for those. If you have a small internal pool, your synaptic scaling is fast and inaccurate. If you have a larger internal pool, you get pretty accurate scaling, but it ends up being unbearably slow. So by kind of tuning these three parameters, the cell can decide how it wants to optimize its synaptic scaling. Now, I've kind of showed you all of this and told you that it works and presented this example. The next question is, how do we know that this works? What is like, when we break this down, when we look at any cell, how can we guarantee that this works? And unfortunately, there's no time to tell you about that here. So I'll have to refer you to a coworker's talk that is happening on October 30th, but that would kind of be the explanation behind a lot of what I presented here. So as always, there's a lot of people to thank, a lot of people working on this project together, and I'd be happy to take any questions. I think my time is probably up.
Yes, I was trying to flash some cards, but, but concentrating on your talk. Fascinating. Are there any questions from the audience here?
There's one in the chat.
I'm trying to see the chat.
So it's a question and answer under.
Yeah, for some reason, oh, here we go. So Conrad says, Hebbian learning with scaling converges to the singular vector associated with the largest singular value. How close is the model to the simple case in practice?
Sorry, can you repeat that question?
Yeah, so Hebbian learning with scaling converges to the singular vector associated with the largest singular value. How close is the model to the simple case in practice?
Well, it depends on how accurate. So in a linear model, we could achieve that perfectly. Then, of course, when we try to make the model more realistic, such as including degradation of AMPA receptors or larger regions to transport across or realistic transport rates, it becomes less accurate. So that's firstly a function of how accurate our model is and how realistic our model is. Secondly, kind of going back to this triangle here, the accuracy does come at a cost of efficiency. So if you allow the model, or if you allow the neuron 5-6 days to scale, then it can scale that perfectly. And if you allow the model to have some oscillations or some overshoots when it scales, you can also have that. You can also have that scale perfectly. I guess the trade-off realistically with a real neuron is that oscillations or overshoots aren't realistic. They're not really observed in nature. They might be pathological. So the accuracy really is kind of, it comes at the cost of these other things. So you can achieve that perfect accuracy, but it doesn't come for free. I hope that kind of answers the question.
Great, thank you very much. We need to move on to the next speaker, who is Özgür Genç. He is a postdoc at UCSF, and he's going to be sharing with us some of his exciting new work on the genetic link between impaired homeostatic plasticity and neurodevelopmental disorder. Thank you.
Thank you, Karina, for chairing the session. Can you see my screen?
Yes, yep.
Okay, perfect. So today I'm going to talk about the genetic links between impaired homeostatic plasticity and the neurodevelopmental disorders. So I just want to give you a quick overview of how I define the homeostatic plasticity. So this is Adriana just gave a quick overview of that. So I'm just going to show you how we can exemplify homeostasis. So a neuron has a certain set point of activity. It can be exemplified as firing rates. And when the system sees a kind of a perturbation that can be environmental or genetics, this firing rate can go down transiently. But in the continuous presence of this perturbation, the systems can restore back to their set points by basically reaching this firing rate. So this is basically how neuronal activity can be controlled homeostatically. So it is quite interesting that these homeostatic signaling systems are very important for brain function, but it's also highly relevant for the neurological disorders. So if this signal is impaired, then the nervous system can be less robust to perturbations that can include the genetic or immunological or other environmental stressors. So I would like to emphasize this phenomena in autism spectrum disorders. So autism spectrum disorders are caused by idiopathic causes, or there are de novo mutations recently in the last decade have been identified that put individuals into this phenotypic spectrum. So here an individual carries the de novo mutation, can be totally fine in terms of phenotypic severity, but in certain cases, this spectrum can be vastly different and can be quite severe. So what determines the severity is a kind of a focus of our research. And I postulate that homeostatic plasticity might act as a phenotypic buffer that might constrain the effect of this de novo mutations in autism spectrum disorders. So the model is, how I define this model is a de novo mutation that puts risk to an individual. And in certain occasions, this mutation can overlap with a genomic variation that is basically inherited in individual's genome. And in this, when these two comes together, then homeostatic plasticity might be impaired. So in practice, this homeostatic buffering is basically lost. And this puts individuals with de novo mutations into the higher end of the phenotypic spectrum. So this is the model. And we want to find out if we can discover this variation, this sources of relevant variation in the genetic background of an organism. So this is quite difficult in mammalian systems, or identifying this random variation in human population is statistically very difficult. But we can design an assay in a model organism like Drosophila and look for this variation in the Drosophila genome. So the way that we design this assay is basically using homeostatic plasticity in a neuromuscular junction system. So here you are seeing an example synapse with the presynaptic button and the postsynaptic receptors. This fluctuation shows the receptor activity, and this is the excitation when an action potential innervates this terminal. So we can perturbate the system by using a blocker of receptors, neurotransmitter receptors, which is called phlantotoxin. And you can see here now when we introduce this blocker, the receptor function is vastly diminished. And there's a transient drop in the contraction of the muscle. And as a result of retrograde signaling, now we can restore the activity levels back to the set point, basically by increasing the neurotransmitter release. So this phenomena is called as presynaptic homeostatic plasticity. And we can also quantitatively plot this phenomena in this plot. As you can see here now in the x-axis, we are plotting the receptor function as the magnitude of miniature EPSP amplitudes. And in the y-axis now we are plotting the neurotransmitter release, which is basically boosted up when the receptor function is dropped. So this exponential plot shows the homeostatic upregulation of the neurotransmitter release. And this can be also shown here as a bar graph. When the receptor function is dropped, as a concomitant increase, there is an increase in neurotransmitter release, basically offsets the reduction in the receptor function. So by using this assay, we wanted to test first if individual autism mutations as heterozygous de novo mutations impair this process. And to our surprise, we haven't found any evidence of impairment in homeostatic plasticity. So these 5 major autism gene mutations that were identified in human population had no impairment in achieving homeostatic plasticity. As you can see in all examples, they are all wild type. They are basically restoring back to the baseline levels. So now the question is, so we put forward this question, if there is a genomic variation that contributes to the phenotypic severity, what if we just basically put 2 mutations together as a heterozygous mutation? And what will happen? This is like a simple compound heterozygosity assay in genetics terms. And now what we found is if we put these 2 mutations together, they are functionally totally separate mutations, biochemically totally separate genes, have no previous biochemical or genetic evidence in their interaction. But now when we put them together, now we see a complete failure in homeostatic plasticity. So this basically gave us the idea about finding modifiers of these AST mutations in the Drosophila genome. And to do that, we use Drosophila as a system because it's a tractable system for genetic analysis. Now, what we did is basically we introduced again, heterozygous mutation, which is here exemplified by RIMS, and now placed a large deletion of the genome, which is called as deficiency. When we put this together, you can see here one arm of the chromosome carry 1 mutation, and the other pair of the chromosome has now a deletion. So if there is an interaction, genetic interaction, now we should be able to find any of the gene that is in this deletion should be responsible. So this is what we did, and we basically, by using this assay, we could basically span the whole Drosophila genome. In this case, I'm showing the 1/3 of the genome, which is the 3rd chromosome, and that basically covers almost 6,000 genes. So by using this assay, we found near 20 loci in the 3rd chromosome that basically show this genetic interaction. So this is basically an example of this interaction. Deficiency itself as a heterozygous mutation has no impairment in homeostatic plasticity, whereas when we put this with the autism mutation together, this deficiency now shows an impairment. So this again shows a double heterozygous interaction, and that is how we define this locus in the Drosophila genome. So now the question is, can we identify the gene in these deletions? And when can we basically find out the mechanism that basically cause homeostasis to fail? So following this, we found in 2 deletions, 2 genes. One of them is a protein phosphatase regulatory domain, PP2R5D, and the other is a protein kinase that we identified in our screen. So then we used the individual gene mutations. So in this case, it's not a deletion, but now it's a loss of function mutation that we used, and we recapitulate the phenomena that we observed in the deletion itself. And this is the case as well for the other gene mutation. So now the question is, we found an interaction between an autism gene and a modifier in the Drosophila genome. Is this modifier will modify another autism gene as well? So this was our hypothesis, and we tested several autism genes that I showed at the very beginning, and tried to see if they will also the homeostasis will be also impaired in these conditions. And surprisingly, what we found when we put this modifier mutation on the background of another autism gene mutation, we also found an interaction that impaired homeostatic plasticity. And 4 out of 5 cases and autism mutations that we tested against this modifier, the homeostatic plasticity were impaired. And we did the same thing for the other gene mutation that we found. Again, in 4 out of 5 cases, we found an interaction that was not previously tested. So we focused on one of the interactions from this interactome, which is the interaction between the modifier that we found and PP2R5D and the gene mutation CHD8, which is one of the top ASD risk genes. So we already found this interaction. As you can see here, homeostatic plasticity is impaired, and we wanted to do some further phenotypic analysis. And we did a bunch of assays like intrinsic excitability, synapse anatomy. They were all normal, except the ultrastructural level. We found very interesting observations and phenotypes, which I'm not going in detail right now. But what we found interesting, we looked at the transcriptional profile of these double heterozygous animals, and we found something very interesting that we followed up, which is an upregulation of a gene that is now shown here, which is CREG, that is a repressor of virus-induced expression. And this gene upregulation basically causes homeostatic plasticity to impair, because when we introduce a mutation that reduces the overexpression of this gene mutation, here shown as a mutation, CREG, now we could restore the homeostatic plasticity. So interestingly, it's kind of surprisingly, at the same time when we found this gene upregulation in the Drosophila genome, a paper that was published in basically single cell analysis of the postmortem human brain of individuals with autism, found in 14 out of 7 cases with idiopathic autism, the CREG is a gene that was upregulated as well. So that was a quite surprising finding for us. And that might be relevant for the evolutionary conservation of several genes that are relevant from Drosophila to human. So today I tried to give you a kind of an overview how we see homeostatic plasticity as a protective buffer that constrains the effects of the autism mutations, the de novo mutations, and reduces the phenotypic spectrum, phenotypic severity. With that, I would like to thank to my lab, Ray Davis lab, Rick Feather, Ray Elena Kulik, Julia Zunino, and our collaborators, Stefan Sanders and Juna. Thank you, and please welcome your question.
Thank you, Özgür. This was fascinating. You will be pleased to, we're unfortunately our third panelist has not yet shown up. We've tried to contact him and he's not yet here. So we can open this to questions and feel free to answer more liberally. If others have questions for Adriano that you didn't get a chance to ask, we can also go back and discuss with Adriano. So let me know if there are any questions.
All right.
So here's a question from Tsivi Atchler. Is it possible that some of the scaling across both talks in this session can be done dynamically during recognition using postsynaptic to presynaptic homeostatic feedback? Oh, sorry, and you'd like to answer this question live. You'd like to answer this question live. Zoom room 1 would like, yeah, feel free to answer this question live. Does that make sense, the question? Do you want me to repeat it?
I'm reading, so I have the screen.
Okay, all right.
I think Adriano probably might also, this is probably some scaling question.
Yeah, so hang on, I'm pulling it up now. It's also that some of the scaling can be done dynamically during recognition using postsynaptic to presynaptic homeostatic feedback. Yeah, that is possible, but one thing that is required for scaling is a global signal that gets around the cell. If you only have it, if you only have one postsynaptic and presynaptic link, you don't know what all the other synapses are doing. So you don't know how much to scale. So I think it is possible to do it dynamically, but you still need a signal with the rest of the neuron that is doing the scaling. I hope that makes sense, and I hope that answers your question.
Are there other questions? So as I was listening to your talks, I was wondering, so I'm a developmental neuroscientist with human children. And I was wondering across your thoughts, across your talks, whether you observe differences across development. So are these scaling properties or these flexible scaling properties, do you see developmental changes, I guess, throughout? I know that you're modeling some of these and maybe you don't have any data to speak to that, but maybe other research speaks to developmental changes in these properties.
That's a really good question. So one thing that we keep constant in our models is the morphology. Now, we have had some thoughts, and this is kind of a side project that we've started working on, is what happens if that morphology is not constant? What happens if you have a neuron that grows, or dendrites that get bigger or smaller or link together or break apart? And that kind of answer, that kind of leads us into the development question, because we know as these cells grow, for instance, pyramidal cells, they obviously start off small, and over the course of like 3-4 weeks, in rats at least, they develop to almost full maturity. So it is a more complicated control problem. There's resources from the cell that are going to scaling in addition to growth and development. So I think it's still a bit of an open question from our perspective, from the balance of accuracy, efficiency, and stability.
Yeah, right. That makes sense. And I guess on the other end of the spectrum, you could see atrophy in aging as well, which may or may not mirror some of these morphological changes in development.
Right.
Interesting. Are there other questions from the panelists? This is your chance, since we have a little bit of extra time, to sort of hash out things. So June, regarding the comment to Adriano's previous answer, does that mean the homeostatic feedback via scaling would affect the whole network, since the neuron being measured could have synapses with multiple neurons?
Yeah, it does affect the whole network. Now, the cell's job from a homeostatic perspective is to make its intrinsic excitability the same. In doing that, it's going to have some synapses get weaker, some synapses get stronger. So it does kind of ripple out into the rest of the network. We believe that it kind of diffuses, like it kind of creates noise throughout the network that kind of diffuses out. We haven't tested this in simulations. We're kind of focused on the single neuron model. So I'm not an expert on how it affects the rest of the neurons in the network, but it definitely does have an effect because it is changing the synapses.
All right, maybe any, yes, so you have questions for each other's talks? Or things that you wanted to add, or?
Yeah, maybe answering maybe part of your question about the developmental aspects of synaptic homeostasis. So I think this phenomena that we are trying to describe, that we are trying to be really careful about whether this phenomena is at some part of the developmental process or it's all lifetime. And we have now evidence that in adult brain of mammalian system in the brain, we observe this phenomenon quite robustly. So although it's quite relevant for the developmental process, because what I basically just proposed here, when the lack of homeostasis, then the system cannot cope with the genetic or environmental insults properly, and that basically can add up, and then the whole developmental process is kind of impaired. But the same thing can happen quite late in the adulthood. We can even think about if the process that degenerates neuron, or that's like stroke or any kind of traumatic brain injury, can induce a transient reduction in the brain activity. But this is again compensated by homeostatic processes, and then individuals can restore back their capacity. And these are all due to homeostatic mechanism. This includes both scaling kind of homeostasis that Adriana just explained the basis, and also now we have evidence that this is presynaptic. It can be also engaged.
Fascinating. Thank you. So this question, I guess you were reading it, Özgür. It's difficult to isolate a cell from another, so it's hard to determine effects of single cells versus circuit, even if you patch clamp. How do you assure effects are due to a single cell or some sort of circuit interaction? Great question.
That's a good question. So the question is, how can we define the so how can we first of all define the perturbation? Which cell is going to receive the perturbation, and or whether it's multiple cells together are receiving the perturbation, whether it's and then if the homeostatic response is coming from single cell or the circuit level, then how are we going to determine this? So it's not easy to determine which cell or which cell groups are contributing, but we can, depending on which kind of assay we are using, if you are looking at, let's say, firing rate homeostasis, let's say a neuron's firing rate. So by simply looking from the firing rate of the somatic recordings for the individual neuron, we can state that, okay, this is a firing rate homeostasis that comes from this particular neuron when it experiences kind of a perturbation genetically. Whereas if it's a pharmacological perturbation that basically perturbs multiple neurons at a time, if it's not a genetic perturbation, then it's possible that multiple neurons contributes, and we need to know at the end whether our assay is detecting an activity, let's say a postsynaptic receptor activity from a particular neuron or not. So I think this is depending on the type of the experiment that we are trying to do. And so again, homeostatic field is quite large. So there are different assays that we use, but we are trying to do this in, let's say, in neuromuscular junction. We have one major synapse, and we know which inputs are giving us the homeostatic feedback and output. Whereas in the more complex circuits like cortex, this is quite difficult. So more cell-specific assays are necessary, like optogenetics or cell-specific manipulation.
Thank you for that question. I don't know, Adriano, if you wanted to speak to that question as well.
Sorry, I had myself on mute. Unfortunately, I don't have much experience with the synaptic scaling wet lab experiments, so I probably don't have anything intelligible to add.
Great, thank you. Are there any other open questions, pressing? Okay. Well, we had a significant amount of time, and I certainly learned a lot from both of you, and I appreciate the data that you've shared. It's fascinating. I'll be on the lookout for more of it. Thank you both, and thank you all of you who attended. I know it's late for some of you and middle of the night for others, and hopefully we will be able to find a way to post the missing presentation up either in slide format or Zoom recording or something like that. I'm not sure whether that's possible, but hopefully we'll make it work. So thank you very much. I look forward to seeing more of your work. Enjoy the rest of your conference. Have a good evening.
Thank you.
Thanks so much. Thank you, everybody. Bye.
Good night, wherever you are. So here we have three really exciting talks coming up. It's going to be a very diverse set of talks. So the talks are going to be spanning three different themes through NeuroMesh. And the speakers, as in all the traditional talks, will have 12 minutes to introduce the topic, and then we have 3 minutes for discussion and questions. So as with all the other talks, you can post your questions in the Q&A and you can post any other discussion in the chat as well, so please make use of the chat. You can also vote in the Q&A for your favorite questions, and I'll relay the questions back to the speakers. Please keep in mind that we adhere to the Neuroscience Code of Conduct, and please be respectful in your interactions throughout the session, in your questions, and in your chat contributions. And also, I want to stress that there is no dumb, silly, or stupid questions. So please participate as much as you like. So now, without any further ado, I'm gonna hand it to our first speaker, Mridula from the National Institute of Mental Health and Neuroscience in Bangalore, India. So I'm gonna stop sharing my screen and then hand it over to Mridula.
Thank you. Yeah, can you see my screen?
Yes, I can see your PowerPoint.
All right.
Yep, perfect.
Here we go. Fine. So I'll just hide this for a minute so that it will be more visible. Yeah, first of all, I would like to thank the Neuromath conference organizers for giving me this wonderful opportunity for presenting my pilot study, which is a part of my PhD study. So I'm working on adult ADHD. The alcohol addiction is also having other comorbidities like substance abuse. So one of the important and the most deliberating one is the adult ADHD. So ADHD is highly heritable and the most common neurodevelopmental disorder in childhood, which passes into the adolescence and adulthood, which we call it as adult ADHD. So in very young children, it's predominantly hyperactive impulsive type. Middle childhood, it's predominantly inattentive type, whereas in late adolescence and adulthood, there will be a combined trait with inattention, emotional lability, and decline in hyperactivity. So in children with ADHD, 15% continue to meet the criteria for adult ADHD by the age of 25 years, whereas 50% meet the criteria of adult ADHD in partial remission with subthreshold symptoms causing the impairment. In the patients with early onset alcohol dependency disorder, the adult ADHD is seen as an endophenotype. So we know alcohol is the most commonly used mood-altering drug worldwide, and alcohol use disorder is a complex and heterogeneous disease which consists of alcohol dependency and abuse. So the alcohol use disorder is mainly of two basic phenotypes. We call it as early onset and late onset. So in India, there is a higher prevalence of adult ADHD in patients with early onset alcohol use disorder. So I was like interested to work on this. And the prevalence of adult ADHD with substance abuse or disorder is around 20 to 50%. So what are the main effects that alcohol and is having on the body? So when we look into the cardiac autonomic dysfunction, so alcohol has a compared to the molecular and also the physiologically, it's very much causing damage to the cardiac system, the cardiac autonomic dysfunction. And in our lab, they also looked into the offsprings who are exposed to this alcohol addiction in the parents, also developed cardiac abnormality as an endophenotype. So they published some of the papers in our lab, which also talks about nearly 26% of the study population with alcohol dependency have abnormal cardiovascular reflexes. So coming to the other abnormality associated with alcohol is cognitive dysfunction. We know the frontocerebellar circuitry is the one which is majorly affected, causes the dysfunction in the alcohol use disorder. So let it be in children or in adults or adolescents, this autonomic dysfunction, dysregulation is very much the core symptom of inattention. So with the basic intro, I would like to move on to what methodology would be liking to work on this. First, we will be going in a stepwise manner, like we will be taking the socio-demographic details, alcohol timeline, follow back assessment, ADHD RS4 with adult prompts, and quality of life enjoyment satisfaction scales. And basically the study groups are divided into three: patients with alcohol use disorder and adult ADHD. Group 2 will be the patients with alcohol use disorder per se, and 3 will be the group matched healthy volunteers for the general population. And study design is a cross-sectional study with the study population. Patients will be recruited from the de-addiction department of NIMHANS, and healthy controls will be my friends and public from the general population. So the work goes like this: like day 1 will be patients admitted to the de-addiction center, 1 to 7 will be on benzodiazepine treatment, 8 to 13 will be the washout for benzodiazepine, 14 will be evaluation of cardiac autonomic functions, and day 15 will be assessed with different psychological assessments, like we'll be doing the color trials and Stroop task, and NIMHANS has developed a neuropsychology battery of tests for both alcohol addiction as well as with adult ADHD. And we will be subjecting them to transcranial magnetic stimulation, that's the paid pulse. We'll be assessing the SSRI, that is what was the excitatory and inhibitory network that was there in the brain, also we will look. And finally we will correlate all the parameters and we'll get a comprehensive idea about how the addicted brain is also affected with the adult ADHD. So very briefly, I would like to go to the physiological significance of the time domain measures. The time domain measures we are assessing here in heart rate variability or STNN, which will give me an idea about the different sources of variation like respiratory, baroreceptor, thermoregulation and activity. Then SDANN index like will be assessing basically the heart rate, how it diffused in the 5-minute segment, and RMSST, the root mean square of difference of successive RR intervals, and finally the PNN 50. This will give me like how the heart rate variability is varying between the subjects, like comparatively between the three groups, and then we will have an assessment like how the heart is responding, or the heart rate variability is varying between the three groups and which group is the most affected. Coming to the physiological significance of frequency domain measures, this is also a part of the heart rate variability, and here we will be using a software for this, that is given by LabChat 7 and 8 software. Will be assessing the heart rate variability in frequency domain. So this will also give me an idea like either there is a sympathetic dominance in the individual or a parasympathetic dominance. So the previous literature tells about there will be like more of parasympathetically mediated reflexes that are more in alcohol. But as of now, we have no clear idea like in a population with alcohol and the comorbid adult ADHD, what was the effect that's mainly like which wing of autonomic system is dominant, either it is a parasympathetic or sympathetic. We have no idea till now, so we'll be dealing with it. So this is a very preliminary pilot study I have done with the subjects of nearly 20. So the very preliminary study I would able to make out the heart rate in a group that is alcohol plus ADHD. It's comparatively more compared to the other two groups. Similarly, the healthy controls are showing better the RMSST values, which is shown, which shows implies that their heart is ready to deal with any other sudden reflexive mechanism compared to the alcohol per se group or the comorbid group. So this is also talking about the heart rate variability in frequency domain spectrum, which tells that the total power, that is the variance, is more for healthy controls compared to the adult ADHD and the alcohol dependence per se groups. So here we significantly found that low power is comparatively less for the comorbid group compared to the healthy control. So this is in our lab. This was one of the subject. So here we were doing the different autonomic function tests for knowing any dysfunction is associated. So this is Valsalva maneuver, the blowing against the closed glottis. And this one is like hand grip test, this hand grip dynamometer to look into the heart rate and BP changes associated with doing a isometric test, hand grip test. This is like orthostatic test. Will be assessing like if there is any hypotension associated with the change in the posture, that is from supine to standing. And this is the tilt table test, which will assess the involuntary hypotension if it is present in this population, the most vulnerable to alcohol autonomic dysfunction associated with alcohol dependency. So one of the other parameter we looked into was the transcranial magnetic stimulation. So here I was only interested to look TMS that's as a diagnostic way, and looked into that is the different excitatory, like inhibitory mechanisms that are associated. And so in TMS, we have taken 3 parameters, that is SSEA, LSEA, and ICF. So SSEA is short interval intracortical inhibition, and LSEA is long interval intracortical inhibition, and ICF is a intracortical facilitation. So these 2 parameters are telling me what was the inhibitory network in these individuals, and this will look into the excitatory network. So finally, what I will be doing is like, first I will do this autonomic function testing, then with the TMS, and correlate and compare it, like what correlation we can draw from the disinhibition that was there in TMS that correlated with the dysautonomic regulation that we got it from the HRV. So another parameter was echocardiography. So in this echocardiography, the literature says like alcoholic patients are most prone for heart abnormalities, which is very much implied in the echo that is done to the heart, the cardiac system. So here with the very small group of subjects, we were able to find a difference in the end systolic volume, which is comparatively high in healthy controls compared to the other groups. Maybe if we can increase the number, we will come to a conclusion like, yes, the alcohol and alcohol plus ADHD could be having a effect on the heart functioning compared to the normal subjects. Next parameter we looked into was the behavioral experiment. This was an online software that was available, which help us in understanding the different behavioral spectrum of the patients as well as the healthy controls. That's a go, no go task. And other one is the stop signal reaction task. So here basically the individual will be asked to press a space bar when he sees instruction on the screen. There's a visual instruction says go, he has to press the space bar. If it says no go, he shouldn't press anything. So this will be assessing like what was the reaction time of the subject. And the other paradigm was the stop signal task. Here initially the subject will be trained to press the right arrow mark whenever they says go. Like either you can customize your own protocol. How I customized this one was, I will be showing 2 different images to the patient. That is, one is an alcoholic bottle that is designed from like normally alcoholic bottle on the screen will design. The other one will be a orange juice bottle, and the other one will be water bottle. So what we will be doing is like intermittently all the 3 stimuli will be displayed on the screen, and whenever an alcoholic bottle was displayed, the patient should control his emotion to click on the space bar. He should not do anything. He just needs to be keep quiet. And if an orange bottle or a water bottle is seen, he has to click. So this will be randomized, and the patient should be very much attentive to focus on the screen. If he presses for the alcoholic bottle, it will assess and give me 2 important values. One will be he is like having that craving for that one image that is associated with the reward. Second will be his inhibitory network was less, like he was unable to stop when he has to stop. So this parameter I will be correlating with that of the inhibitory parameters which I assessed from the TMS, and I just wanted to look like is there any correlation between them. So coming to the summary and conclusion of my preliminary study, patients with alcohol use disorder and adult ADHD showed the trends of moderate to severe cardiac autonomic dysfunction, decreased parasympathetic activity, decreased end systolic volume, decreased reaction times, And so we need to recruit more patients to get more clarity on the effects of the pathologies on cardiac excitability measures by transcranial magnetic stimulation. And does alcohol use disorder and ADHD affect the cardiac autonomic functions and also the reaction times? And the preliminary results suggested that there could be a possible correlation between the cardiac autonomic and neurobehavioral dysfunction between alcohol addiction and adult ADHD compared to the control subjects. So I hope the entire world is suffering with corona, the outbreak. So similar with India as well. So we were not able to recruit patients within time. So hope for the best, and the entire world will enter into peace and the COVID goes off. So we'll do our research further way. So here I go. And this is my institute. I'm very thankful for NIMHAS providing me the institute fellowship for continuing my study. Thank you. Thank you so much.
I'm clapping for all the attendees. Thank you. Thank you for your talk. That looks like a very comprehensive and massive undertaking there. If you have any questions, please post them in the Q&A. I'm also watching the YouTube chat, so if you have any questions there, you can post them in the Q&A, just put them there. While we're giving the audience some time to write them down. I was wondering, as you said a few times, this was a pilot study. What are your next steps? So do you have some grand plans for a follow-up on this?
Yeah, so I have written a grant for getting a fellowship for doing it as a genetical study. So I was mostly interested to look into previously we have done a study which was related to parents who are addicts, like alcohol addicts. Their offsprings at the age of around 8 to 12 years we have taken, and we assessed the cardiac functionality. So to our wonder, we came to know that these kids are also having an autonomic dysfunction and cardiac abnormality at that age group, even though they are not addicted. So there is a very strong genetic makeup that is making them to develop this substance abuse when they become adolescents or adults. So yeah, this once I get my grant, I would be interested to take out the blood sample and do more genetic studies, so that I think it will be useful, like in putting an eye on these kids before they develop any other addiction. That's my plan for next.
Great, thank you. So in the interest of time, we're gonna move on to the next speaker. Thank you again, Rodula. And our next speaker is Patrick Ichipi-Foucault from Delta State University. Do you wanna stop your screen sharing?
All right.
And then, we are swapping the schedule, I see. Patrick is having some audio issues that we're trying to figure out. So we're gonna swap the two remaining talks in the session. So our next talk is gonna be Tom Gorham from Rizzo University.
Okay, thank you very much. Okay. So thank you, everybody, and also, Merdula, thank you for that talk. It was really interesting. So, cheers, thank you, and Patrick. I hope we get the connection issues all worked out for next time. So, but I'll just jump ahead. For my talk, I'm going to be doing an interactive section. So I will be talking for about 5 minutes, just introducing the idea, and then for the remaining time, I'd like to open it up for more of a discussion and a kind of a Q&A with everyone. So I'll be talking today about mind, brain, and education science, or MBE science, and how translational research is improving education. Again, my name is Tom Gorham, and I'm an associate professor at Rishio University in Tokyo, Japan, and I'm an English teacher in the Faculty of Letters. Also, I'm a part-time PhD student at Kyoto University, where I'm studying in the Department of Social Informatics, and I'm focusing on learning analytics. Now, today I'm going to be talking to you about mind, brain, and education science. And what this is this is a transdisciplinary field that combines three disparate fields. So neuroscience, psychology, and education or pedagogy. And so it's as a transdisciplinary field, it's one where there is a back and forth exchange of information from each of the fields. It was started at Harvard Graduate School of Education. The two of the founders were Howard Gardner and Kurt Fisher. Gardner, you may know of as the founder of the multiple intelligences theory, and Dr. Fisher is famous for his dynamic skill theory. And so I did my master's degree here at in this program, in the MBE program, and while I was there, I had the opportunity to be a teaching fellow in Dr. Tracy Tokuhama Espinosa's class on the introduction of MBE, and so I basically from there. I'd like to talk to you a little bit about how this impacts educators mainly. That's kind of the area that I'm coming from with this. And so as educators, we interface with the MBE field in a few different ways. So on the more academic side, we have the International Mind, Brain, and Education Society, or IMBES, and this is their journal. And then on the other side, we have kind of more informal sources of information. So for example, Dr. Ginger Campbell's Brain Science podcast is one that's quite popular with a lot of educators. Beyond that, there's a lot of kind of popular science books that a lot of educators will use to access this. For example, Mary Helen Imerdino Yang and Tracy Tokuhama Espinosa are quite popular. And there's a lot of other ones, for example, David Sosa as well too. Now, I'd like to introduce the group that I'm involved with here. Here in Tokyo or in Japan, I'm a member of the Japan Association for Language Teaching, and in that, I'm a member of the executive board of the Brain SIG, and so this is the special interest group for mind, brain, and education. And with our group, what we do is we have a lot of outreach as far as workshops and conferences. And so recent and upcoming ones we've done are reaching out to do events in India, Brazil, Oman, Philippines, Korea, and we've done dozens of events here in Japan as well. Beyond with these events, these can be targeted both at people in the secondary level and below, so high school, junior high school, elementary school teachers, as well as the post-secondary level, so university teachers too. Beyond that, another thing that we do is our organization we publish a journal that's geared towards educators who are busy and may not have the time to dig deep into kind of formal academic journals. So we do have an academic journal, but we also have this Mind Brain Ed. We call it a think tank. And so these are a few of the issues that we've had. And what it is it's a way to introduce topics connected to psychology and neurosciences, but make them easier to digest for teachers and easier to access for them. And so currently we've got about 1,600 subscribers in over 70 countries, and it's become really quite popular as a way for teachers to find out about this. If you're interested in seeing the work that we do, please visit mindbrained.org, or you can scan the QR code here if you'd like. I could also post it in the chat as well too. Okay. Beyond that, I think that brings me up to my 5 minutes here. So I'd like to open this up to discussions. And so a few questions for discussions. I'm really interested in interfacing with all of you here. I've got kind of two different sides of questions I'd like to pose here. So one would be your experience as a student. I think this is quite interesting about how were you first introduced to your brain when you were a student? So is this something that your teachers talked about when you were in elementary school or junior high school, or didn't you not find out about how it functions until you were older? Do you think that your understanding of how the brain works was sufficient as a student? So, for example, were you taught about how exercise or sleep or things like that can impact your learning? Were you ever taught about myelination and its connection to things like a growth mindset in education? So that's one side that's interesting that I think is kind of your experience as a student. The other side to this would be the second two questions I'd like to open up for discussions are kind of how these two worlds, this mind, brain, and education that's kind of heavily focused on teachers and educators who may not have this strong background in either the psychology or the neuroscience. And for these final two questions, how can computational neuroscience impact educational practices? And secondly, are there ways to connect this community of MBE or mind, brain, and education researchers and practitioners to the Neuromatch Conference and Neuromatch Academy communities for educational outreach? So these are kind of a few ones that I'd like to turn over to you, but I'd be open for talking about anything. So if you'd like to post any of your questions or turn on your mic too, I'd be happy to open for that. Okay. Thank you.
Thank you, Tom. I don't think there's an option for attendees to turn on the mic, but please
I see.
Your questions and comments in chat or in the YouTube chat, and we'll look at them. You're free to leave your slides up as well, I think, with the discussion questions. And then
okay, yeah.
That might stimulate the audience. Meanwhile, do you already have ideas in this case, especially to point number 4, I think, which is highly relevant here? Like, do you have any suggestions on how to connect these two?
Right. Yeah, no, that's a great question. Thank you. So, and it's Tal, right? I want to make sure I'm pronouncing your name right. Okay, great. Thanks, Tal. So, yeah, no, that's a really good question. And so, I actually signed up this past summer as a participant for the Neuromatch Academy. And one thing that I found is it's interesting for, I think, the design of that. Well, first, making all of the materials open is really wonderful, I think, and so that's I think as a teacher, anytime that there's open and free resources, my ears perk up, and a lot of teachers do too. It's like, oh, how can we do this? Or how can we use this? And so I think that's quite nice. The timing is a tricky one. I think that kind of the intensive summer courses can be a little bit challenging, and so I wonder if there might be some ways of looking at the content that's available from Neuromatch Academy and seeing are there ways to kind of break it up and kind of target it more specifically for educators, or for maybe not even just educators, but for people who have less experience with the computational neuroscience or the kind of the computer science background, perhaps. So.
Yeah, that's and one of the things you said struck me, like the making it openly available. To me, that might be one of the ways that we can really impact things, right? There's in my mind, there's a lot of time wasted by everybody preparing their own course on basically the same topics or very similar topics. So if a lot of this content was made publicly available, that would save a lot of people a lot of time, I think.
Definitely.
We have a question in the chat, which is not a silly question. There's no such thing. But yes, we swapped the last 2 talks in this session because one of the speakers is having audio issues. So Tom's talk did start earlier than planned, according to the schedule.
So, yeah. Oh, is that another question that's popped up? Okay. Okay. Oh, sorry. Well, actually, I see is it Vinicius maybe? Who said, sorry, you missed it. No problem if you missed it. It was actually really quite short. It was just 5 minutes. So the kind of the meat and potatoes of this is the kind of the back and forth or the interaction on this for this session. So just to give you a quick overview, it's I'm coming at this field of neuroscience from the perspective as an educator and looking at how knowledge and understanding about where kind of the science of where neuroscience can interface with education and what it can. Are there things that we can pull from the lab to influence the way that we make pedagogical decisions? And so one of the things that I found and my colleagues have found is that there's a real hunger among educators to find out more about what we're learning in the laboratory about the brain. And I think what is interesting is that education has been going on for, as long as human history. And so there's a lot of kind of intuitive choices that have been made. And so it's been refined through practice, and that being able to find kind of these correlates in neuroscience about why certain practices work. So for example, looking at kind of like the affective neuroscience and kind of what are we learning about how kind of like the social neuroscience or affective neuroscience, and how we can leverage those insights on making decisions in the classroom. And so that's one of the things that really kind of gets me excited about being able to connect with this community here, is being able to see that if we can find more and more ways that we can connect back to this kind of cutting edge science that's happening, and see if we can have kind of a back and forth kind of interaction between the two communities.
Can I jump in with a question from the audience? There's a question from Audrey. How can you make neuroscience accessible to young learners when the content can be challenging to understand? The example is that they didn't learn about the brain until college, but think it would be helpful to approach certain aspects about how the brain works earlier. That is, how and when is the best way to learn, and how do drugs interact with the brain and body?
Yeah, that's fantastic. Excellent question. Yeah, because it does kind of feel as though, like neuroscience is intimidating, I think. And that you say like, well, should we do it for high school students? Should we do it for junior high school students? Should we do it for even younger? And what I found is that, or what we found, our colleagues have found, is that even with quite young students, for example, if you show there's a great GIF or GIF, depending on how you pronounce it, an animated GIF or GIF from the Wikipedia page on myelination. And it has two neurons side by side that are rotating, and one's fully myelinated, the other one isn't, and it's animated to show the signal passing through. And it's really clear for even young people to say like, oh, wow, the signal moves faster if there's insulation on this. I get it. How can we encourage myelination? Well, through things like engagement and practice, through repetition, through studying, through working on whatever you're working on. You can increase the efficiency and effectiveness of these networks in your brain. And even young kids can say, oh, I get it. And if you talk about kind of what do inefficiencies in these networks translate to, it's a higher caloric cost for processing. And you say, well, oh, that's why if I am starting something new, I get tired while I'm studying it. So I think that that's just a simple example, but I think there's ways that you can approach these ideas to really spark young learners' imagination about it. I hope that answers your question. I know we're kind of coming close to the end of our time here.
Could you, there's another question that I think follows onto that really nicely, is could you give a couple of examples of insights from basic neuroscience that are useful for education?
Oh, fantastic. Yeah, great question. I'd say like one would be looking at the science of sleep. And so if you talk about this idea of cerebrospinal fluid being flushed through the brain while you're sleeping to remove kind of I guess the leftover kind of like how would you say it? I guess basically like cleaning out your brain while you're sleeping, that being able to look at that on one side, and then the other side, the idea of consolidating long-term memory formation while you're sleeping, that kind of this idea of hygiene in your brain as well as consolidation of memories. These are things that I think if you explain it to both educators and to teachers, I know I've met a lot of teachers, particularly who teach junior high school age students, who get angry if their students are sleepy in class. And I think changing it and being more sensitive to things like chronobiology and these kind of looking at making junior high school students or adolescents come in early to class when their chronobiology actually has shifted so that they tend to be more active later in the day and less active in the morning. And that I think that there's things like that I think we could make changes in the way that kind of the schools are structured, but also even just kind of things about being more empathetic about how we react to students who may be tired in class. I hope that makes sense. Sorry, that was a little bit all over the place, but.
Well, I think, yeah, no, that's good. So before we move to the next question, I have the unfortunate news that we have to skip the final talk. We have to forego the final talk of this session because a speaker is having audio issues and we can't.
Oh no, that's too bad.
But it gives us more time for discussion here because it seems like there's a bunch of good questions coming in. So I'll have another question for Tom from the audience. It's more maybe more of a comment than a question there, where it would be nice to have tools in different languages and not only in English, especially if you want to impact in high school or in lower grades, as not all the teachers and of course students around the world speak English. So do you have something to add to that? And do you maybe have examples of resources that you can point to?
Right. Yeah. No, that's a fantastic point. Yeah. That and I think that right off the top of my head, I guess I'll say two things. One would be I know that Dr. Tokuhama Espinosa has a lot of good bilingual content both in English and Spanish, and I can add I can add that to the chat here if you're interested in Spanish content. The other thing, and I'll put this back. Let's see. With this content here, in the Mind Brain Ed think tanks, this is all English-based writing, but it's designed to be. All of the articles are quite short, and it's designed to be simplified kind of to the point where lay people can read and understand, so that this might, although it's not multilingual out of the box, it may work well with translation software. So that's one possibility. Also, the way that these think tanks work is that they always begin at the outset with kind of a surface level introduction to the topic and then a deep dive. And so they often have things like YouTube clips. And so those ones, a lot of them will have multiple translations for the subtitles in there. So that this, I know it doesn't exactly answer all of the questions in there directly, but it might give you kind of a stepping stone for accessible content that may be more easily translatable.
Fantastic. Thanks for your great answers, and thanks to the audience for a great discussion here. Thanks for
great
shooting up your questions. And with that, I would like for you to join me in thanking the speakers of today again, which a great session here. So it's unfortunate that we had to miss the final talk.
Sorry, Patrick. I was looking forward to seeing yours. So sorry to hear that.
And I think there is now a couple of hours of a break in the schedule, and it looks that the